wishing you a morning filled with joy, kindness, and endless possibility. I welcome you all to the third Pharma Summit Drug Discovery and Community Trial Conference organized by APR, Association of Hello everyone, I am Dayu and I want to tell you about Biolix. Biolix is a non-profitable professional association which prominently promotes research and development. Biolix conference bring together the professional wizards and leaders who have explored all avenues to research the fields of life science and medicine technology. Bilex conduct events worldwide which help in enhancing the skill set of the people from diverse industry and also from a common platform for eminent personalities, physicians, researchers, doctors, and academicians, professionals, business figures, and much more. Biolage conference encourage better comprehension about improvement and progression over the world through worldwide conferences with the speed of science and technology. Motto of creating a better tomorrow by organizing conferences and creating a network which will help grow a better tomorrow with the help of advanced technology and achieve. And let's talk about the conference. This conference aims to bring together leading academic scientists, researchers, and scholars to exchange and share their works and research results on all aspects in pharmacology, pharmaceutical biotechnology, pharmacogenomic, drug delivery, biodrugs, pharmacovigilance, drug safety, pharmaceutical microbiology, pharmaceutical research and development, pharmaceutical analysis and quality corrective, biotherapeutic, radiopharmaceutical, vaccine design, formulation technologies, clinical pharmacy, industry pharmacy, pharmaceutical chemistry and pharmaceutics, and that's all. The drugs discovery industry has become such a competitive market that it continually faces a challenge to find better drug discovery technologies. This industry must discover and develop innovative drugs for a wide range of diseases and in a marketplace that is likely to experience growing regulatory challenges, pricing pressure, and various other bottlenecks. Pharma Summit will be welcoming brainstorming research papers from across the world in the field of pharmaceutical science and drug discovery. I would like to welcome all honorable dignitary and eminent keynote speaker for this event. The first keynote is Dr. Ramon Kumarajput, former Vice President Candila Pharma Limited, Ahmedabad, India. And then Prof. Murali Yalapu from the University of Texas, Rio Grande Valley, Sioux Falls, South Dakota, United States. Mr. Girish Nihalani, Teva Pharmaceutical in USA, Dr. Gelgel Wirasuta, Department of Pharmacy, Faculty Mathematics and Basic Science in Udayana University, Indonesia, Dr. Paraka S. Godanapar, Professor and Vice Principal, Department of Pharmaceutics, Sri Adi Hunchanagiri College and of Pharmacy in India, Dr. Kartik Anatharaman, Vice President of International Business Apollo Hospital in India, Professor Dr. S. Duraival, Principal, Guru Nanak College for Pharmacy, Dehradun Uttarnath, India, Dr. M. Sunita Reddy, Principal, Center of Pharmaceutical Science, ZNTUH University College.
Now I would like to welcome our conference session chairs. Dr. Dilip Kumar Bharati, Professor and Principal, Pharmacology, JNCP Bhopal, India. Dr. Saranya Punyakoti, Professor, Savita College of Pharmacy, Savita Institute of Medical and Technical Sciences, India. Dr. M. Padma Parakash, Principal, the Oxford College of Pharmacy, Bangalore, India. Now, thank you for joining us today in imparting expertise with us. A warm welcome to all the esteemed participants who have joined us for this conference. We are honored to have such a diverse group of thinkers, leaders, and innovators sharing their insights and experiences. Together, we will explore new ideas, forge connections, and shape the future of our field. Thank you for being part of this important event. I would like to welcome all the attendees in joining us today. Once again, welcome you all to this third Pharma Summit conference. Now I call upon Professor Dr. Nala E. Ismail, Dean, Faculty of Pharmacy, AIMST University, Malaysia, to give the inaugural message. Ma'am, over to you, please.
Okay, thank you so much, Chairperson. Am I audible to everyone now?
Yes, ma'am, you're pretty audible. Thank you.
All right. Okay. Distinguished guests, speakers, delegates, organizing committee members, ladies and gentlemen. Assalamualaikum tuan-puan, namaste, waalaikum, kamusta, selamat pagi bapak-bapak, ibu-ibu, and good morning. It is an honor to welcome all of you to our third Pharma Summit, Drug Discovery and Community Trial. For year 2024, 27th and 28th of March, commencing on hybrid mode all the way from Bali, Indonesia. I recognize that this annual conference is principally designed to enhance the sustainable development of research in pharmaceutical science and drug discovery, in tandem with this year's Essence theme, Unlocking the Technologies in Pharmaceutical Science and Drug Discovery. Pharmaceutical industry has a significant role in helping patients to get better through their medication. It helps in discovering and developing new drugs which save many lives. Many novel technologies and emerging trends are going to cause a great impact to the pharmaceutical industry. The pharmaceutical sector has to embrace these new technologies in order to provide a better future in medical evolution, including digital and artificial intelligence in health. Therefore, our excellent program in these 2 days will be delivered via a faculty of prominent, enthusiastic, high-caliber speakers presentation from highly recognized organizations, including from the academic institution, Udayana University Indonesia, University Technology Mara, UiTM Malaysia, Acropolis Institutions India, the University of Texas, Rio Grande Valley, South Dakota, USA, Sri Adi Chunchanagiri College of Pharmacy, India, Guru Nanak College of Pharmacy, India, JNTUH University College of Pharmaceutical Sciences, India, Adi Chunchanagiri University, India, and Chettinad School of Pharmaceutical Sciences. Not to forget, from the healthcare and pharma industry, we have speakers from Cadila Pharmaceuticals Limited, India, Apollo Hospitals, India, Teva Pharmaceuticals Incorporation, USA, Middle East Organon, UAE, Altia Global, UAE, Janssen from the area of Europe, Africa, and Middle East, also UAE, WHO, World Health Organization Center for Biomedical Research Population, New York, and many, many more. The commitment of being professionally excellent, keeping up to date, and continuously seeking improvement is the key of optimizing a professional's career's opportunities and development. Continuing professional development program and activities are the way to improve professional competence and sustaining it. We need to be intellectually alert, have appropriate knowledge base, as well as being accomplished in the necessary coping mechanism and capable of translating science and innovation into practice to close the gap between evidence and practice and to improve indispensable performance for the benefit of our end user, the customers, our patients. This virtual hybrid gathering enables the building of a productive dialogue between the participants and the representative of various pivotal stakeholders. It is also provides an invaluable opportunity for networking and fruitful contacts between respective delegates and participants of this conference. Worth to mention, the proceedings of this conference will be submitted to the Web of Science Book Citation Index, BKCI, and to Scopus for evaluation and indexing. I would like to express my gratitude for your commitment, your seeking excellence in your practice, and your interest in attending these informative, valuable, thought-provoking sessions catered by this conference. I am confident that all of you will enjoy the admirable theme and leave the conference with excavate knowledge that you will be able to apply at your own current and future practice. I wish to acknowledge all delegates and observers for their full cooperation and contribution to the third Pharma Summit Drug Discovery and Community Trial. I would like to take this opportunity to thank Association of Pharmaceutical Research APR, scientific partner BioLix, research partner United Innovators, and various generous sponsors for providing the necessary funding and kind assistance. Appreciation goes to the organizing committee members for their diligence and untiring support and effort to ensure the glory of this summit. I wish all the participants a very fruitful and productive sessions. Terima kasih semuanya. Thank you. Bahut syukriya. Mika Nandri.
All right, thank you so much, Dr. Dilip Kumar Bharati. And our next session chair is Dr. Saranaya Punio Yakoti, Professor Savita College of Pharmacy, Savita Institute of Medical and Technical Science, India. The time is yours.
Now I would like to welcome our keynote speaker of today, Dr. Pramod Kumar Rajput, former Senior Vice President, Cadila Pharma Limited, Ahmedabad, India. Sir, over to you, please.
Thank you so much. Good morning, good noon, Good evening and Assalamu alaikum, ladies and gentlemen. Conference Chair Dr. Nala Ismail, Professor Engine, Bursar and Council Member, Academy of Pharmacy, from the Department of the Clinical Pharmacy, Practice and Pharmaceuticals, amidst Malaysia, my fellow speakers and the faculties, esteemed members of the Association of the Pharmaceutical Research, distinguished guests and fellow researchers. It is indeed an honor to be before you today through an online platform as we gather here on the hybrid mode for the third Pharma Summit, that is on the drug discovery and community trial, being organized by such a wonderful company, BioLegs, along with the APR. This prestigious conference, as we heard from the chairperson, is going to start from today, that is on 27th, and will continue till tomorrow, 28th, in this beautiful city of Bali, Indonesia, and also on the online, because we all have joined you on the online mode, which aims to bring the brightest mind in the field of pharmacology, which is the need of the hour. As we heard with the chairperson, that our primary objective for this summit is to create a platform where leading academics, scientists, researchers, and scholars can exchange ideas and share their groundbreaking work and research results. Because this diverse range of the topics covered in this entire 2 days of the conference reflects our sheer commitment to explore all aspects of pharmacology, which is the real need of the hour. From the latest development in the pharmaceutical biotechnology to the existing world of pharmacogenomics, drug delivery, and the biodrugs, this wonderful summit of 2 days will provide a comprehensive overview of the cutting edge advancement in our basic fields. Moreover, we will dwell into the critical areas of pharmacovigilance and the drug safety, which is very important, pharmaceutical microbiology, and the crucial aspects of the pharmaceutical research and the development, which is again a part and parcel of this conference. We also aim to shed light on the essential disciplines of the pharmaceutical analysis and quality curatives, biotherapeutics, and the vaccine design, formulation technologies, clinical pharmacy, industrial pharmacy, and integrate world of the pharmaceutical chemistry and pharmaceutics will also be explored in depth, which I'm sure we have such a learned speakers are there. We are going to make a lot of change and the people are going to understand, listen, and resonate it. Because as we gather, a lot more intellectuals from across the globe are going to share their valuable knowledge. Let us embrace the spirit of collaboration to learn and update the real need of the hour. Together, we can push the boundaries what is possible in the field of pharmacology by fostering an environment of open dialogue and intellectual curiosity. We have the power to shape the future of the medicine, which is again really needed. In addition to the comprehensive range of the topics, I want to emphasize the significance of the collaboration and innovation in the field of pharmacology, because it is the real need of the hour. As researchers and the scientists, we are at the forefront of discovering new treatments, improving drug delivery methods, and enhancing the patient care. However, we cannot achieve this advancement in isolation. That is the reason that the need of the collaboration. So by collaborating with the colleagues from the different disciplines and the background, we can leverage diverse perspective and expertise to tackle the complex challenges which we face. This Pharma Summit provides an excellent opportunity to foster these collaborations, enabling all of us to develop the groundbreaking solution that can transform the lives of the patients worldwide. Moreover, it is highly crucial to acknowledge the role of the technology in this accelerating drug discovery and development. In recent years, advancement in the field, such as artificial intelligence, machine learning, and data analytics, have revolutionized the way we approach the pharmaceutical research. By harnessing the power of these technologies, we can analyze the vast amount of the data, identify the patterns, and make informed decisions with greater precision and efficiency, which is the real need of the hour now. During this conference, we will also explore the latest innovation application of the technology in drug discovery, formulation, and the clinical trials. By embracing these achievements and advancements, we can expedite the development of safe and effective therapeutics, ultimately improving the patient's outcome. As we delve into the intricacies of pharmacology and drug development, it is imperative that we address the ethical consideration and prioritize the patient safety, because ultimately it is the patient safety which makes a difference. So research and development in the pharmaceutical come with a great responsibility. It is our sheer duty and responsibility to ensure that the drugs we develop are safe, effective, and meet the highest standard of the quality, which is again the real need of the hour. Throughout this wonderful conference, we will discuss the essential aspects of the pharmacovigilance and the drug safety as well, emphasizing the need of the robust regulatory framework and the rigorous clinical trials, which is going to help for the patient safety part. By adhering to the stringent ethical guidelines and maintaining transparency, we can build trust among the patient, healthcare professionals, and the regulatory bodies all, because we are confined to all the three segments, which is very important. Finally, let us take a moment to celebrate the achievements of the researchers and the scientists who have made significant contribution to the field of pharmacology. A lot of development had happened. So my salutations to all of them. Their dedicated work, hard work, and the groundbreaking discoveries have paved the way for the advancement in the healthcare and improved patient outcomes. Further, let us inspire and encourage the next generation of the researchers which are listening, which are going to listen, which are participating here in this Pharma Summit, because it provides a platform for the young scientists and the scholars to showcase their work, exchange ideas, and learn from such established and esteemed experts in the field, which are here as one of the speakers here. So by nurturing and supporting these emerging talents, we can ensure a bright future for pharmacological research. I urge each and every one of us to seize this great opportunity to engage with our peers, learn from their experiences, and forge new connections. The future of the pharmacology lies in our collective effort. That's why I said a collaboration. And by uniting our knowledge and expertise, we can make a meaningful contribution that will impact the life of countless individuals around the world. In the conclusion remarks, I will say, let us embrace the opportunity presented by the third Pharma Summit, Drug Discovery and Clinical Trials. Together, we all can push the boundaries of pharmacology through collaboration, innovation, and the responsible pursuit of the knowledge. As we gather in the Bali and also in the hybrid mode in different parts of the world, let us be inspired and fuel our passion for improving the global health. Thank you once again to the Association of Pharmaceutical Research and Felix for organizing this extraordinary event. I extend my heartfelt gratitude to my fellow speakers and the faculties and my best wishes to all the participants. And I hope this wonderful summit will make a resounding success, leaving a lasting impact on the field of pharmacology. Let us make the most of our time together for forging new connections, sharing knowledge, and embarking on the journey towards a brighter future. Thank you and wish you all a very productive and enlightening 2 days wonderful conference which have already started. Thank you so much once again.
Thank you so much, Dr. Pramod Kumarajput, for your informative presentation. Let's move to next speaker, Prof. Murali Yalapu from the University of Texas Rio Grande Valley, Sioux Falls, South Dakota, United States.
Good morning, everyone. Can I share my slides? Okay. So good morning, everyone. Can you able to see my slides?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Oh, wonderful. So good morning, everyone. So again, I'm an associate professor and also co-director for the Department of Immunology and Biology and the program coordinator for South Texas Center of Excellence in Cancer Research, School of Medicine, the University of Texas Rio Grande Valley, Texas in the United States. The existing ones. Hello, can you hear me?
Sir, your PPT, we can hear you, but your PPT is not coming, sir.
Okay, so I think something. Okay.
Yeah,
something-
earlier it was coming, sir, but now it's not coming.
Okay. Okay. So maybe I will reshare it
Yeah,
again.
correct, sir.
Okay. So share.
Yes, sir.
How about
Visible.
good now?
Visible, sir. Visible, sir. Visible.
Okay. Good. So without further ado, my topic is on nanopharmaceuticals, how these nanopharmaceuticals are different for regular pharmaceutics. Okay. So I will be giving some of the basics of nanotechnology, and then I will be introducing some of the FDA approved formulations, and then I'm going to introduce what we do at the ETRGV Texas in the United States. Okay. So when we talk about nanopharmaceuticals, we want to uh, provide the rationale why we want to use nanopharmaceuticals for cancer therapeutic applications. So here the slide is showing you the better statistics in each year, how many new cases of cancer has been diagnosed at the United States, and also how many people have been died with this particular disease. You can see 1.8 million people diagnosed each year, and then at the same time, 600,000 people would die because of the cancer. So when we talk about nanomedicine or nanoparticles that are contained drug molecules, you can imagine, how these pharmaceutics are different than the regular one. So these are nanoparticles. They have very tiny in size. You can see the clearly 1 nanometer to 100 nanometer. These are such a tiny particles. In these tiny particles, you will be encapsulating your drug molecules or therapeutic agents. It could be protein or small molecules or any nucleotide or any other biomolecules. But those are you either encapsulate or you formulate with those. So therefore, you can achieve a better therapeutic delivery. So that's what the concept is. But when we talk about a delivery, first you need to identify what are the issues that are exist. So there are some issues when you talk about a cancer drug delivery. Number one, it could be a drug low solubility. Next one, it could be a very high potent molecule, but at the same time, it may have lower bioavailability, or maybe it is producing non-linear pharmacokinetics. And also you can also recognize that there are some of the targeting issues, and also some of the cancer cells develop the drug resistance. To overcome some of these issues, are not only some, but majority of these issues that can be overcome by employing nanotechnology. So here, so there are some of the examples here I'm listing, like, from the right from 1 to 10 nanometer, dendrimers, metal nanoparticles or carbon nanotubes, and also like various other polymeric micelles, nanogels, hollow nanoparticles, and so on. Those are lying in between 10 to 200 nanometer, or at the same time 100 to 300 nanometer. So whereas the bulky nanoparticles, those are like, microparticles or microgels. So these are all like, it can be categorized based on the either the nature of this nano material itself, or based on the size itself, right? So whenever we talk about the cancer therapeutics, the nanoparticles all have the inherent passive targeting activity that is called enhanced permeation and retention. So where the most of these particular EPR effect has been explored widely in the nanopharmaceutical fields, where you can see majority of the FDA approved formulations are explored by this mechanism. And also there are so many new ways of nanomaterials that are being delivered to the cancer cells. Those are primarily by targeting active way rather than passive manner. So these active targeting manner, it could be a ligand putting a ligand or any decoration on these nanoparticles. So those will be directed specifically at the tumor site. So at the bottom of the slides, you can clearly recognize how the passive targeting is happening, where you are all very tiny nanoparticles, they are in the circulation, blood circulation, and they are finding the leaky vasculature at the tumor site. Therefore, they are going to go inside the tumors and then they are locking up. That's why it's called like enhanced permeation you will be achieving, and also it is remain or retained at the tumor site. Therefore, you call it as a EPR mechanism that is nothing but enhanced permeation and retention effect. So whereas the active targeting that will take the advantage of the active, I think I can hear some background. Can you mute everyone, please? So all these nanoparticles, they will be on the surface of the nanoparticle, they have an active moiety that can specifically recognize the tumor cells. It could be a folate receptor or by using a folate, or maybe HER2 antibody to recognize the HER2 protein and the cancer cells and so on. So you have these two basic mechanisms that are highly explored for cancer therapeutics. So when you talk about the nanomaterial, we're all imagining the nanoparticles are very spherical in nature, but that may not be true. In general, they can be in a different, structurally different, or it could be a rod shape or spherical nano rods, and you name it. There are so many wide variety of those, but majority of the nanomaterials so far we discussed are in the literature are based on the spherical particles. And also you can see the surface modification can be achieved by putting the hydrophilic or hydrophobic molecule, so that it can improve the biological targeting or the suspension of these nanoparticles, stability of the nanoparticles, and so on, by modifying these surfaces. And also the nanoparticles, they have either positive, negative, or neutral charges based on the surface coating that you are providing. So why these are important, whether it is a tiny particle, whether it is a hydrophobic in nature, or maybe it's a positive or negatively charged nanoparticle, all they have some of the clinical importance. Because if you see this slide, you can see very smaller particle size, like 1 nanometer. So those nanometer, very tiny particle, they have a tendency, or they have a easily cleared by the liver. So where you have a 10 nanometer nanoparticle, those will be cleared by renal mechanism. So that means kidney. So that means when you're talking about, or when you're creating a nanomaterial for cancer therapeutics, you need to keep in mind that for a drug delivery purpose, the smallest size is not the best. So therefore, you need to create nanomaterials or nano formulation. Those are in the size range of greater than 10 and also less than 220 nanometer in general. That means, so the above about 10 nanometer and then close to 200 nanometer or 100 nanometers are the best choice for those can be easily obey the EPR effect. So that's what we need for cancer drug delivery. So at the same time, you see on the top of the chart, right hand top of the chart, you will see the EP. If you have a lower solubility, those nanomaterials can be recognized by the macrophages. It can be taken up the macrophages and then cleared easily from the body, right? And also you can see on the right hand side of the slide, you can see if you have a highly positive charge nanoparticles, so those will be toxic for the majority of the cells in the body. So for example, somebody is administering nanoparticles, those are highly positively charged in nature. So whenever you inject these nanoparticles into the body, they meet first with the RBCs. So those RBCs can be interacted with the nanoparticles. Because of this highly positive charge in nanoparticle, they may have a tendency to poke inside, make a holes and then poke inside, go into the RBCs, and then the RBCs will be destroyed first. Okay, so that means, like, you need to keep in mind that not only the size, but also the charge plays a major role apart from the solubility or dispersibility of this nanoparticle. So these are some of the basics that you think, it is important when you create some of the nanoparticles, right? Then you might be thinking, we talked about so many varieties of nanoparticles, variety of smaller size, bigger size, and so on. But what type of nanomaterial that you want to choose for specifically drug delivery purpose? So based on the classification of the nanomedicine, considering these physicochemical properties as well as their applications in drug delivery, all together, so we have some information by combining all these with the case of like drug delivery strategies, how many physiological, designs, and also the near challenges that are needed to be considered for drug delivery application. Considering all these characteristics, if you think about liposomal formulation, they obey almost all the characteristics. Those are best for the drug delivery purpose. You all might be thinking, even the mRNA vaccines or COVID vaccines are widely developed with this particular type of nanomaterial, that is liposomal formulation. Next is the best for the using polymeric micelles or maybe polymeric nanoparticle. So if you know that majority of the nanomaterial, either it could be polymeric nanoparticles or polymeric micelles or the liposomal formulation, if you know all these three major category, you might be knowing that the importance of these nanoparticles in drug delivery application. You can modify the surface, or you can also attach the on the surface of the nanoparticles with the specific ligand or protein or any other aptamers or antibody and so on for specific drug delivery applications in targeted way at the tumor site. Same way you can design like in a particular organ specific, like for example, you want to deliver some nanomaterial to the brain, you can design such a way. If you want to deliver specifically at the kidney or liver or lungs, you can design all those based on the modifications on these surfaces of the nanoparticles or uniquely designing of various type of nanoparticles. So this is the basic information that I want to give today. The next session that I want to deal with, I want to give you some of the examples what are so far the FDA approved formulations of the nanomaterial that are widely used in the clinic. But before I go there, just I want to give one of the example, that is aspirin. I think all pharmacy students and everybody might be knowing this drug molecule, right? Aspirin in any one of your life at any time point, at one time point, at least, we have consumed this particular molecule, either orally or, any other way. Either it could be in the liquid form or in a pellet form and so on. But, this was created more than 130 years ago. But you see the leverage of the same drug molecule, but into a different formulation. Right now, they made a nanoparticle-based formulation that is giving a quicker or burst release of this drug molecule through this aspirin near tablet that is made with the um, sodium carbonate-based pill, okay, or a tablet. So what it does, like, it is quickly releasing so that it can take you at the inflammation site. Therefore, you may experience less within a short time period, you can get the relief from the pain. Okay. So now I want to show some of the FDA approved formulations. So you see the general concept, it's a number of FDA approved formulations for available for clinical application. It could be a paclitaxel, docetaxel, or various doxorubicin or any other liposomal based formulation for various near drug molecules for cancer therapeutic application. At the bottom of this slide, you can see the number of clinical trials that are underway that is exploring the use of antibody or aptamer or fragmented molecule that is going right on the top of these nanoparticles so that they can facilitate the directed towards the tumor cells and also minimizing the systemic toxicity. And these are like advanced concept, but I want to give you brief information of some of the FDA approved formulation. If you talk about doxorubicin, it's a highly water soluble nature and also highly toxic. Okay. If anybody gets this drug molecule as such, in the solution form, that will go and highly accumulate at the our heart. Okay. So they have you have a smooth muscle cells or cardiomyocytes. This drug molecule not only going to the cancer cells, but also going to the heart, smooth muscle cells, and also cardiomyocytes. So where they produce toxicity because they don't have a mechanism that only works on the cancer cells, right? So this molecule it works on the normal cells, but also on the cancer cells. So that is the major drawback of this drug molecule. Therefore, the nanotechnology come into the play. They make this formulation of doxorubicin and put it in the nanoparticles, and then you could see the complete options of the cardiotoxicity, right? This was the one of the major side effects that you experience from the doxorubicin. Like same way, considering same technology.
Sorry, Dr. Murali, is your slide supposed to be moving or just remain on the slide where the wording of our laboratory focus?
Oh, I couldn't get it. What is it?
No, I mean, now the slide that we can see is only the slide that, having the title is our laboratory focus. And then, and then that's it, there are pictures of nanotechnology facilitate cancer prevention, treatment, and detection. That particular slide, this is the whole slide that you are telling us? Or the slide not moving from the previous slide?
Oh, you're unable to see the
No,
slides?
sir, you are only on the very first slide, which is there. That's what the professor is telling. So the entire
Oh,
thing, what you are elaborating, it has got stuck up here.
okay. Looks like it's stuck from your side, looks like.
Yes.
So once
Sorry,
again, sir,
okay. I'm referring.
sir, once again, you can reload it and we can see it, sir. That is better, sir.
Okay.
Okay. So yeah, so then I won't be referring to the slides, but I will be talking. Okay.
All right.
So when you talk about the doxorubicin, it has a side effects on the human cardiomyocytes. So therefore, the nanotechnology liposomal formulation was made so to decrease the cardiotoxicity. Okay. So the next example, the same technology, liposomal technology, has been implemented for a number of other drug molecule, for example, amphotericin B, and so on. So when we talk about paclitaxel is another anti-cancer drug molecule. It is widely used for various type of cancer cell treatment. It could be breast cancer, ovarian cancer, esophageal, lung cancer, and cervical cancer, and pancreatic cancer, and so on. So what is this molecule does? It inhibits the mitotic spindle assembly. Okay, and also it stops the cell division. So how that happens, it stabilizes the microtubule polymerization. Therefore, the cancer cells stop the growth of these next division of these into 2 or 3 or multiplication. So therefore, so this is a very potent molecule, but the major side effect of this molecule is it has a lot more systemic toxicity, not only just from the paclitaxel molecule itself. In addition to that, when we make a formulation that is primarily used polyethylene based castor oil, where it also gives some of the adverse side effects. To overcome these two side effects, so a paclitaxel based nab-paclitaxel formulation has been made that is nothing but the paclitaxel has been conjugated to human serum albumin. And then final formulation, you would be able to achieve 130 nanometer in size. But these nanoparticles not only exploring the EPR mechanism as I mentioned before, the EPR mechanism is nothing but when you inject any formulation into the bloodstream, they'll keep going, circulating into the blood throughout the body, but wherever it sees the tumor, at the tumor site, your blood vessel formation, like you have a leaky vessels there. Therefore, nanoparticle goes and locks up there. Okay, that's called EPR mechanism. So these nanoparticles that were generated with the human serum albumin conjugated particle, that is Abraxane, it has 130 nanometer in size. As I mentioned before, the nanoparticle size somewhere between 10 to 200 nanometer. Those are good for achieving the EPR mechanism. So this is obeying that EPR mechanism, and also it also obeys some of the active targeting mechanism, because the albumin can bind to specifically GP60 proteins, so that are widely overexpressed in the majority of the tumor cells. So that means you are getting two different properties just by using one formulation, that is Abraxane. Okay, so that's one another example. Apart from all these, so you are also improving the bioavailability and biodispersibility or solubility of the paclitaxel, because paclitaxel is not at all soluble in the aqueous medium. Okay. With these two examples, just I want to make it clear that the nanoparticle size, charge, solubility, dispersibility, and its all the surface modifications are very, very important when you consider making any new formulation. Either it could be for cancer therapeutics, or maybe it can be for any other therapeutic application. Primarily, nanoparticle technology can enhance the solubility and improve the PK/PD of your drug molecules, and also it can improve the targeted delivery at the desired organs. Okay, you can specifically design, for example, you want to deliver something to the brain. Okay, you can design your nanoparticle and then put on the nanoparticle some of the APOE1, a protein that is, that can take you to the brain, or cross the blood-brain barrier. Or maybe, for example, if you want to deliver any drug molecule to the bones. So when you see, majority of the bone metastasis from primary tumors, you can experience a lot more pain. So you can also design such a way all your nanoparticles that way, and you name it, like you can construct all, specifically design all your nanomaterial, and then you can deliver at the tumor site. I'm very sorry, I couldn't show my slides to all of you, but I could see from my side, I could, move forward one by one, but, I couldn't realize that, you are unable to see my slides. But, with this, I would stop here, and then I would be happy to take all your questions, whatever you have, or you can also email me at any time, or I have given my email address. You can communicate if you want to discuss more about nanotechnology, how we can implement in various basic and applicative research. Either it could be a smaller or big project. I'll be happy to help all the participants. Thank you. I'll stop here.
Thank you so much, Prof. Murali Yalabu, for your informative presentation. We move to next speaker, Mr. Giris Nihalani, Teva Pharmaceuticals, USA.
We couldn't hear you, Dr. Girish. Yeah, Girish, I think we couldn't hear you.
And I hope this brief presentation will be helpful to everyone in some or other way. A little bit about me. I completed my master's in pharmaceutical sciences from Bombay College of Pharmacy in year 2005, and thereafter I have been in the development of solid oral dosage forms, the most part modified release generics, and to some extent 505(b)(2)s and NDAs. So my today's presentation is on the oral route of drug delivery for the peptides and why it is getting more attention, and what are some foreseen challenges and ways around it to practically make it feasible, the oral route of delivery for some of the peptides. So, needless to state why we should be preferring oral route or solid dosage form. The first and foremost reason is the patient compliance. For every drug medication, every dosage regimen, the more emphasis is given from patient compliance point of view, specifically considering pediatric and geriatric formulation. Pediatric and geriatric population, because the oral route is the convenient route of administration. One can self-administer the medication versus parenteral. Talking about the cost of manufacturing compared to parenterals is significantly less, and solid dosage form has a very enhanced shelf life, and specifically for a pharmaceutical industry, when a drug product is in solid form, there are multiple options existing for its manufacture, to get it manufactured from a third party, from a CDMO, from a CME, like those, and it helps in the product portfolio. And from IP perspective, for to formulate a solid oral dosage forms, there are multiple ways of designing it and strategies around it. Compared to the synthetic drugs, why or the small molecules, why peptides are gaining more preference and more attention in today's world? As the peptide, they resemble more towards our endogenous substances, the endogenous peptides. possess a very high safety profile, remarkable efficacy, and a specific in biological actions. Their metabolic pathways, as they are hydrolyzed by proteolytic degradation and primarily cleared by renal filtration. The metabolic byproducts, which are amino acids, they are not regarded as toxic to our body, and hence the safety profile is much better than the other drugs on the market. And moreover, in about, I would say, last 5 to 10 years, there has been many advancement in the synthesis technology of the peptides, in the technology of to get the purified form, to isolate the required structures in terms of advancement in the analytical techniques, which has made it possible to obtain the purest form of the peptides as compared to some of the other recombinant technologies for proteins, which are very difficult to guarantee a high purity, a high quality product of the proteins or peptides. And as this is a new field gaining more popularity for industry, there are advantages from IP perspective in uh, in combination with specific or unique formulation design, and these have been mostly employed in the metabolic disorders and the cancer. Based on one cited article, the compound annual growth rate for oral peptides is projected at the rate of 7.9% and to reach uh, a 50 billion target by 2027. Looking into some of the basics between the peptides and proteins, they are basically the similar molecules with similar structures. The basic differences being the peptides are relatively smaller molecules than the proteins, which consist of the peptides consist of 2 to 50 amino acids, whereas proteins, they are much larger molecules with more than 50 amino acids. And peptides and proteins, they both are the fundamental components of our cell constituents, and structurally they both have the peptide bonds. Within the peptides, which they can be further subclassified as oligopeptides and polypeptides. Oligopeptides with amino acids up to 20, and polypeptides between 20 to 50. This slide represents some of the examples of peptides based on their molecular size and structure. As you may notice, the structure of peptide itself can bring a lot of issues or problems in terms of the formulating them, or in terms of stability, or in terms of physiological factors affecting them. There are so many free functional groups present in the structure, and molecular weight is significantly high, and hence from pharmaceutics point of view, there are challenges in formulating these chemical moieties. So talking about the challenges, the structure of the peptides itself bring a lot of challenges, like starting from gut to intestine. They are susceptible to the pH, they are susceptible to the enzymes. Due to their large structure, they possess very low absorption through mucus or intestine, and their unstable structure or too sensitive structure makes it makes it a challenge from manufacturing process perspective. And even if they get entry into the plasma after crossing the intestine, they are readily hydrolyzed by the plasma proteolytic enzymes. And as the number of amino acids or the chain increases in the peptide structure, they do possess some level of immunogenicity. So in summary, the core issues with peptides are their bioavailability, their stability, which is due to their physicochemical properties, free functional groups, their sensitivity to enzymes and pH. So other than the challenges we just talked about, the other challenges are from analytical perspective. As a molecule is so big, the chain is big, there happens a permutation and combination of what possible impurities could exist in a pure form of drug, and hence advancements in analytical techniques such as GCMS, UPLCMS, NMR, mass spectroscopy. These are all being applied in the characterization of peptides. This schematic diagram shows different biochemical and physical barriers for the absorption of peptides, ranging from our mouth, the gut wall, the stomach, the intestine. There are a number of enzymes present to which peptides are susceptible for degradation. And if we look at the structure of our gut wall or intestinal wall, there are tightly packed epithelial cells surrounded by mucus, and these all limit the absorption of peptides. The mucus contains mucin, which is glycoprotein, which is negatively charged, and majority of the peptides, they are anionic, and hence mucus becomes, or the mucin becomes the first barrier for the absorption of the peptides. Looking at the different intestinal cells, which range from enterocytes, globular cells, M cells, these in the recent years have been targeted for the target specific delivery by carrier mediated drug transport. And even if a peptide gets an entry through the epithelial cells and enters into the plasma, it is metabolized by the proteolytic enzymes, and the enterocytes contains cytochrome P450 system, which again metabolizes peptides, and the peptides are thrown away by the efflux system. Looking at the mode of transports or the absorption, what could be the possible pathways of absorption for any molecule? It could be a small molecule or it could be a peptide. So basically active transport and the passive diffusion. So for the active transport is the carrier mediated transport, and the passive, for the passive diffusion, there are two options, either transcellular or paracellular pathway. So for peptide, to follow the passive diffusion, it is either by transcellular or paracellular. The hydrophilic and the larger peptide molecules, which have very low permeability through the gut wall, they tend to follow the paracellular pathway, and smaller and lipophilic molecules, they tend to follow the transcellular pathway of the absorption. And for the active transport, which has to be a carrier mediated, mostly by a protein which requires a receptor specification typically at the apical edges of those cells. So we are going to just brief based on the so many issues with the peptides, like their sensitivity to the enzymes, their sensitivity to the pH, their very large molecular structure or large molecular mass, their lipophilicity, and the free functional groups, which makes them unstable. Knowing all those issues with the peptides and knowing what we have from, I mean, what we can apply from pharmaceutics point of view in terms of selecting the ingredients, in terms of selecting the polymers, in term, the different designs of rotating the peptides, either from the pH or from the enzymes, or increasing the bioavailability, combining all possible approaches. Those are grouped basically into these four: either chemical modification, addition of effective agents, drug carrier system, or medical devices. I'm mostly going to brief only on the chemical modification and addition of effective agents in drug carrier systems. So within the chemical modification, we have lipidization, ketonization, PG glycation, and prodrug formation. Lipidization, as we know, the more the lipidic molecule, the more the permeability exists. So making the peptide molecule more lipidic in nature by addition of another lipidic molecule or by conjugating with a lipidic molecule does increases the permeation of the peptide. But however, in practical world and based on the efforts done so far, it was noticed that doing so lacks the specificity of the peptides. The second approach within the chemical modification is the ketonization. Ketonization, because we know mucin is negatively charged and opposes the absorption of the peptides. The ketonization of the peptides make it more possible for the absorption. Like, but the side effect of ketonization is it encourages the immunogenicity, and as a result, the peptide molecule could be metabolized much faster than expected. PG glycation is nothing but a covalent bond with the PG. In recent years, this approach has been applied to many molecules, including the insulin. Attaching a PG molecule to the peptide increases the molecular mass, which is the pharmacokinetic and dynamic properties of the peptides. But again, due to the non-biodegradable nature of the PEG related products, it can have its own downsides of either lack of specificity or increasing the viscosity. Another approach within chemical modification is the prodrugs. Prodrugs are nothing but the related derivatives of the main drug, but with better physiochemical properties. For example, one can go for the esterification of either carboxylic amino groups on the protein, and the esterification will result in more lipidic molecule, which will have more permeability than the actual drug, and at the same time, the esterification of the prodrug will protect the drug from the proteolytic enzymes and will dissociate at the desired pH of the intestine. The peptide cyclization method basically involves creating a lipophilic with the help of lipophilic linker to cyclize those free bonds onto the peptides, and thus the more sensitive carbon and nitrogen bonds, which are more susceptible to the proteolytic enzymes, if they are cyclized, the molecule gets protected from the proteolytic enzymes, and this results in higher stability. One of the another examples, like converting the L-peptide to D-peptide. L-peptide has the amino group on the left-hand side, which is more susceptible for the proteolytic degradation. and converting them to D-peptides enhances the shelf life and the stability. And within the addition of chemical agents or absorption enhancers, this is the most widely or attempted technique in the recent years. And absorption enhancers, they range from a normal surfactants to glycerides, salicylates. The list is pretty long. Every surfactant has its own downsides because the mechanism by which these absorption enhancers work, they either disturb the epithelial tightening within the intestinal structure, or they change the viscosity or make the peptide more permeable by either enhancing the solubility or chemical modification. There is a lot happening within this category. Absorption enhancer, the two absorption enhancer which have been used with success are the short form is SNAC, the sodium A2-hydroxybenzoyl or sodium caprylate in short, which by adding this enhancer, the advantage with this SNAC is there is a minimal destruction of the epithelial cell lining is reported, which is a very important for the peptide drug delivery, because if there is more disruption of the epithelial cell lining, there is a possibility of systemic toxicity as the as a more drug and the more surfactant will get entry into the plasma. And the addition of these enhancers also protect peptides from proteolytic degradation. Another enhancer is the BICNAC or 8-aminocapryllic acid, and in the literature there are successful outcomes reported with calcitonin. What it does by forming a complex with calcitonin which is insoluble at the gastric pH, and hence the molecule remains protected in the gastric pH, and while in the intestinal pH it dissociates, and this particular enhancer is known to increase the transcellular absorption pathway of the peptides. Another category is modulation of the pH, which the peptides are more susceptible to the enzymes as well as the acidic pH of the stomach, and hence one can think of applying a delayed release coating so that the drug gets protected before the drug gets protected up to the small intestine, and in the small intestine it can dissociate to get absorbed. And in combination, one can think of adding citric acid. Citric acid in the literature has been reported to protect the peptides from the proteolytic degradation, and hence combination of this both clear coating citric acid and maybe enhancer can have a very fruitful outcome onto the enhancing the bioavailability of the peptides. Proteolytic enzyme inhibitors, there are a few like aprotinin, which inhibits enzyme trypsin and chymotrypsin, lupeptin, which inhibits enzyme plasmin trypsin papain. PEG has been also used as a mucolytic agent in thinning of the mucus and increasing the permeability. Another category is the drug carrier or ectotransport mediated. In this, the trending technologies or the most research work which in the recent years has been reported is on the SMEDs, microemulsion self microemulsifying drug delivery systems. and nanoparticles. Nanoparticles, due to their particle size, they are more better ways of drug delivery through the intestinal wall. Hydrogels consist of a cross-linked polymer with peptide and can absorb a lot of biological fluids and hence remain insoluble in the GI wall, protecting the peptide from pH as well as proteolytic enzymes. Liposomes, which we know are basically the phospholipid molecules with hydrophilic ends, and the peptides can be encapsulated within those structures, and liposomes then can be used as such for the drug delivery. Based on the structure of the different cells present in the small intestine, especially the enterocytes, which we looked in one of the slides, based on those structures, one can make use of specific proteins with receptors in targeting those cells for the delivery of the peptides. Some of the other formulation considerations in terms of formulation, like manufacturing process, to choose dry process, obviously the first choice for peptides. In cases where the dry process or is not feasible, wet processes must have for certain good mixing of the ingredients. So one can think of like wet granulating only the excipients and dry mixing with the drug, and some of the aspects which we just talked about, the use of ingredients in the formulation. Basically, one can combine all possible ingredients to leverage to protect the peptide from the proteolytic enzymes, from the pH, adding the absorption enhancers. So in the market, there are more than 80 peptide drugs, many of which exist in the parental form. They already occupy like 30 billion market in of the world. Coming to orally delivered peptide approved by the FDA, we can see in this slide molecules like cyclosporin, semaglutide, octreotide, voclosporin have been approved. And if you look at the formulation mechanism, the is basically SMEDS, self-microemulsive and drug delivery system, adding a permeation enhancer with SNAC or sodium caprylate, which is C8, or cyclization. These are the FDA approved orally deliverable peptides. And the more peptides in the pipeline with application of either permeation enhancers or nanotechnology. This slide. Among the surface enhancers, it's very important to choose the one which has minimal disruption of the epithelial cells in the GIT wall, because enhance the choosing the concentration of permeation enhancer is very critical. Apart from the techniques which we just talked about, the different ways of enhancing the bioavailability of peptides, the new trends or new direction in the peptide structure which are being explored are multifunctional peptides, especially combining more than one peptide or coupling of the functional groups of the two peptides, and so as to leverage more output or more benefit or reduction of the side effects. And the peptide drug conjugates, which are basically carrier mediated transports, where kind of a small protein structure, which is maybe only 10 amino acid, is used as a carrier to carry a peptide, and then targets specifically or binds specifically on the receptors on the intestinal cells, which then facilitate the absorption of the peptide. And within the parenterals or injectable, the research is still going on to make the peptides more stable. And PEG glycan, which we also talked about, is still remains one of a greater tool to enhance the stability of the peptides, to protect the peptides from proteolytic enzymes and enhance the permeation. I hope my presentation was useful. Thank you so much.
All right. Thank you, Mr. Giris Nihalani, for your informative presentation. Let's move on to next speaker, Dr. Gelgel Wirasuta from Department of Pharmacy, Faculty Mathematics and Basic Science, Udayana University, Indonesia. Good morning,
Okay.
Dr. Gelgel Wirasuta. The time is
Yeah,
yours.
can you hear me? Hello?
Yes,
Yes,
yes, I can hear you.
yes, and the slide is, can you find my slide in screen? Okay. Hello? Can you see my
Hello?
slide on your, yeah, can you
Oh,
see my slide on your screen? I've shared the slide. Hello? Is my slide you can see on screen?
it's not see, Doctor.
Yes, see. Okay, I will back. I'll try again. Yeah. Sorry.
Sorry, doctor, do you have any trouble?
Yes, I couldn't share my screen. May you help me send I will send
Yes.
the
Can you send the slide to committee, doctor?
wait a moment. I've sent the slide to the committee, Mr. Cynthia, Miss Cynthia. Could you help me?
All right, Prof, the slide is show right now.
Okay,
Then you can continue
thank you
your
very much.
presentation.
Can you slideshow? Okay, thank you. I apologize me that the trouble with the instrument. I cannot so quickly to send my presentation. I give thanks for the committee to give me chance to present my research in Udayana University. That I'm give the title Control Quality and Ensuring Stability of Herbal Medicine by Key Marker Approach. Next slide, please. Could you slideshow my slide? Make the slideshow presentation. Next slide, please. Okay, if we see the market challenge of the herbal medicine, this is the new emerging market. The needed of herbal medicine is quite high, and especially of the market herbal and also wellness, and then in the specific of complementary alternative medicine. So for the quality of the herbal medicine, please the next slide, please. Yes, we should take the challenging to make the quality control of herbal medicine. We have the influence for the external challenging and all challenging, and also for the external challenging that making few poor quality of finishing product of herbal medicine. So we try to propose and use of the key marker on for the controlling quality of product herbal medicine. Next slide, please. Yes, the herbal medicine challenging solution, we use the quality marker. That means we control it for the external and internal challenging, making by quality marker. We try to find the marker of the herbal and control from harvest of the herbal, and then phase post of the harvest, and in the GMP area. And also, next slide, please. If we see the standardization compedia like pharmacopoeia, there are many factor controlling. One of them is HPTLC profile or HPTLC picture and profile of the herbal medicine to control of authentication and also quality of the herbal and originality of the herbal. And then we control also the adulterian and quality of the marker or secondary metabolic of the herbal medicine. Next slide, please. So the quality marker is the structure of the target of the marker. We try to use the phytochemical constituent of the herbal, and then we try to make the quality and efficacy, safety of the herbal medicine. We try to make the fingerprint of the herbal, and then from fingerprint we try analysis the quality, and then we have the key marker that is traceable, transitive, specific, effectiveness, and measurable. Next slide, please. This as one of our research, we try to differentiate of the pepper beetle with the other pepper from our region. Then we try to make authentication of the pepper beetle from and the quality of their for the uses of the antifungal activity. With the TLC profiling of the pepper beetle, we can differentiate of the species different pepper beetle like pepper beetle, pepper aduntum, cubeba, and also other pepper species. Next slide, please. So we try to introduce of the Ipomea batatas. Ipomea batatas is well known as the one potential pharmacological effect was found in that is as anti-diabetic mellitus for the herbal medication. Sometimes we can find the report the use of the herbal, also the use also the number of the Ipomea batatas. We can try to find out which of the chemical phytochemical substituent that have a function and have a pharmacological effect. Through the review analysis, we can found that flavonoid had a function in the liver, pancreas, and also the other organ. The liver can increasing of the secretion of the GLP-1, and also will decrease of the HbA1c that they can decrease of the glucose blood level. That mean the Ipomea batata has potential for the treatment of the uses for the anti-diabetes type 2. Next slide, please. We can try to find out the fingerprint of the people of the Ipomea batata extract by heavy TLC blood or densitometry, and also spectrum in situ spectrum of uh, the extract. This picture is we can give the name of the profiling of the herbal. We have before derivation and after derivation. We find out different color. We can use this for the identification of the originalitas and also of the quality of the marker. And then the spectrum we can use for the identification of each peak of the uh, chromatogram. Next slide, please. So we can identify the spot by color reaction that we can find which flavonoid substance have on the extract. Like this, we can find out the first peak is anthocyanin till the third peak, and the other is flavonoid. So we can find that which flavonoid constituent have of this extract. Next slide, please. Next slide, please. Next slide, please. We should evaluate of this variety of the fingerprint. We should quantify which is the chemical marker of the peak with have correlation to the extract from the area under curve of the peak. We can try to make regression with the difference quantity volume which apply to the plate, and then we identify which is the peak chemical marker or constituent there. And then next slide, please. After we identify, we can use this Andrea undercook, and then for analysis that which process have influence to the totally process in GMP. We can find out here Simplicia have the same peak marker or the profile marker with the extract and also with the product capsule. We can find out here have the similarity around more than 65%. Next, please. So we can present here that we have the chromatography with different area depending on the peak, the wavelength of the scanning of the chromatography. At the first, we make scan in the 330 nanometer, and at the second we can make 34. The first chromatogram is correlated with the flavonoid content, and the second chromatogram have correlated of the quantity of anthocyanin. According to the review, we found out that flavonoid and anthocyanin has reported have potential or have responsible effect to the GM. And then we use this area under curve of the collected peak to quantify of the quantity of flavonoid by polylinear square of this linear list square for the district, and we found out that three different volume we have applied to the plate have linearity, good linearity. So this experiment we can use to the control quality of the production in the industry. Next slide, please. So we can use also to the control of this profiling of the fingerprint of the raw material extract and also capsule. We have the same similarity of the profile, and then we can also test it of the this on into the preclinical study. We got here that the preclinical study with the different quality of the total flavonoid we can use to control which doses that better to use for the remedy in the capsule. And we can found that for the uses of the 700 milligram of total flavonoid content can reduce better than the others and also better also with the positive control of the remedies. Next slide, please. If we use further, this can be used to find which of the key marker by identify the marker, chemical marker, and then predict active molecular. After that, we can predicting of the molecular mechanism pharmacology by in silico docking. Next slide, please. In this experiment, we can find out that this molecular have pharmacological effect to the expression. Please, next slide. Yes, the conclusion and presented, the efficacy and safety of the herbal medicine depend on the quality of the raw material, the quality control and standardization currently, and stability of the for the Simplicia extract and herbal medicine preparation is inadequate considering that the determination of one or several marker component is not sufficient correlated with the efficacy or chemical selected or for the quality control of the extract and not representative of the drug as whole. Determining correct key marker can identify the change chemical component during the preparation development process, and also it can guarantee the efficacy and the safety of the herbal product. The potential analytical method uses determining key marker is combining by TLC and HPTLC densitometry. Combining the TLC method, spectrophotodensimetry as an in situ scanning process is able to produce quality data and form spectrum maximum wavelength to support the RF value and qualitative data from the AUC of the chromatogram, where the various data can be used to determining optochemical fingerprint. The result of the analytic by using combination of those two methods are able to analyze the similarity of the sample based on chromatography this residual. Based on the advantage of the happy TLC method comparing to the happy LC method, determining key marker by using happy TLC as more visible industry application. Next slide, please. The conclusion is this is our reference we can use, and then thank you for the all for the chance that give me to present our research. Thank you very much. I give back the time for committee.
Yes, thank you so much, Dr. Gagal Wirasuta, for your explanation and informative presentation. Let's we move on to next keynote speaker is Dr. Prakash S. Gondanavar, Professor and Vice Principal, Department of Pharmaceutics, Sri Adi Chunchanagiri College of Pharmacy, India.
Good morning. Let me share my presentation. Give me a half minute, please. Hope the presentation is visible.
Yes, sir.
Okay, thank you. Thank you again. Let me start with my topic. I'll just be highlighting on the recent advances in drug delivery system for targeting brain tumors today, which can improve the therapeutic efficacy and patient outcomes. Myself, Dr. Prakash Gaudnavar, I belongs to Adi Chunchanagiri University, India. Presently, I'm working as a vice principal in the Faculty of Pharmacy. So let us start with a brief introduction on brain tumors. As everyone of you know that these brain tumors are abnormal growth of cells within the brain or the central spinal canal. And these brain tumors can be non-cancerous, called as a benign, and also can be a cancerous. With malignant tumors basically poses a significant health risk. According to the American Brain Tumor Association, I'll just go to the next slide so that I can explain there. So you can see here, this is as per the US data. Around almost 7 lakh plus people in US are living with this primary brain or central nervous system tumors. So in that, around 5.5 lakhs with benign tumors and 1.
Class sir இருக்காரு மேம் தியாக சுந்தரம் சார்
Yeah, hope other the participants can mute themselves so that I can explain. Thank you. So in that 7 lakhs, around 5.5 lakhs are with benign tumors and around 1.38 lakhs with malignant tumors. So basically, the primary these brain tumors, they originate in the brain, so which are more challenging to treat compared to the other metastatic brain tumors, which basically spread from the other parts of the body. Of course, there are so many treatment options available for brain tumors, which includes surgery, radiation therapy, chemotherapy, and targeted drug therapy. Despite so many advancement in treating these brain tumors, but still these brain tumors remain a significant cause of morbidity and mortality worldwide. So again, depend upon the size and shape or the type of a brain tumor, the symptoms can vary, like say vision loss, the hearing loss, seizures, the headaches, fatigue, sensor loss, right? So these are some of the symptoms based on the type and size of the brain tumors. Now coming on to the main topic, that is the importance of targeted drug delivery. There are so many conventional drug delivery methods are available to treat these as a treatment option for the brain tumors, but these conventional drug delivery methods they often result in a low drug concentration at the tumor site. So because of that low concentration at the tumor site, because most of the concentration of a drug is lost in the surrounding tissue of the brain tumors, so which can leads to suboptimal treatment outcomes. And also, since some of the drug concentration is lost in the neighbor the tissues of brain tumors, there is increased systemic toxicity. So that's why the targeted drug delivery systems are developed to aim to overcome these all limitations basically of the conventional drug delivery systems, right? So by enhancing this drug delivery to the brain tumors, the targeted approaches can be improved, right? And or it can enhance the therapeutic efficacy, you can say, and reduce side effects as well as enhancing the patient quality of life. This is the how the drug is transported across the blood-brain barrier. So there are so many mechanisms involved, like paracellular, the transcellular pathway, then the transport of proteins, the flex pumps, right? So these are some of the mechanisms of a drug transport across blood-brain barrier. Now, coming on to the one of the major part of this my presentation is challenges in developing drugs for the brain cancers, right? So there are so many limitations associated with the drug therapy for the brain tumors. So one is the barrier, that is blood brain barrier. Along with the blood brain barrier, even the blood brain tumor barrier, the tumor microenvironment, then the heterogeneity of a drug distribution, tumor mutations. So these all are the challenges which basically come across while developing the drugs targeting the brain tumors. Compared with the peripheral tumors, the other tumors, the treatment of brain tumors, it faces, as I said in the previous present slide, that is many challenges. So on one hand, you can say all these physiological barriers like blood-brain barrier, a blood-brain tumor barrier, and the overexpressed reflex pumps. So these all prevent drugs from entering the CNS so that they reach the tumor site. So one of the major challenge is the barriers of brain as well as tumors. On the other hand, some of the characteristics of a brain tumors, such as say infiltration, invasion, high heterogeneity, drug resistance, and immune escapes. So these all which are caused by a tumor microenvironment, which I'll be explaining in the further slides. What do you mean by tumor microenvironment? And one more, the characteristics of the tumor environment is the cancer stem cells. So further, they inhibit the therapeutic effects, which leading to high failure as well as the recurrence of the brain tumors. So this is, you can go through this blood-brain barrier and blood-brain tumor barrier. These are the two main barriers, basically, which inhibit the entry of most of the drugs to the CNS, so that they fail to reach that the brain tumor site, right? So there's so many mechanisms included, like say paracellular barrier, like say transcellular barrier, some of the enzyme barrier, right? These all limit the drug permeability through the blood-brain barrier, so contributing to the drug resistance in the brain tumors. And as the brain tumor progress, there is a development of a blood-brain tumor barrier, so which further again limits the entry of drugs to the target site. As I said about the infiltration and invasion, the tumor microenvironment. So brain tumor cells basically exhibit certain aggressive behavior, like say they infiltrate to the surrounding tissues. Apart from the tumor site, the tumor cells, they infiltrate to the surrounding tissues and forming tumors through a series of steps. So the process of this infiltration begins with the migration and accumulation of tumor cells near blood vessels. So these all factors basically disrupt the normal contact between endothelial cells and the basement membrane. Yeah, one more challenge is brain cancer stem cells. These cell subsets within the brain tumors, the brain tumor cells, they exhibit stem cell-like characteristics. And these brain cancer stem cells, they also possess certain key characteristics like say aggressiveness. So these the brain cancer stem cells are highly migratory. They migrate to the surrounding tissues. So that is called as invasive. And there are these brain cancer stem cells, they are also resistant to chemoradiotherapy. And these the cancer stem cells are self-renewal and differentiation. Similar to the normal stem cells, right? So these the cancer stem cells can renew themselves and differentiate into various types of cancer cells. And one more key characteristic of the cancer stem cell is the drug resistance. So they demonstrate the multi-drug resistance by mechanisms such as DNA damage repair, and efflux transporters. Then so you can see this figure. These the cancer stem cells, like say, they are self-renewed compared to the other normal cells. Despite the efficacy of standard therapy in killing most tumor cells, these cancer stem cells, they invade the brain parenchyma, which leads to the disease recurrence due to their invasiveness. So I was explaining about the infiltration and invasiveness. They have the resistance to the most of the drugs. They are self-renewal and differentiation. So eradicating tumor stem cells is a crucial research focus. So apart from treating the brain tumor cells, these stem cells, right? So eradicating these stem cells is again one more research focus to overcome this disease, right? Now, the one more challenge is the immune escape in brain tumors. So basically, the central nervous system traditionally considered to lack some of the normal lymphatic and dendritic cells, which are essential for the antigen presentation. So there is always the compromised immune system in the central nervous system, so further inhibiting the active immune reservoirs. So this immune privilege within the CNS creates an environment conducive to the tumor growth. Since there is compromising of immunity in the CNS, so this creates an environment which is conducive for tumor growth. So as tumors can evade immune detection and destruction. The immunotherapy has shown therapeutic potential in various solid tumors like melanoma and some of the small lung cancer cells, but its effectiveness in brain tumors remains limited because of the conducive environment in the central nervous system. One of the primary reason for this limitation is the inability of immune components. Some of the immune components inability, like say antibodies and immune cells. So this is immune invasion by the tumors. So which was I was explaining as the antigen preparation bodies are limited in the central nervous system, which can inhibit the immune system. Next is one more challenge is tumor microenvironment. So this comprises of various components, which includes the tumor cells, tumor stem cells, blood vessels, lymphatic vessels, immune cells, right, and extracellular matrix. And this TME, it plays a critical role in supporting the tumor growth, its proliferation, angiogenesis, and metastasis. Right. So this tumor microenvironment protects the tumor cells through several mechanisms. The one is the increased vascular endothelial growth factor activity. The second one is interaction with the cytokines and the growth factors. The third mechanism how this tumor microenvironment protects tumor cell is through extracellular matrix cross-linking. So these are some of the mechanism through which the tumor microenvironment it always protects the tumor cells. So thus it becomes a challenge, to develop any suitable drug delivery system to treat the brain tumors. Now, moving on to the design strategy of a brain tumor targeting nanodrug delivery systems, right? So till now we discussed about some of the challenges in treating the brain tumors, some of the limitations which we come across while designing any drug delivery system to treat the brain tumors. Now, I'll just highlight on the design strategy of a brain tumor targeting nanodrug delivery systems. These nanodrug delivery system, of course, they offer some unique advantages in drug delivery. Now, some of the common design strategies for this nano drug delivery system is targeting a brain tumors, including it includes like say optimizing some of the physicochemical properties of this drug delivery system, overcoming the blood-brain barrier and blood-brain tumor barrier, and introducing some stimulus responsive functional groups in the drug delivery system, and targeting specific organelles. So these are some of the design strategies for the targeting the tumors with a nano drug delivery system. We'll see, you can see here some of the nanoparticles which binds with us some specified receptors, like say liposomes, some polymeric nanoparticles, dendramites, polymerosome, nanogels, exosomes. So these all are nano drug delivery systems. Now the first the strategy, designing strategy for optimizing the physicochemical properties of nanoparticles. Now, so optimizing the size, optimizing the surface charge, optimizing the morphology and surface modification of nanoparticles can significantly influence their circulation in the bloodstream, and so that they can accumulate in the brain. This is one of the crucial consideration in the nano drug delivery system design. So basically these nano carriers, they accumulate in the brain tumors, as I said in the previous this one, through passive targeting mechanisms, and their accumulation in the brain tumor is influenced by their size and surface properties. So optimizing some of the physicochemical properties, like say the size, so keeping a size in between 10 to 100 nanometer, so which will be efficient for them to penetrate across the blood-brain barrier, and keeping the charge, that is in a positive size. So positively charged nanoparticles, again they enhance the interaction with blood-brain barrier. And again morphology, optimizing the morphology of these nanoparticles. So like say rod-shaped particles, unlike the other spherical or the cubical shaped particles, the rod-shaped particles are more superior in cell uptake, so which can enhance the drug delivery efficacy to the brain tumors. The second design strategy is to overcome the blood-brain barrier and blood-brain tumor barrier. So again, various strategies can be employed to facilitate drug delivery across this blood-brain barrier as well as blood-brain tumor barrier for effective brain tumor treatment. So these strategies include, like say, carrier-mediated transport, utilizing some specific transporters like say TFR or glutathione 1, are going for receptor mediated transport or absorption mediated transport and cell mediated transport, right, employing some leukocytes and stem cells. Additionally, efforts to reduce drug efflux by you can inhibit efflux proteins and utilizing the enhanced permeability and retention. Then there is one more strategy called as dual targeted drug delivery system, which can target both blood-brain barrier as well as blood-brain tumor barrier. So these are crucial for selectively delivering the drugs and minimizing the distribution to the normal brain cells. So now the latest technology is dual targeted drug delivery system. There is one more strategy called as stimulus responsive nano drug delivery system. So again, the stimulus responsive, the emergence of stimulus responsive nano drug delivery system. This is nothing but introducing the sensitive groups which can undergo physical and chemical changes when they are stimulated at the pathological sites. So upon the stimulation, these groups, they can enhance the drug delivery efficacy. There's one more strategy called as pH responsive systems. So because of a tumor microenvironment, which we discussed in the previous slide, so there is a decreased pH. So there is a decreased pH in the tumor environment. So employing some of the pH sensitive materials like say acetals, the mites, or orthoesters, using these pH sensitive materials, this can trigger the drug release at the target site, that is tumor site. And there is one more called as redox responsive systems, which exploits the high concentration of a glutathione, right? So this is one more strategy, for nano drug delivery systems. This is about the stimulus. As I said, some stimuli response functional groups can be added in the drug delivery system. So upon reaching the tumor site, that is environment, they undergo stimulation and enhance the drug release onto the target site. There's one more strategy called as organelle targeting in nano drug delivery system. This is targeting organs like say nucleus targeting, the mitochondria targeting, right? So exploiting the high mitochondrial membrane potential in tumor cells to selectively deliver the drugs. One more is called as a lysosome targeting. The last one is called as endoplasmic reticulum targeting. So organelle targeting is also one of the basic strategy to enhance the drug delivery to the tumor site. There's one more called as a Golgi targeting, so which disrupt the signaling pathways involved in the tumor growth, invasion, and metastasis. So targeting to the different organelles can also enhance the drug delivery to the target site. There's one more strategy called as a drug combination strategy, which is based on the multifunctional nano drug delivery systems. In recent years, this combination therapy has emerged as a promising approach to improve the efficacy and reduce the side effects of cancer treatment. So this is basically relevant in context to the brain tumors, where as we discussed, there is so much complexity of the disease, which is presenting a challenges for single agent therapy. So going for a combination therapy, like say which includes the chemotherapy, targeted therapy, the immunotherapy, and gene therapy. So these all are investigated in clinical research for brain tumors. But still, so many combination therapies, there remains a need to optimize existing combination therapies or strategies to address the challenges associated with treating brain tumors effectively. Some of the advantages for drug combination therapy. Hope I have some more time, at least 5 more minutes. Okay, I'll take 5 more minutes, please. Some of the advantages of a drug combination therapy. One is enhanced efficacy. So combining drugs with a different mechanism of action can target multiple pathways which are involved in the tumor growth and survival. So this improves overall treatment efficacy. So combining drugs with a different mechanism of action can enhance the efficacy of the drugs, reduce toxicity, so lower doses of individual drugs. So basically now when the individual drugs are given for treating a brain tumors, right, the dose will be always high. So now going for a combination where lower doses of individual drugs can be used in combination therapy, so which can minimize the toxicity and side effects associated with the high dose monotherapy. Prevention of a drug resistance. Again, the use of multiple drugs can reduce the likelihood of tumor cells developing a resistance. Basically, one of the major challenges is tumor cells developing that resistance to the single drugs, right? So that can be prevented. Now, targeting tumor homogeneities, again, combination therapy can target different subpopulation of a tumor cells. Application of multifunctional nano drug delivery system in combination therapy. Of course, we discussed about combination therapy, giving drugs in a combination which is more effective or more efficacious in treating the brain tumors. Now, application of a multifunctional nano drug delivery system is one more the recent approach, right? So combination of a small molecular chemotherapy drugs. This multifunctional nano drug delivery system can deliver 2 or more chemotherapy drugs with a different mechanism of action so that it can improve the treatment efficacy. The control release and targeted delivery capabilities of these nano drug delivery system can optimize the drug distribution, and also it can give the synergistic effects because the combination of drugs are used in this multifunctional nano drug delivery system. The second approach is chemotherapy immunotherapy combination. Again, nano drug delivery system can be engineered to co-deliver chemotherapy drugs as well as immunotherapeutic agents. Again, there is a combination of chemotherapy drugs and immunotherapeutic agents which are combined and given as a multifunctional nano drug delivery systems. And one more is chemotherapy gene therapy combination. So again, the nano drug delivery system can facilitate the co-delivery of chemotherapy drugs as well as gene therapy drugs, for enhancing the targeted delivery of therapeutic genes to tumor cells. So these are some of the recent approaches like combination of drugs using a multifunctional nano drug delivery system. Of course, there should be some considerations in designing multifunctional nano drug delivery system. We need to consider some of the aspects like neurotoxicity. A selection of materials to minimize neurotoxicity is essential. Drug leakage, strategies we should produce. I mean, we should come out with some strategies to prevent drug leakage during transportation that should be implemented, and protein corona shielding or off-target effects, minimizing the off-target effects through precise targeting and control release mechanism. So these are some of the consideration while designing a multifunctional nano drug delivery system. So, so we discussed about the challenges in treating the brain tumors and some of the recent approaches for treating this the brain tumors. So the conclusion is, of course, still a brain tumors present a formidable challenge in oncology due to their complexity and lethality. So overcoming all the obstacles which are related to the blood-brain barrier, blood-brain tumor barrier, tumor heterogeneity, invasiveness, immune escapes, tumor stem cells, and tumor microenvironment. So it requires innovative approaches in drug delivery. So that's why we discussed a multifunctional nano drug delivery system or a promising solution. That too again multifunctional nano drug delivery system involving the combination therapy, involving the multi drugs is a promising solution by addressing these challenges, so that enhancing the therapeutic outcomes through combination therapy. So future directions: continued advancement in understanding the brain tumor physiology, identifying new therapeutic targets, developing novel anti-tumor drugs, and refining material science and nanotechnology will drive the translation. And one more important future direction is a personalized administration and precision treatment strategies, which can further optimize, therapeutic outcomes for patients with the brain tumors. With ongoing process in emerging therapies such as immunotherapy, gene therapy, and cell therapy, the era of effective combination therapy for brain tumors based on multifunctional nano drug delivery systems is a future direction. I thank you, the organizing committee, for giving me an opportunity to present my topic in this drug discovery and community trial conference.
All right, thank you so much, Dr. Prakas Gonda Navarro, for your interesting topic for today. Let's move on to next speaker is Dr. Kartik Anantaraman, Vice President of International Business of Apollo Hospital, India.
Hello, thank you very much, and I hope I'm audible. Ida, if you can please confirm if my audio and video is fine.
Yes, your audio and your video is clearly
Thank you.
fine.
Thank you very much, and thanks for having me at this third Pharma Summit. And I think Dr. Prakash had a very lucid presentation, which is extremely informative. And therefore, what I would like to do is I'd like to base my talk around, while he has given a lot of insights about what is happening in biotechnology, implications of biotechnologies on immunotherapy, targeted therapy, etc. In fact, we do a lot of work every day in terms of managing cancer patients at Apollo hospitals, where we take very specific measures in terms of not just therapeutics, but we're also now giving higher importance towards preventive medicine and prevention of cancers as well. So a lot of our consultants have now been very specifically, you can say, taking specific measures to go and talk with physicians in the community as to how we can make a difference in terms of preventive cancer, or one of the other things in terms of cancer management or other management of any disease which involves the immune system, is in terms of early detection, because curative intent is much better and the chances of cure is much better once these kind of conditions are detected early. So therefore, what I'm going to do is over the next couple of minutes, so I'm going to talk about the technology applications which are changing the world of life sciences and healthcare today. And it would encompass biotechnology, pharmaceuticals. It would also bring together, in a way, technology, which is typically today driven through artificial intelligence and machine learning platforms. And what are the key application areas of AI and ML in the life sciences space today? And which are the organizations that are working in this space that are making a difference to us today? So that's going to be basis of my talk today. I will just share my screen. Kindly let me know if my presentation is visible. So the presentation would actually read opportunities in life sciences. Ida, if you could confirm if my screen is visible, I'll start my presentation. So I'm going to be looking at some key applications that we have in healthcare, in life sciences, which basically have been transformed by technology. So if you broadly look at the digital healthcare ecosystem, so the digital healthcare ecosystem consists of these core components. So one set of core components are basically related to patient-generated data, which would include mHealth wearables, etc. One set is related to data visualization, which is data analytics. Another set is related to decision support, which is where artificial intelligence comes in, which is all around knowledge and insights that you would gain and actionable insights that you could have in healthcare. The fourth one is clinical, which is around precision genomics, health records, and claims data. And the fifth one is around social determinants and economic data. So these are the five main pillars on which digital healthcare ecosystem is defined. As you can see, there are many organizations that have specialized, and you can see by specialty, there are these organizations that have been doing excellent work in this particular space. So you can talk about oncology, women's health, wound care. So you can, depending on each of these respective, you can say specialties, you have, specific startup organizations, well-funded organizations who've been working in this space. And these are some of the top 20 digital therapeutic companies that are well funded today, which are working in the field of digital therapeutics. And as you know, digital therapeutics today has been accepted by the drug regulator as well as a technological advancement or a technological, you can say, support to pharmacotherapeutic, which incrementally enhances positive clinical outcome in patients. And these are some of the companies that are in that space. And these are the top organizations which are in between digital therapeutics, mental wellness, telehealth, B2B sourcing, measurement and testing within the healthcare domain. And if you're interested, I'll be happy to share this across with you. And these are the top 20 to 21 top organizations who have had very good technologies in digital therapeutics. And these are well-funded organizations, most of them based either in the US or in parts of Europe. And there are many that are now coming up in India as well as we speak. So therefore, the digital health space is basically a collaborative effort between telehealth or telemedicine providers who also have telepharmacy built into it, teletherapy with a direct-to-consumer approach, having either a hybrid or an in-person or a virtual care approach, including remote monitoring, diagnostic and devices, as well as virtual digital care enablement. And large organizations like CVS, and from an Indian context, including Apollo 24/7, Tata 1mg, etc., are basically now combining a combination of care coordination, chronic disease management, diagnostic and therapeutics, healthcare IT, home health, and virtual care integrated into one digital healthcare delivery platform. So there are also multiple publications and key influencers that have been working diligently in this space. And these are some of the references that one can refer to if they're interested in this particular domain. The five key ways in which digital health is influencing healthcare industry are: number one, AI, augmented and virtual reality application, remote patient monitoring, bioprinting, telehealth, and teleassistance. And I'll also talk about the impact of some of these in the instance of drug discovery and its impact on pharmacotherapeutics and in terms of diagnostics. And the five key tech trends that are impacting the healthcare industry are primarily wearables, increased availability of broadband, rapid growth of genomics and gene editing, rapid advances in AI, and remote working through virtual meeting software and services. So these are the five main drivers or disruptors here. And India is no longer a laggard. In fact, we have the best of internet penetrations in the world today. And you can see that the ability of Indian doctors, Indian consumers to use internet-led and technology-led healthcare consumption has gone up significantly. And you can see by state, we now have not just internet penetration, but we also have consumption data of you can say, telehealth services, including telepharmacy, digital pathology lab, consumption, etc. patterns marked across the country. And not it's not only the younger age group that is searching for healthcare services and solutions online. You see that even the older age group, even 40 plus until 74 or 75 years of age, the research has shown that people are using technology to avail healthcare products and services. And the key immediate opportunities that we have as a country to really explore is to look at what could really replace Codex and possibly compete with Microsoft Azure in the OpenAI service, because natural language based no code solutions are the way to go. And GPT-3 features, while it does include powerful search and conversation and idea generation, it still provides larger headspace over there. And some of the other opportunities is the example of how Google has been working closely with Mayo Clinic to build a data lake with anonymized data. And similar platforms is something that we are exploring at Apollo, and other larger healthcare institutions with access to large data can certainly look at some of these, including some of the more recent things that we have done with Cohere Med, where we have actually developed a sepsis predictor, looking at large data sets that's gathered from patients admitted to the ICU for sepsis and severe infections. Some of the other opportunities are to look at, lookalikes of Nuance and Doc AI. As you know, these are very strong digital health platforms that enable capture of real-world medical and life data and their polyomics data pipeline. And this really gives a lot of data analytics-led insights as well. And AR technology has significantly disrupted the connectivity between doctor and their patients. And this has been proven in very extreme of situations like, the ongoing war between Russia and Ukraine has disrupted normal life, has disrupted healthcare as well to a great extent, but still technology comes to the rescue where doctors are still able to connect, and telehealth or connected health-based healthcare services are still kind of saving the day and saving a lot of lives over there. The opportunity that rests with conversational AI in healthcare is huge as well. And as you can see, conversational AI is growing extremely well. We've had an experience in our oncology department where through conversational AI, we are able to actually build tools and methodologies, which actually helps in much faster screening of patients seeking, targeted therapy, immunotherapy, or even, qualified for chemotherapy. And typically, as you know, most of these therapeutics are instituted either, just prior to surgery, if it's a neoadjuvant, or if it's an adjuvant immediately post-surgery. So the appropriate timing, the appropriate responses that are seen in these patients are monitored through conversational AI applications, which we have seen. Pharmaceutical companies like, and also medical device companies like BD have been also partnering with some of these leading organizations to basically identify, insights through patient data. And they have been doing post-market surveillance on their own products at BD to fuel innovation and unlock faster and more efficient paths to the market as well. Cohere for AI is another great startup, which is actually creating language-based models that rivals OpenAI. So something that people that are interested may go after. Patient-controlled electronic health record is another very important technology that is disrupting healthcare. And this is giving hospitals like us a lot of data and insights about how patients are doing after they have gone out of the hospital or after they've left the OPD after the consultation. So having that insight is very important to plan for the prognosis and the follow-up investigations and the follow-up results that we actually deliver. Personal health monitoring through point-of-care devices is another important thing to look at as well. These are the top health innovator platforms that are doing exceptional work there. And then now I'll come to the core area, which will be very much in line with the conference or the Pharma Summit today. What are the key applications of AI in drug discovery? What are the core successful examples over there? What are the applications in medical imaging, diagnostics, clinical pathology, and finally clinical trials? So I'll show you some of the examples, and these are real-time examples that are working very well today. The first example that I can talk about is in silico medicine. I think in silico medicine is amongst the first companies to basically adopt AI and they have started their first human clinical trial on a study drug, which is a candidate drug that's identified for pulmonary fibrosis. And this drug, this target or candidate drug discovery happened completely using an AI design model, right? So this is the very first, I would say, success stories that one has in terms of how AI is able to disrupt drug discovery. And this is the very first, world's first AI discovered compound to enter the clinic, and more compounds are expected to come in the near future. It also significantly reduces the time to discovery and time to get into preclinical, which earlier used to be anywhere between 48 to 60 months, which is now shrunk to almost 18 months, and a significant cost saving as well in that whole process. So these are the steps that it actually follows. So there is data input, then there is Pharma AI, which actually works on end-to-end drug discovery as a platform, and then it goes through early preclinical and, as you know, effectively API formulation, and finally into human clinical trials. Uh, the second best example that we have is a partnership between Okin, which is the AI tech platform organization, and Sanofi, which is a large pharma company, as all of you know. So they have entered into a strategic multi-year collaboration to seek new cancer therapies, and they're looking at target discovery using artificial intelligence. And the core focus areas here have been NSCLC, or non-small cell lung cancer, as we call, and TNBC, or triple negative breast cancer, mesothelioma, and multiple myeloma, which are the key target oncology, you can say, areas that they wish to seek support from AI technology platforms in terms of new drug discovery. Recursion is another platform which has partnered with Roche and Roche Genentech. Path AI is another platform that has also partnered with Roche. So Recursion is on the Roche pharmaceutical side for development of novel targets, whereas Path AI is on the Roche diagnostic side to look at AI-powered digital pathology solutions that Roche has on the Roche Diagnostics business. Likewise, Benevolent AI is another top platform that's been helping, and their recent collaboration with AstraZeneca and Eli Lilly has led to some very good target discoveries. Cellularity Map is another, great example in the same space, which one can look after. And Isomorphic Labs, so as you know, is a dedicated platform dedicated to AI for drug discovery, and plans to reimagine the entire process here as well. So those are some of the, you can say, examples of drug discovery that's enabled through AI. Now, can AI do a lot more? Yes, there are many other areas that people are looking at leveraging AI. I'll go through them one by one with actual examples. This is the example of AI improving the use of hearing aids. So therefore, it has an application in the audiology market. AI is seen to reduce hospital stay, which means the length of stay of the patient in the hospital is coming down, so recovery is better. And this is all completely driven through AI analytics and data insights. AI is streamlining care in hospital by reducing these hospital stay, and naturally, length of stay comes down. It's able to predict clinical outcomes much better. And today, in fact, at Apollo, we have a partnership with an AI-enabled financing platform, because a lot of cancer patients, as you know, need health funding or health financing, because cancer treatment is not easy, it's not cheap. So patients many times need funding support for cancer treatment. We have partnered with an AI-enabled platform service provider who's able to look at the patient reports, look at the hematology reports, look at the PET-CT, look at the biopsy reports, and look at the sequential treatment the patient has received in the past. And it's able to come up with a predictive analytics on what's the expected outcome in this patient. And we then have outcome-based funding gets offered to the patient as well. So that's the other application that we see. It's now able to predict tumor regrowth in cancer patients, a very, very important aspect of monitoring prognosis in cancer patients. So therefore, this is what is driving what we call customized medicine or precision medicine. And these applications are extremely well developed now. There are great applications in imaging, especially CT, MRI, PET-CT, where you can now do functional imaging of the organs, especially for the brain, the heart, and most importantly, functional imaging of targeted organs that are inflicted with primary site of cancer. It's also helping autism kids. So there is AI, artificial intelligence that is supporting autism detection and therapy in autism. AI neurofingerprinting is also something that's an upcoming area, and AI has applications in management of Parkinson's disease as well. All of these are proven applications which are currently being used at various leading hospital sites across the world. Uh, then FDA has also cleared technology which is called as a Strive PD mobile app, which is used to monitor mobility data in patients with Parkinson's disease. So therefore, this clearly helps in monitoring, mobility and neurological functions in patients with Parkinson's, and it's able to monitor therapeutics really well basis that. It also has a great application in colorectal patients with colorectal polyps and colorectal potential colorectal cancer. And these applications are helping in early discovery and hence early management, surgical followed by radiation if required. Then there are advances in spinal cord injuries and surgical management of such injuries. There are applications in neurosurgery. There are applications in cardiovascular disease, including applications in retinal scans, which helps detect macular degeneration, helps detect early signs of diabetic retinopathy and things like that. And there is significant implication in early detection of breast cancer, which is one of the most advanced, I would say, forms of early cancer management that we have seen as an application here. There is application in radiology. There is application in mental health. There is application in radiology. I think I need to talk about this because this really helped us during the COVID times. Differentiation between COVID pneumonia and non-COVID pneumonia was an extremely important application that we were looking at, during the COVID times. And this application basically helped us, in early detection. And there were AI applications that could read through high resolution CT or HRCT as we call, which could differentiate between bacterial pneumonia versus COVID-19 pneumonia so that targeted treatment could be started on time. And there are multiple Indian organizations that have this technology as well today, which is applicable on both digital X-ray platforms and on CT platforms and MR as well. And then we have the detection of fracture. As you can see, this is a radiology application where something that was missed by the human eye was detected by AI. And thereby we have multiple applications across oncology as well as non-oncology applications that we see here. And broadly, finally, I'm going to come to the last part of the presentation, which talks about pathology, where we've had multiple technologies that are being used in vitro diagnostics, making pathological diagnosis more sharper, more precise. And these are some of the examples that you can see, Page being one of the more advanced platforms in this that has been partnering with some of the leading, biotechnology companies as well. Broadly, what I can say is that technology has been a great disruptor. It's been a great disruptor in clinical trials as well. AI is able to now cut short the time taken to conduct of clinical trials and hence cut short the time and the cost as well. And we are seeing all of these clinical trial platforms, digital CTMS platforms, leverage AI significantly these days. A lot of the global CROs are also using AI-enabled technologies in their clinical trials, especially phase 3 human clinical trials. And we're seeing that this is truly helping this one. Last one is actually on Amgen and Ofkin partnership in early detection of cardiovascular and cerebrovascular events. You can see this is a great example there, and NeuroKey brain-computer interface, etc. So therefore, these are the various, I would say, applications that are there. And finally, training of medical and paramedical staff is also something that's extremely helpful. And this is one of those applications there. So that's what I had in terms of the presentation I wanted to share today in terms of how technology is actually disrupting, the way how drugs are discovered, the way how diagnosis happens, early diagnosis and more precise diagnosis happens, and the way how therapy is actually being given in a more targeted, in a customized and a precision medicine, as we call, in a precise way, so that the outcome in the patient is also that much more, accurate. And at the end of the day, the patient should be detected early, treated early to have the best outcomes, and wherever possible, where there is a curative intent, to get a cure from that disease. So once again, thank you for a patient for listening. I hope this was interesting for you. And I hope India would basically develop a lot many new scientists and new healthcare enthusiasts that can actually contribute to how technology can be leveraged for furthering patient care. So thank you very much. So Ida, over to you. In case there are any questions, I'll be happy to answer.
Yes, thank you so much, sir. Yes, thank you so much, Sir Dr. Kartik Anantaraman, for your informative presentation. And next, we move on to next speaker, Prof. Dr. S. Duraival from Principal Guru Nanak College for Pharmacy, Dehradun, Uttarakhand, India.
Can I hear my voice, please?
Yes, sir.
Yeah, thank you. Very good morning to all the participants, and I am very much thankful to APR team and BioLeaks, the co-host Shravani, Hamid, and Anita. Really, you people are doing nicely, and my association with APR is since 2018. I'm traveling along with this big event and conference management people. I'm very much thankful once again. My concern topic of concern is nanomedicine. See, almost all the speakers who spoke previously have covered almost maximum informations about nanotechnology, drug delivery system, and other things and all. And but my concern is like I'm not comparing with the Indian medicine with allopathy or something like that, or the pros and cons, or like benefits or other things and all. Rather, I feel since I am concerned about, I am working on herbal medicine and nanomedicine, so I'm very particular about this topic. I hope I've got a lot of slides, but I don't think I can cover all the slides since I already got another assignment. My slot is little bit extended. Anyway, we have got no like nanotechnology. That is a widespread terminology. Nanotechnology that is not only involved in the pharmacy or pharmaceutical manufacturing. The nanotechnology will be used in almost all the fields, all the departments. So we cannot concise the nanotechnology, but we are making use of this technology to deliver the drug, or making the nanomedicine and making use of the benefits of this technology for the use of healthcare system. Well, what is this nanomedicine and nanomaterials? These are all the terminologies we are coming across nowadays. Nanomaterials, it can be like active pharmaceutical ingredients, or it may be like any other excipients that can be utilized for the diagnosis, or the monitoring the patient care, or it is for the treatment or therapeutic value. I said already know that nanomaterial is once again we can say that anything that is comprising between the size of 1 to 100 nanometers. The European Commission is defining this nanomaterial as the material that is natural or incident or manufactured. It may be aggregate or individual icon that is having the size of 1 to 100 nanometers. So the thing is like the size must be like distributed more than 50%. That this there is this within this number I'm talking about. Then we can call it as a nanomaterial, something like that. Okay, that is what the European Commission's definition. And World Health Organization and FDA, they are not very particular about the definition of this nanomedicine. or nanomaterials, they are all having their own. But what is the concern about this nanomedicine or nanomaterials in the healthcare system? So as you know, all the once the size is reduced, uh, then it has got the surface energy, surface charge. So the when the material is in bulk, its physical chemical properties are different. The same material is reduced to the size of nano range, then its physical chemical properties are changing widely or maximum. So that once again is no impact on the pharmacodynamic and pharmacokinetic parameters in when we are talking about drug delivery or therapeutic system. This nanomedicine, when what I'm talking about is Ayurvedic, uh, all basmas, they are all in nano range. See, these basmas may be like natural source, it may be mineral or the material that has got very toxic in nature, but they are making it in the process, various process, and making it like oxidized form, and it is going by its process in the nano range. It is not it is like it is reducing itself to that particular size ranges. So by virtue it is a medicine, natural medicine. I am working on Abrag Basma, that is silica, like a plastic. I mean, not plastic. It is um, mica. Mica. They are making so many processes on this mica and making it as a basma. That is called Abrag Basma. It has got rejuvenating property and more other properties. Even it is having anti-cancer activities, and we have worked on this Abrag Basma. Well, this ancient medicine, no, like when we are talking about Indian medicine, it is Ayurvedic, Siddha, and Unani. The Ayurvedic medicine starts from the like 5,000 years before, and it is a part of Rig Veda. Ayurveda is a part of Rig Veda. And my concern is how many of us are aware of these natural medicine or these Indian medicine. And I feel like we can have the balance of not only the allopathic medicine, we can also make use of this homeopathy and Indian system of medicine, so that we can make the healthcare system more efficient and less toxic and patient compatibility and so many other ways. Well, almost all the countries are using this natural medicine, and it starts from 10,000 BC and prehistoric period, and slowly it has been developed, and now we are making it as a like a what to say that uh, yeah, now we are using the even still we are using that natural medicine. Well, these slides are like how this the traditional medicine from the prehistoric or historic background has been brought over here, and how many what is the health organization is saying, and how many people the percentage of population using the this alternative medicine. See, all these slides are talking about this thing, like no. Well, once again, I am going to the see. These are the countries who are all using the traditional medicine. Still, they are in practice. Uh, so, my concern is when we are talking about nano. I said no, the nano range. The fundamental thing is
So please to all the participation to it.
So can you hear my voice?
Yes, sir.
Sorry, suddenly power shut down. Well, so I'm talking about the nano medicine and nano range, the 1 to 100 nanometer, the size of the particle. Uh, when we have discussed already, and all the other experts have discussed about these nano particles, uh, and then its impact on the health. So what is the concern about the three fundamentals of what I said is size and distribution and surface area. So these three which is managing this nanoparticles. When we are talking about the size, I said the size ranges must be from 1 to 100 nanometers, or it is 60 meters square by meter cube, centimeters cube. That is the way they are making it the size ranges. Well, then the size distribution. So this is polydispersed. We cannot say that all size, I mean, my material or particles are in the same size, but it will be distributed to 1 to 100 nanometers. Some of the particles are 10 nanometers, some of the are 80 nanometers, and maximum number of particles, how much the number of particles are in one particular range. So that way we can call it as a size distribution. So this distribution is having the great impact, whether it is a delivery system or uh, the nano delivery or nano, I mean, therapeutic way, how it is acting on. So this is distribution. Then the surface area, the surface volume, it is relational to the size and distribution. So we can make it all these three fundamental things that is very much needed for the nano materials or nano medicine. See, that which I talked about the Abrak Basma. We have worked on this Abrak Basma, and we subjected these Basmas to the various analytical techniques, because no, when we are calling about the natural medicine, no, the standardization is very important. As far as Ayurveda is concerned, they are having very rudimentary analytical methods to say that that is in this shape and this size, and if it is meeting that criteria, then they are calling that is attained the formulation. They attain the formulation of Ayurvedic formulations. So that way it is very rudimentary, or maybe it is not scientifically proved. In that way, the doubt will be created. So that is why we have subjected this into the Raman spectra, DX, then edX, and same so many other mass spectra, FTIR, and we have interpreted all these informations, like how this nanomaterial, the nanomedicine is behaving, what is its physical properties and physicochemical properties, and we have derived based on these different modern analytical techniques, and then we have gone for toxicological and even anti-cancer activities on pancreatic cells. My concern actually, my idea was to treat diabetics, and since it has got rejuvenating action on the cells, we thought of sending this material to the pancreas, then we can rejuvenate the beta cells, so that we can by nature we can treat the patients in that way. So that way we have decided, but later on we have changed it to the anti-cancer activities on pancreatic cells. So cell lines, we have used cell lines, and it has got a very good action on pancreatic cancer. And also, like it has having the less toxicity, even more than uh, 50 grams can be used. That is not having any toxic effect. But when we talk about it is mica, then but it is in a processed way. When we are it is having the minerals, that is toxic minerals or contents, that is but not having any toxic effect. Rather, it is having the anti-cancer activities and less even toxicological studies we have done. I couldn't display all these materials, since I rush through these last minute. I just added up the things. Anyway, so we have worked on already the abractmasma, and we concluded that the traditional medicine is having the value, and it has got its own nature of way of formulations. That formulations are not meeting the modern way of formulations, like in the nanomedicine. I mean, modern medicine, like liposomes or lipid nanoparticles, solid lipid nanoparticles. So they are making it in a very modernized way, like giving the active pharmaceutical ingredients in a nano size to treat the patients. But this by which these Ayurvedic medicines are by which it is in nano range, and it has got very good effect on the human health. So I think I can talk more on these. These are all the slides which how these ancient medicine are developed, and how different countries how they are utilizing using it, and even now all these countries which we have shown, no, they are all using this natural medicine. I'm not talking all these things now. So I'm concluding what are the concern about this nano medicine in a modern era. So we should there are a lot of awareness created in the public, and they are also coming to the this natural medicine for specific treatments. And I hope in future this will be included in the in a large way of treatment for the treatment of the human public. The concern is like whether it is one particular formulation I have made, like the toxicological studies thing and all, and like that so many other formulations in Ayurvedic formulations or Siddha formulation, Unani. So we have to go for searching it, like studying it, what is its effects and any toxic effects are produced, or how we can make the standardize these formulations in a modern technique, so that we can bring it out like in a very safe manner. All these formulations can be used for the treatment of various diseases. So the futuristic need is like the standardization and toxicological effect. And since it is in a nano size, the nano the biocompatibility, like the body has to assimilate, like it has to take up the materials, and it should not make any serious effect on the body, since it can be easily absorbed. There is no barrier for crossing the any biological membrane for this nanomedicine or nanomaterial is concerned about. So that way, like we have to work on the further toxicological studies and therapeutic way, so that we can explore more on this natural medicine for the benefit of the human public. So with that, I just conclude my talk, since I already have another assignment. So I thought of going by the time Hamid has called me and asked me to this. Once again, I thank all BioLeaks and APR teams for giving me an opportunity. In any other opportunities, we can talk very in a brief and in a broad way, like what are my studies, already my research work, and what other things are there in the Indian system of medicine. With that, I conclude my speech. Thank you very much. Thank you much, BioLeaks and APR team.
Thank you, sir.
Thank you. Thank you.
Yes. Thank you so much, sir, for your informative presentation. And next, before we continue the presentation by our keynote speaker, let's move on to the technical session. I welcome all participants and our session chair. Without further delay, please join me in welcoming our first author for the poster presentation, Jyoti Pharma, with the title of the paper, Lipid Nanoparticle Strategies for Modifying Pharmacokinetic of Central Nervous System Targeting Drugs, Crossing the Blood-Brain Barrier to Manage Neurological Disorder.
Good morning, everyone. My name is Jyoti Verma. I am a bachelor student at Galgodia's College of Pharmacy, India. Uh, Today, I am going to represent a poster about lipid nanoparticles, strategies to modify the pharmacokinetics of the cell to modify the curing the central nervous system neurological disorders, crossing the blood-brain barrier in strategies. Here we are now. I am going to share my screen. I am unable to share my screen due to the already shared screen.
All right, wait a minute. Prof. Dr. Duraivo, can you stop your share screen, please? Sorry, Prof. Duraivo, can you stop your share screen, please? Okay, thank you so much, sir. Let's continue for Jyoti Pharma.
Okay. Let's start it. I am audible.
Yes, it's visible.
Okay. Like brain diseases and the neurological disorders are leading to a burden in the healthcare system. According to the World Health Organization (WHO), brain diseases like Parkinson disease, Parkinson disease, Alzheimer, and a stroke are the major cause of the death worldwide, because the major uh, limitation of the treatment of the neurological disorders is difficulty to deliver the drugs to the brain, because as we know that our brain is protected by a blood brain barrier which protect the invading organism and unwanted substances. So through this poster, I will try to explain the some basics of the blood-brain barrier. So blood-brain barrier is formed by endothelial cells of the cerebral microvessels. So it acts as a barrier between the peripheral circulation, blood, and our central nervous system. So blood-brain barrier, endothelial cells of the blood-brain cells are the tightly junction. So the most of the drug molecules are not able to cross the blood-brain barrier. Uh, Basically, there are a drug delivery systems like local delivery system. Local in the local delivery system, we directly deliver our drugs. Drugs is API, active pharmaceutical ingredient, directly delivered to a brain by the injection or a intranasal administration. Intranasal administration receives a lot of the interest because it directly targets the brain, bypass the blood to the brain barrier. First of all, uh, when we take our medicines by a nasal activity, it diffuses through a olfactory nerves, and it get to the brain. There are the many physiochemical properties of the nanoparticles, but the nanoparticle can easily cross the blood-brain barrier due to its physiochemical properties, like its reduced size, due to its reduced size and a biocompatibility nature, non-toxic, and the mucus penetration. By this way, it can easily cross and and adhere to the affected area and starts to cure that area. After that, lipid nanoparticles are of types lipid. It can be a lipid nanoparticles or a polymeric nanoparticles, inorganic nanoparticles. Nanoparticle easily cross the blood brain barrier due to its the smaller size than a 100 nanometers. So it can be the concluded that the drugs of the encapsulated solid lipid nanoparticles or the nano carrier lipid efficient approach to circumvent the limitation of the blood brain barrier, as they facilitate the passage through it, improves the bioavailability of the drugs in the brain, and consequently drugs efficiently.
Jyoti,
Thank you.
okay, Jyoti, am I audible?
Hi, yes, ma'am.
Yes, ma'am. Can you please tell us about the applications where you are planning to apply for what kind of drugs and the indications for that particular formulation?
It can cure a lots of the neurological disorders.
Okay, can you give me an example of a drug for which you are planning for this formulation, or is it available already?
Actually, ma'am, I have no idea. I have uh, only given it information.
Okay. Can you show your poster once again, please? Please don't
Oh,
close
sure,
it before we end our conversation.
sure.
Okay. what are the pharmacokinetic parameters that are modified by this formulation? Which parameters could be modified? Pharmacokinetic parameters. Is my question clear?
Pharmacokinetic parameters may be uh, drugs we take, which can it can be the easily dissolve in a blood, but cannot crosses the barrier.
No, my question is about, you said, show your title once again. Yes. So you have mentioned it like lipid nanoparticle strategies to modify the pharmacokinetics of CNS targeting drugs. What are the parameters or what are the pharmacokinetic parameters that could be modified by these lipid nanoparticles? That is the question. Which parameter could be modified?
Ma'am, it would be the lipid structured.
Okay, Jyoti. Thank you. Please refer about that. Thank you.
See, one question to you. This is Dr. Padma. I wanted to know you've written for neurological disorders. So, what dose you are planning to try? Have you done any experiment, or it is just the review what you have collected? Hello.
Jyoti, are you listening? Jyoti?
Yes, ma'am.
Yes, Padma ma'am is interacting with you. Dr. Padma is interacting with you. Yes, ma'am.
Yeah, I asked you a question to whether you have written that this is study for the neurological disorders or neuro diseases you wanted to try. Whether you have done any experiment or it is just a review article.
Ma'am, actually, I'm unable to hear your voice.
You're not able to hear. I think my voice is loud or audible because I'm on mute, not on mute. Now you are able to hear?
Actually, I'm not.
Okay. Then we'll drop. Next participant we will take. Organizers, we can take the next participant. If any other sir is there who can, one more sir is there as a chairperson. If he's interested, let him also ask some questions. Dr. Dilip Kumar Bharti. Hello.
Hello. Sorry,
Okay, ma'am.
ma'am, I'm unable to hear your voice.
Okay, Jyoti ma'am has dropped her. Ma'am has planned to move to the next candidate. Thank you.
Okay, thank you, ma'am.
Okay, thank you so much, Beauty, for your presentation, and thank you for the chair for the question. And we move on to next presentation uh, with the author, Dr. Anita V, with oral presentation. With the title of paper is Standardization of Purification Process of Chrontum Ticklium nervalam.
Good morning. Can you hear me? Hello.
Yes.
So myself Tyagarajan, I am the co-author of this paper. So I am presenting now. Can I start?
Yes, you can start now.
Yeah, thank you. Can you see my screen? Is it visible? Hello.
Yeah, it is not visible.
Yeah, one second. Can you view my screen? Hello?
Yes, sir, it's visible.
Yeah, okay, thank you. So myself, R. Tyagarajan from Sri Sairam Siddha Medical College and Research Center, and my author Dr. Anita, and here we are presenting a topic on standardization on purification process of nair volume, which is known as croton tiglum.
Sir, excuse me, sorry for the interruption.
Yeah,
Can you please move to the slideshow, please?
yeah, sure. One sec. Yeah, can I start? Is it visible now? Hello?
Yes, sir, it is visible. Please
Yeah,
proceed.
thank you, thank you. So, Siddha system of medicine is one of the earliest Indian system of medicine to the mankind. There are a lot of formulation developed in Siddha system of medicine by various practitioners. Now, it is a global demand that modern standardized procedure was to be used to ensure the preparation, identification of the drug and purity and the shelf life of the medicine. So the global market for the Siddha formulations appears to be impressive now, but need of the art is to ensure the quality of the medicine. So here, Nervalam, which is also known as Proton Tiglam, is used in various Siddha formulations like Agasthyar Kurambu, Nervala Ennai, Ashtabhairava Matrai, Nava Uppu Melgu, and Kaushigar Kurambu. These medicines are mainly used in treating Vada Pitta diseases and mainly used for purgation. So aim and objective to determine the purity of the raw materials. Hence, the main aim of this presentation is to standardize the Siddha drug Nervalam, which is known as Propantiglam. So as per the Ayush guidelines, we conducted this study. Methods. In Siddha literature, purified Nervalam is compared with the Amritam, that is known as Amrita. So, unpurified Nervalam, which is not used in medicine preparation, hence it is purified and then used for preparation of various medicines. So, process of purification, first the Nervalam is collected and it is boiled in cow dung solution and then it is transferred to cow's urine and then boiled for couple of hours and then it is transferred into lime juice for boiling method. After boiling it 3 days and it is dried over the sunlight and hence the purified air volume is obtained. So purified air volume which is obtained by this purification is drained powder form and which is sent for analysis. This process of purification is mentioned in our so these are the physical characteristics of the nail bottom, which is solid, coarse in nature, rough, and blackish in appearance. So these are the physiochemical evaluation which were done. So it is insoluble in chloroform and ethyl acetate and soluble in ethanol, water, and DMS. So it is the final test report of the analysis of nail volume. So results and discussion. The values are which are found is normally to the API standards, and this is conclusion. Standardization of nerve volume was carried out by the analysis of hash values. So these are the values which are under the API standards. Hence, it is concluded that it is standardized drug. So these are the references of our project. So acknowledgement, I thank management of Sairam institutions, Dr. S. Kumar and Dr. V. Anita for this support, and I thank this opportunity for presenting in this conference. Thank you.
See, I would like to know what is the percentage of cow dung you have used, and
So
then
cow
why
dung,
cow urine is used. Why two are used? Because for purification, either one can be used, no?
yeah, ma'am. But in our literature, they mentioned the process. First, we want to purify in cow dungs, and then it should be purified with cow's urine.
Okay, what is the percentage of cow dung you have mixed?
So actually,
Any percentage you have
no, ma'am,
stabilized?
no. It is mixed with
Huh.
water and made into liquid form, ma'am. Then it is boiled. So no percentage.
Okay. No percentage. Just
Yeah.
like that, you have taken
Yeah, ma'am.
the cow dung and mixed with water and
Water and
added the
we,
seeds and boiled.
yeah, ma'am. We added the seeds in the
How
cloth
long you
and
have boiled?
tied about 1 hour, ma'am.
You have put the seeds in the cloth and tied
Yeah,
and put inside that
tied
cow
and
dung
the
water.
cloth, yeah, ma'am.
Mm-hmm.
Then it
Okay.
is cleaned and then transferred to cow's urine and last in lemon juice.
Oh, that is okay. Then how can you say that? Did you do any experiment to say that it is purified by this method and the without purification in cow dung and cow urine? What is the difference? Did you do that study?
No, already the papers are available based on the toxicity of the unpurified and purified. Many
Okay,
papers are
what
available.
toxicity it gives? What
Actually,
toxicity it gives?
arrow poison and it has many alkaloids. It is very toxic and poisonous in nature. So actually,
Alkaloids are all. Hmm. Yeah. Proceed.
yeah, without purifying, it gives more diarrhea or purgation and allergic reaction. After purifying, there is no allergic reactions. No.
But you have not done that study. You have just
Yeah,
standardized that with the
yeah,
help of. Uh, but see, when you are taking standardization, you are telling then you should standardize with respect to the marker compound.
okay.
You have just done hash value and total extractive values. That's all.
Yeah, we'll do it in future, ma'am. So we started now as a first project and we'll proceed.
Okay, fine. So, what you plan to use this purified for? What you are going to use?
So we prepare various medicines using this Nervalam, ma'am, like Agasthyar Korambu, which is used for purgation therapy. So before treating a patient, we give purgation to stabilize the vata. So on 3 doses, so we give as a medicine to stabilize, ma'am, for purgation.
Okay. Yeah, thank you.
Yeah, thank you.
Sir, is that given for all the patients who visit the hospital or only for the hospitalized patients? How is it?
So in our IP, we are giving this medicine, ma'am, based on the patient's age and the sex, ma'am. So we are not giving for all patients based on the patient's age and their complaints. We are giving this medicine.
Now, you have only studied. Now you have studied about the purification of the Nedvadam seeds, right, sir?
Yeah, ma'am.
And what is the mode it is administered? What is the formulation you give it to your patients?
It is like paste form, ma'am. We will give in early morning to the patients for the purgation. Combined with other medicines, we will give.
Okay. Thank you.
Okay, thank you.
Can go for the next presentation.
All right, ma'am. Thank you for Dr. Anita Fee for the presentation. And when we move on to the next author, there is Dr. Mayuresh Joshi with oral presentation with the title paper of evidence-based evaluation of Soneratia apetela bucham as hepatoprotectant. Okay, we will wait for 1 minute left for Dr. Mayures.
Excuse me, ma'am. Hello.
Yes.
Oh, okay. So I wanted to tell that Dr. Mayur Joshi has been stuck in some work, so he might be joining shortly. So I am there after him. So can I begin my presentation?
Oh, yes, of course, you can.
Okay. Should I share my screen?
Sorry, the update is Dr. Mayures is joining now. So please wait a moment.
Okay, okay, no issues.
Okay, please, Dr. Mayures Joshi, for your presentation about evidence-based evaluation of Soneretia apatella Buchham as hepatoprotectant. The time is yours.
Okay, I'll just share the screen.
Yep.
Please, may I know if the screen is visible?
Yes, it's
Is the screen?
visible now.
Okay, okay, I'll begin then. Sorry for the delay. First, I was on examiner at one place, so I've just taken a 10-minute off and I'm here. Here I go. Oops. So the topic for today's presentation, evidence-based evaluation of Soneretia abetala, Buchham as a hepatoprotectant. So, first of all, mangroves, we know mangroves are environment important. It's a very stressed habitat, and hence, actually, it has a lot of potential because secondary metabolites responds to stress. So recent studies have shown that they have a lot of amount of novel compounds, but of course, scientific data on mangroves is very less. That's the pharmacological importance of such plants. So the plants that were selected here was based on the literature which is available, the lack of the same thing. The traditional and documented medicinal claims, scientific absence of scientific data here. Okay, complement target mechanisms which were studied, and complete absence of standardized data on this said plant, which is Soneretia apetala. And why this plant? Again, traditionally, Sandeksha has been reported in tribal communities as an hepatoprotective agent, okay, and as treatment of hepatitis. But scientific evaluation of this plant has not been done. That's the reason why Soneretia is taken up. There is a subspecies of Soneretia cassiolaris, not from India, that has been studied to possess, hepatoprotective activity using Hg2 cell lines. On apetala, which is very common in Mumbai and Maharashtra and the whole coast of India, is not been studied. That's why the plant was selected. So this is the plant that I'm going to talk about today. So apetala, a medium-sized tree, India, China, Myanmar, Bangladesh, the eastern countries, and all that patch also has, you can see the IUCN database of the availability of this plant. It is not currently an endangered plant because it's available in plenty where it is available. But of course, being mangrove species, it is under stress of industrial usage also. So the proposed part of this plant is not the whole plant. The proposed medicinal usage is for the leaves. These leaves can be now repurposed, which has not been done. So the objectives were collection authentication, establishment of quality parameters, because there is nothing about standardization on this plant. Then development of an HPTLC fingerprint, marker estimation, because marker can be now repurposed to actually attribute the medicinal purpose that I will be proposing. A safety study in rat, in vivo hepatoprotective study in rat again, and finally the pharmacokinetics of which marker is working. All the methods have been USFDA validated, and all the studies have been authorized from an IIC committee through CPC-ACA, which is an international body. So this plant material was selected, collected from 7 locations, including Sundarban scannings for the purpose of the study. That it was validated, authenticated from a regularized and an authority which can authenticate plants. Quality control parameters were established for foreign matter, ash value, acid insoluble, water insoluble, loss on dryness, all these primary parameters. Also, the primary phytoconstituents were extracted from the plant. The presence or absence of this phytoconstituent was analyzed. And it was observed the alcohol content on the plants is very high. Heavy metal analysis was also done. It was found to be within the prescribed limits by Ayush. And lead content was slightly higher, but of course, it being a coastal area, the quantity was high, but it is within the permissible limits. Based on what was present in the plants, based on the constituents that were observed to be present in the plants, a fingerprint, HPTLC fingerprint was developed using a simple toluene methanol ethyl acetate 9.1.2 mobile phase and 10% methanol sulfuric acid as reagent. These are the photo documentation, and 5 markers with oxalic acid, beta-sitosterol, betulinic acid, betulin, and lupiol. They were simultaneously quantified using an ICH validated HPTLC method. By quantifying this HPTLC method, which was found that lupiol is completely absent from the plant, which was notified, but it is not there. And these markers, now selection of these markers was based on the pharmacological activity which has been targeted, which is hepatoprotective activity. So all of these markers are individually well known as hepatoprotective agents and well established as the same. Then extract was standardized to find out which extraction mechanism is actually going to function the best. And it was found that ethyl acetate extract is actually giving me highest amount of yield, which is 5.843 plus or minus 1.973 milligrams per gram from the various extracts that we selected. Further, this standardized method was validated as per ICH guidelines, industry-based ICH guidelines. The LOD and LOQ values for the material using HPTLC was found to be 1 and 5 mg per ml, 1 and 5 ppm. A linear range was established from 5 to 50 mg per ml. Regression equations can be observed for all 5 markers. This is a simultaneous experiment. So simultaneously, all 5 markers have been validated using ICH guidelines, the Q2R1 guidelines from ICH, and these are the related parameters. The validated method then was extrapolated to Swadeshi Patala, which was collected from all 7 regions against these 4 markers. As we discussed earlier, nuclear was found to be absent, and it was observed that the BKC sample is actually containing 8.13 milligrams per gram, whereas the Sundarban sample actually has lesser amount of markers. That's of course understandable. That also tells you that Sundarban is lesser in, it has lesser strain on the plant because biomarkers actually do arise from the biological strain which is applied on the plant, and hence the medical value also increases that way. Finally, once the extract was standardized, quality parameters were established. The standardized extract was then taken to safety experimentation. This is the letter of the IEC committee. Safety was carried out using standard OECD guidelines 420, single dose procedure, 2 grams per kg body weight of plant material. Extract was given to the animals, rats here, observed for 7 days post dosing and 7 days prior to dosing, and any deviation which is more than 10% was marked out. So we observed that there was no mortality observed, no toxic symptoms, no significant increment in body weight or food intake, water intake also. Cage-side observations were also found to be stable and within the permissible limits. Hence, the extract was then found to be safe for the efficacy study. This is the permission letter for the efficacy study from the IAC under CPC-SCA. Ethyl acetate extract or ethyl acetate was the standardized extract of the leaves of Swadeshi Patala. Now, why leaves? Because leaf is a renewable resource, and hence the plant is not directly going to get affected by this. Actually, the plant is going to be repurposed. So the protection and conservation aspect also is taken care of. If it was anything else, stem or root or something, then this can damage the plant. And again, this being a mangrove, such a damage is of course not advisable. So the leaves extract of ethyl acetate extract of Sonareshwar petala was taken, performed into three parts. You have 50, 150, and 300 mg per kg body weight. The whole study was divided into 7 groups. You have the first is normal control, then or the sham treated group, which received only distilled water. Then the CCl4 induced hepatotoxic group, the induction control. A natural recovery group, which was kept for 7 days post dosing, also to find out how naturally liver is recovering, because it's a CCl4 model. CCl4 model is an acute reversible model for damaging liver cells. The fourth was hepatotoxicity with a comparative positive control, which is silymarin. Silymarin is a well-established compound for hepatoprotection. And 5, 6, 7 were Sonareshwar petala at different dosages. Retroorbital plexus was used for blood removal. 6 per group animals were kept. 7-day study, and blood biochemical parameters were analyzed post after the end of the study. Both histopathology for light microscopy and electron microscopy was also carried out. So I'll go through the results for blood biochemistry. We have liver markers. One simple pattern which was always observed is normal control would actually show what should be there, and silymarin, the PC group, showed the most closest of this to this results. The plant groups SA1, SA2, and SA3 always showed a dose-dependent outcome, whereas the highest dose and the second highest, the 250 and 500 dose, showed very good results. Natural recovery, those showed slight amount of increment, betterment. They were not as strong as, of course, they were not as strong as treatment groups. These are the markers for AST, ALT, and AST, ALT, and ALP also, alkaline phosphatase, which is also required. And the asterisk symbolizes the significance rating, which was calculated using Tukey's test of parallel after done it. Then you have bilirubin, total and direct bilirubin access were done to analyze the condition of the liver tissue. Again, it was the same thing was observed. IC after induction, CCl4 damages the hepatoprotective cell and makes them more permeable. Therefore, a lot of bilirubin had leaked out. This is the total bilirubin count in B. Natural control shows that the bilirubin is coming down, but PC and then the SA2 and SA3 were very efficient in controlling the bilirubin, and hence reducing and reversing the condition of jaundice, which was the symptom of jaundice, which was initiated because of the CCl4 administration with paraffin. Then we have direct bilirubin, which is also calculated in the same fashion. Lipid profile was carried out for these doses. You can see 4 parameters in the lipid profile. You have cholesterol, you have HDL, which is the good lipoprotein, then you have TGs, triglycerides, and LDL. TGs is actually one of the most prominent problems in lipid recovery. Cholesterol and HDLs can be used when even LDL gets managed. TG is something which has always troubled, researchers who are trying to find out medicines and trying to find out a cure for liver conditions. But in our case, the TG, which was naturally found to be 51.69 UI per L, was found to be recovering very well in comparison and better than your PC control or silymarin control, which had the TG levels of 63.4 UI per L. Whereas in SA3, you can find the levels to be at 59.97, which is actually better than silymarin also. The other 2 groups were, and other parameters were found to be well in control and recovering quite nicely. We also did analysis for GGT and G6PD-H, which follows the same dose-dependent conditions, and protein and liver glycogen levels were also evaluated. These are, so once biochemical parameters are analyzed, you come to histology, and you can see this is the natural histology of normal control groups under 100x and 400x, where you can see the portal triad and the architecture as it should be. CCl4, the second photograph that you observe, CCl4 actually showed a complete disarray, and sinusoidal distortion had happened. Hepatic artery is punctured out, so you can see blood being leaking inside. Binucleated hepatocytes are very, very visible. This is natural recovery group, which shows kind of a recovery, but not so much. Positive control was the best recovery that we observed. And then you have the comparative low, mid, and high doses of for petala. You can see a dose-dependent increase, wherein the mid and the high dose especially shows an architecture which is much closer to what is found in the normal control group, suggesting that Sonareshwar petala in high dose of 500 mg per kg body weight is actually giving fantastic results. And this is a crude extract, which can, of course, be enhanced further in the study. To support the light microscopy, electron microscopy analysis was also done. This is the electron microscopy analysis and electron micrograms for normal control and CCl4 control. In the CCl4 control group, you can actually see the lipid globules which are formed inside the cell matrix also, suggesting the increase of lipid levels and lipid profile going very bad in case of the CCl4 infected induced group. Natural recovery shows minor recovery. Mitochondria are seen and RER is seen to be coming back, but Of course, not as well as it should. Positive control shows one of the best recoveries here, again at 25,000 times magnified. You can actually see mitochondrial number also increasing, suggesting that the cells have gone to regeneration and they are processing very well. The protein content has gone well. And in Sun ratio also, we can see RER coming up. You can see the 12,000x grouping showing a good RER matrix and mitochondrial network also coming up nicely, which suggests that good amount of recovery is going on. Glycogen molecules and globules can also be seen in the electromicrograph. That's again suggesting that Sonartia petala at the higher dose is actually showing fantastic results. Finally, just to prove that this thing is actually working, we need to correlate it with the markers and how they're behaving inside the body. That comes as the part of the pharmacokinetic study, wherein acetonitrile was used to do a protein precipitation from plasma. And from plasma itself, we have tried to find out post-dosing what is the content of the marker which is there. Again, this study has also been validated from CPC-SEA. These are the US FDA bioanalytical validation method validation parameters. You can see LOD, LLOQ range equations from 1 to 30 mul and a coefficient correlation of 998 for betulinic acid and 0.996 for ursolic acid, suggesting both of them are actually giving fantastic correlations and the method works fine. The LQC, NQC, and HQC levels were found to be 1.3, 5, and 24 mg per ml. These are the pharmacokinetic parameters. The whole animal group was, the sampling was done using something called as fast sampling techniques, wherein blood is withdrawn from the organism, animals, which are grouped into 4 groups. And blood is removed at 0 hour. You have 10 minutes, 15 minutes, 20 minutes, and 30 minutes, 1 hour, 2 hour, 4 hour, 6 hours, 8 hour, right up to 24 hours. And the blood which is collected, it is extracted, plasma is drawn out from it, plasma is purified, precipitation is done, and the established US FDA methodology using HPLC was used to estimate the content of betulinic acid and ursolic acid. Now, this whole study is to understand how is the molecule actually going inside and what is the speed at which it is actually working. So you have the first graph, which is about betulinic acid. You can see that it has reached Cmax in at 28.54 from the dosage that we had given. In just 20 minutes, at 0.33 hours, at 20 minutes, it has actually reached maxima in the blood. Whereas ursolic acid actually shows a dual blood pattern wherein it reaches Cmax of 27.94 at 2 hours, suggesting that the plant is working so well because it has multiple molecules which are reaching their Cmax at different times, and hence it is providing a long-lasting effect which is helping the recovery that can be seen. These are the parameters and HPLC chromatograms showing the presence of betulinic acid and ursolic acid. Glycyrrhetinic acid A was used as the internal standard to pre-validate the method as per US FDA. So, just to conclude, this study actually establishes hepatoprotective activity of Sonartia petala, which has not been done. So, scientific data is established for quality assurance also and further research also. The results shall assist to create scientific awareness regarding mangroves because most of the people look at mangroves as only for conservation for environment. But if the medical value of mangroves can be actually pulled out and actually established, then there is a better chance of getting pharma industries to actually work together and conserve the plants. So that's the whole reason behind searching for medical values. Now, this data can also be compiled to prepare a monograph. Observations justify the traditional ethnomedicinal usage also for hepatic disorders. So what has been done by tribals is correct. The evidence provides a rationale for the proper usage, and the immediately dependent population, the Korees and every population which is immediately surviving on mangrove land also, they can actually benefit by setting up a small control cultivation and regeneration units also, and crude extracts can then be used for formulations. These are a few of the references, and thank you so much. Any questions, if you have, please do ask. Shall I stop sharing?
Right, there's a online and questions are over.
Sorry, I can't actually hear. Can you please be that 2-3 people speaking? So I can't hear.
Yeah, Dr. Joshi, one question from my side is that already this plant fruit is already reported to have hepatoprotective activity. Even polysaccharide is isolated from this plant, which is shown to have a hepatoprotective activity. I'm correct?
Yes. But not from Apetala.
Then what is
Apetala
no, from
Apetala
Apetala
is
only.
reported.
No, from Apetala only
Okay.
the hepatoprotective activity is reported.
Mhm. So, ma'am, here
You can
the,
have
the purpose of doing this is because you see, fruiting also the leafing aspect, because regeneration is activated, it actually provides a better result. Though I have not done a comparative study between fruit and leaves here, the results that we have got on leaves are extremely promising.
Okay. Okay, that's all.
Thank you, ma'am.
Sir, you have collected samples from different parts of the
Yes.
country, correct?
Yeah.
Have you also taken samples from the Andaman and Nicobar Islands where we have a lot
No,
of
we could not reach for Andaman Nicobar directly because we need permission for that. We got permission on India land, and we have taken samples from western coast also and Sundarban.
Okay, fine. Uh, you mentioned that the extract is used in herbal medicine. Traditional, they have been using it as traditional medicine
Yeah,
among
yeah,
the
the
tribes.
tribals do use it.
Okay, is it only particular to the tribes, or only the tribes are using, or others in the country are also using as a traditional
No,
practice?
no. When we spoke to the people, who live around that area, those people told us about the tribal usage of these plants. People who come and in and out of mangrove land also. So no, in mainland, of course, we have not seen the usage of this plant.
Okay, thanks, sir. Okay, fine. What are the indications for which they have given it? Is there any idea about the indications for which this extract was given among the tribes?
Among the tribesmen, they are using it against jaundice. So they don't actually, they simply say kave. So any yellowing and jaundice, any liver condition, they give this plant. The leaves are directly crushed and given.
Okay, okay. Okay, thank you. It was a good presentation.
Thank you, ma'am.
We can go for the
Yeah,
next.
we can proceed.
Yes.
Yeah, but please ask the persons to restrict the time limit to 10 minutes. That is also very important.
Yes, ma'am.
Sure, ma'am. Sure, we will tell them.
All right, we go next to next author. There is Prerna Kanchandani with oral presentation with the title is Evaluation of Standardized Tripalakurna Extract as a Antigenotoxic Agent using Allium sativum cells.
Okay, so good afternoon, everybody. Is my screen visible?
Yes, it is visible.
Okay.
You can go ahead.
Yeah. So good afternoon, everybody. Myself, Prerna Kanchandani. I am currently pursuing BSc in Life Sciences from KC College, Efficiency University, Mumbai, India. And my research topic is evaluation of standardized triphala extract as an antigenotoxic agent, which has been done under the guidance of my mentor, Dr. Mayuresh Joshi. So to proceed. So as you all know that in the current city environment, we all are knowingly and unknowingly exposed to a myriad range of harmful genotoxicants that can lead to various health risks such as cancer, CVD, sterility, and multifactorial diseases, etc. Hence, a safe and commonly available, regularly used genoprotective agent is the need of the hour. One such well-known traditional and Ayurvedic formulation is Triphala Churna. Triphala Churna is a common and well-known health tonic in the Indian context, which is being used and readily available over the counter. The ingredients of this formulation have been reported individually to possess various medicinal benefits, including genoprotective potential. Thus, the current research work provides the scientific data for repurposing and repositioning the therapeutic utilization of Triphala Churna as a genoprotective prophylactic agent.
Right now, your slideshow is not visible.
not visible. Okay, just a moment. Is it visible now?
Yes.
Oh. So Triphylla churna consists of three plants, that is Phyllanthus emblica, Terminalia belrica, and Terminalia tabula. These ingredients were collected and used to prepare Triphylla churna as per AFI, that is Ayurvedic formulation of India. These are the aims and objectives of the current study. First, the ingredients were collected, authenticated, and processed, which is used to prepare the formulation. The quality was assessed in terms of powder microscopy, extractive values, and phytochemical detection. The presence of gallic acid was also established from the formulation. Finally, a genoprotective ability of in-house Triphylla churna was evaluated in allium sativum cells, that is garlic root cells, against the chemical mutagen, which is mercury chloride. These are the observations of the powder microscopic analysis. Your specific characters were observed in the ingredients, which were also be observed and present in the formulation. Thus, the powder microscopy is an important parameter to prevent an adulteration and assess the quality of the formulation. Further, for the presence and absence of major group of secondary metabolites was evaluated and the test was given by Khandelwal et al. It was observed that though certain metabolites which were not detected in the individual ingredients, they were showed their presence in the main formulation of the Triphylla churna. The metabolites are alkaloids, terpenoids, and saponins to be observed prominently. Here to standardize the extraction, the solvents before the actual assay, the extractive values of the ingredients and the formulation as well was evaluated in the solvents ranging from highly polar to non-polar solvents was done. And the distilled water and methanolic extract showed maximum extractive potential, which was followed by ethanol and hydroalcohol. Here, the methanolic extract was used for TLC, whereas the aqueous extract was used for the antigenotoxic studies. Here, using a liquid-liquid extraction separation method, which is by a softlet extraction, which was prescribed by J.B. Harvard. Here, the percent content of phytochemicals in the ingredients of the formulation was quantified, and it was observed that though certain ingredients and formulation are rich in alkaloid content, further it can be noted that numerous alkaloids have been reported to possess antigenotoxic potential. Here, as an important tool of quality standardization and evaluation, a rapid reproducible TLC method was developed for TLC fingerprinting of the ingredients as well as the formulation. These are the optimized chromatographic conditions for the fingerprint development. The TLC fingerprint was developed by using toluene-to-methanol as the mobile phase. It was done on the Merck silica gel TLC plate, and the photograph was taken before and after the derivatization. These are the photographs of the plate, which was pre and post derivatization. Here, the distinct band variation can be observed within the ingredients, which was labeled as 1, 2, and 3. These are the ingredients, and the fourth one is the main formulation. So here, distinct bands can be seen in one of the ingredients.
Good afternoon to all of you. I am Dr. Priya Shrivastava.
Sorry. So the bands can be seen in the ingredients as well as in the main formulation. Thus, these bands form the basis of the quality assurance and quality control tools. The presence and absence of specific bands are useful And they also assess the quality and the quality of the formulation which was prepared, and also prevent any adulteration. Here, the plate 3 shows the gallic acid marker, which was detected from the formulation, and it was observed at 254 nanometers without derivatization, and the RF was observed at specific wavelength. The track of gallic acid, that is of 20 ppm, was considered for the confirmation of this band to be a gallic acid band. After completion of the standardization process, the genoprotective potential of the aqueous extract of triple tuna, which was previously done and evaluated in the alien sativum garlic root cells. Here is the following protocol for the antigenotoxic studies. First, the garlic bulbs were grown in regular water and the mitotic index was established. It was divided into 3 groups. The group 1 consists of normal control. The group 2 was induction control, and the group 3 is simultaneous treatment. So here in group 1, at 9:30 the roots were cut and fixed in carnose A solution, and for the induction control, 1 hour treatment with 1 ppm solution of mercury chloride was given before the roots have been cut and fixed in carnose A solution. And for the simultaneous treatment in the group 3, 1 hour treatment of triple churna extract and mercury chloride was given in 1:1 ratio before the roots were cut and fixed into the carnoid A solution. So these are the results. A total of 470, 492, 431 cells were well separated.
Sorry, Pranana. Sorry for interrupt you. Your slot time was over. Can you close your presentation?
Can I just conclude in a moment? Hello.
Yeah.
Can I just conclude in a minute?
Yes.
Okay. So thus our current research work focuses on the triple tuna which was prepared and standardized. The study presents that a significant evidence with regard to the genoprotective activity of the triple tuna, and this study also provides regards to the genoprotective activity of the triple tuna, and it also opens the lead to prepare a stronger proprietary formulation using the established triple tuna data as a baseline. Here are the references, and thank you.
What's your conclusion? Yes,
Hello.
ma'am, you can proceed now. Padma ma'am.
Hello.
Yes, ma'am.
Hello.
Hello.
Prerna, can you hear the
Yes.
question?
No. The question is not being audible.
All right.
You were speaking about Tirupala Churnam
Yes.
formulations, preparation and standardization. Then you studied about the activities also.
Yes, I have performed. Basically I have done the research project wherein I have done the standardization and preparation of the triple tuna by myself.
Okay, this Tripala Churnam is available. I mean, it is very well established the use of this churna is very well established in Indian system of medicine, right? What is the purpose of choosing this particular churna? Any uh, lacunae in the scientific data available?
Uh, it has various medicinal properties, as we all know, and but wholly combined Triphala Churna was not been put up as a genoprotective potential, as it has a genoprotective potential. So, in my research study, I have seen there are three ingredients in this project in this Triphala Churna, and some one or two elements shows the potential of genoprotective potential. So that is the main point of choosing Triphala
Okay, how
Churna.
are you planning to take your research further?
I am basically using this data as a baseline to repurpose the value of Triphala Churna as a genoprotective agent.
Okay, Prerna, thank you.
All right, that's it for Prerna presentation.
See, Prerna, I wanted to know, one second, I'll one question for her. See,
Yes,
this
of course,
Trifla
ma'am.
Churna is known, people are taking from long time. So what is the use of finding anti-genotoxicity? Because if it is toxic, people would not have used. This is a common churna which is used very, in Ayurvedic, if you go for any treatment, this churna is prescribed. Many doctors prescribe this churna as a digestive, as a immunity booster also. So why you have done genotoxicity now?
Ma'am, so my project is basically on the anti-genotoxicity study, and as you all know that in the current city environment, we all are exposed to many harmful pollutants, carcinogens. So that made me strike out that I should do something on the previous data, which is already a well-known Triphala Churna, as it has various medicinal properties and it is Ayurvedic as well.
Okay, fine. Okay, we can proceed with the next person, next presenter.
Okay, thank you, ma'am. We move on to next presenter, Dr. Priya Sirvastafa, with our presentation with the title of Targeted Intracellular Delivery of Anti-Tubercular Bioactive. There you go.
Good afternoon to all of you. I am Dr. Priya Srivastava from Lakshmi Narayan College of Pharmacy, Bhopal, MP, India. The title of my oral presentation is Targeted Intracellular Delivery of Antitubercular Bioactives via Transferrin and Mannose Functionalized Nanoliposomes, its formulation strategies, ex vivo and in vivo assessment. Moving on to the next slide. So we all are aware about tuberculosis. What is tuberculosis? Tuberculosis, a ubiquitous and highly contagious granulomatous bacterial infection, is a leading killer worldwide. It is caused by an opportunistic infectious intracellular pathogen, which is Mycobacterium tuberculosis. It is one of the most pestiferous and enduring human pathogens, killing millions of infected people every year. It is an acid-fast bacilli. Two of the major problems associated with TB and which are closely related are lack of adherence to therapy and the emergence of multi-drug resistant strains to antibiotics. To circumvent these challenges, it is essential to improve the antitubercular activity of the existing bioactives by combining them that have different mechanism of actions to attain synergistic effects. To some extent, DOTS therapy addresses the problem. However, the currently available bioactives are only partially effective owing to the impermeable nature of the mycobacterial cell wall and the aptness of the MTB to develop resistance. Furthermore, the lengthy and prolonged treatment duration and the uses of the high doses of the bioactives results in severe side effects and patient non-compliance. The conventional dosage form-based therapy often fails to establish and maintain therapeutic level at the target site. On the contrary, non-specific distribution of the bioactives to the non-target sites results in contraindicative manifestations. The factors like rapid clearance, short plasma half-life, poor water solubility, and low bioavailability further limit the effectiveness of the bioactives. So, these clinical challenges could be addressed by ingenious designing and developing novel strategic drug delivery modules. This is the research advised. The encapsulation of antimicrobials or chemotherapeutics into liposomes can enhance their activity while minimizing toxicity. However, their use is still limited owing to ineffective and inadequate loading strategies. Several bioactives which are non-ionizable and poorly aquasoluble cannot be easily encapsulated into aqueous core of liposomes by using conventional means. Such bioactives, however, could be encapsulated in the liposomes by forming their water-soluble molecular inclusion complex with cyclodextrins. In this study, rifampicin and 2-hydroxypropyl beta-cyclodextrin molecular inclusion complex was developed. The rifampicin and 2-hydroxypropyl beta-cyclodextrin interaction was assessed by computational analysis molecular modeling. In this study, we worked on two antitubercular bioactives, rifampicin and isoniazid. The HP-beta-CD rifampicin complex and isoniazid were co-loaded in the small unilaminar vesicles in the aqueous core of the SUVs. Further, the developed system was functionalized with transferrin and mannose, that is para-aminophenyl alpha-D mannoside, and was extensively characterized. The molecular docking tool software were applied to analyze the orientation and interaction of the bioactives moiety rifampicin and 2-HP-beta-CD. The interaction was analyzed by using AutoDock Tool 2016 software. The docking analysis revealed an estimated free energy for binding for the best fit. The docking score was minus 5.9 kilocalorie, which is significantly good. Thereafter, the molecular inclusion complex of rifampicin and 2-hydroxypropyl beta-cyclodextrin was prepared by solvent evaporation and freeze-drying method and extensively characterized for solubility study, for FTIR spectroscopy, for NMR spectroscopy, for XRD analysis and phase solubility and other DSC analysis. Thereafter, the process variables were optimized for the preparation of the SUVs using the response surface model approach using a 3-factor 3-level box Pancon design. 3D response surface curve and 2D counter plots were plotted and it showed that the independent variables have a mixed effect on the vesicle size of the developed small unilaminar vesicles and the polydispersity index. Thereafter, small unilaminar vesicles were prepared using various lipids like DSPC, cholesterol, and DSPE in the ratio 2:0.8 ratio 0.2 by using lip and thin film hydration method and functionalized by transferring using carbidimide chemistry. For another formulation for mannosilated SUVs were prepared by using DSPC, cholesterol, and DSP PGNS2 and functionalized with para-aminophenyl alpha-D mannoside through a Schiff base or imine formation. Further, the developed formulation was extensively characterized for transmission electron microscopy. The study revealed that the developed nanoliposomal formulations were spherical in shape. However, on transfer and conjugation, it was observed that the TEM image of the SUVs appeared opaque, spherical, or uniform in size. The opaqueness of the electron beam may be due to accounted for the effect of transferrin surface conjugation. Further, the development of SUVs was also confirmed by atomic force microscopy. There was no visible aggregation or fusion of the vesicles seen in the photomicrographs, indicating the method selected for the nanoliposomal formulation, transferrin mannose conjugation, as well as the bioactive encapsulation did not influence the liposome's morphology or shape. The average vesicle size of the plain SUVs and transferrin functionalized SUVs was found to be 138.8, 0.11 nanometer and 201.5 nanometer respectively. The average vesicle size of TF SUVs was marginally larger compared to plain SUVs that could be ascribed to transferrin surface conjugation. A low polydispersity index less than 0.3 was obtained for both the formulation, indicating the narrow size distribution. The formulation was characterized for the zeta potential also. Similar pattern was observed in the case of mannosilated SUVs also. The formulation was characterized for the FTIR spectroscopy, NMR spectroscopy. The drug entrapment efficiency, the formulations were studied for the drug entrapment efficiencies also. The plain SUVs could entrap 48.12 plus minus 0.1% for INH and 50.34 plus minus 1.2% for rifampicin. However, for TF SUVs, the percent drug entrapment is quite low because of the decreased in the percent entrapment of drugs was marginally decreased in the case of TF SUVs. It may be due to the loss of drug during functionalization. The formulation was further studied for the drug release. The dual drug loaded SUVs demonstrated a simultaneous, however, sustained release of both the drugs over a protected period of time. The relatively slow release of either of the drugs from TF SUVs as well as MN SUVs as compared to plain SUVs could be due to transferrin conjugation or mannose conjugation that constituted an additional barrier to the drug's diffusion. Further, the ex vivo studies were performed on raw 264.7 macrophage cells. The results of the cellular uptake studies of the different formulations, pre-rhodamine solution A, rhodamine labeled plain SUVs, and rhodamine labeled TF SUVs using flow cytometry were recorded. There was a substantial difference recorded between cell-associated fluorescence measured after incubating raw 264.7 cells with plain SUVs and transferrin functionalized SUVs at 100 micromolar concentration for 1 hour, 4 hour, and 24 hour. All SUVs demonstrated significantly higher mean fluorescence intensity in comparison to free rhodamine solution. However, TF SUVs exhibited yet higher mean fluorescence intensity in comparison to plain SUVs and free rhodamine solution. This convincingly signifies the role of transferrin as a targeting moiety that could improve the rate and extent of macrophagic uptake and accumulation of the SUVs through receptor-mediated endocytosis. Similar pattern was observed in the case of monocylated SUVs also. The intensity of fluorescence was significantly higher in the case of monocylated SUVs as compared to plain SUVs.
1 minute remaining.
Yes, 5 minutes. Can you give me extra 5 minutes? I'll cover in.
I will give you 2 minutes. How about it?
Okay. Further, the formulation was characterized for the confocal laser microscopic studies, and the results are well nice. And both the formulations were characterized for cellular competitive inhibition studies, assay also. And further, because there's a short of time, so I can conclude. The formulations was characterized for superoxide generation. The animal studies were also conducted for the bronchoalveolar lavage study and the intracellular biodistribution studies and the in vivo antitubercular activity study. So all SUVs formulations tended to accumulate in macrophages involving active as well as passive uptake. The results are comparable for transferrin conjugated as well as monocylated SUVs. There was a significant difference in the intracellular fluorescence accumulation in case of both the SUVs, transferrin and monocylated. However, on antitubercular activity evaluation, the antitubercular activity was recorded manifold higher than plain SUVs as seen in the CFU count. The cell-specific accumulation in the case of transferrin conjugated SUVs was higher than monocylated SUVs. They exhibited the high potential for phagosomal uptake as well as reflected in antitubercular activity. This could be possible due to subsequent intracellular transportation of the contents of the static endosomes to the phagosomes. However, this needs an appropriate study design for the confirmation of the mechanism involved. Thus, it is concluded that the developed formulation succeeded in macrophagic targeting of both the drugs, possibly with subsequent phagosomal delivery, thus resulted in manifold effective antitubercular activity when compared with the plain drug combination. This establishes the therapeutic as well as clinical need of the combination drug therapy targeted to the phagosomes of infected macrophages, could seemingly eradicate the intracellular bacterial burden. The approach oblique strategy may be inseminal in treatment and management of tuberculosis and also in its eradication when given in low doses for a short period of duration, thus ensures both therapeutic efficacy as well as patient compliance. This is a reference. Thank you for giving me such wonderful opportunity to present my research work here.
See, regarding anti-tuberculosis activity, you
Yes,
have done this rifampicin, correct?
Yes.
What is the dose which you try to do in the encapsulation method, whatever lipophilic SNV,
Ma'am,
whatever you
5 to 6 milligram per kilogram of the body weight.
5 to 6 milligram?
Milligram per kilogram.
Please.
That's it, ma'am.
Ma'am is not audible, I guess.
Myself, Dr. Ponmalar, can I present, ma'am?
Yes, ma'am, you are audible.
Okay, thank you, ma'am.
Yes, ma'am.
All right, can we go for the next presentation?
Yes, ma'am. I am presenting, ma'am.
Oh yeah, all right. Shenawali.
Okay, thank you, Dr. Priya.
Thank you, ma'am. Thank you so much.
Ma'am, my screen is viewing.
Hello.
Yeah,
Hello.
it's
Okay, ma'am. Myself, Dr. Ponmala, Professor Sri Sairam Siddha Medical College and Research Center, West Tambaram, Chennai 44. My topic is antispasmodic activity of some herbs commonly used in Siddha system, a comprehensive review. The main aim of this review is to focus on the medicinal potential of plants possessing antispasmodic activities with their proposed mechanism of action. Methodology. Siddha is a form of holistic medicine that is focused on promoting. Yes, yes, ma'am.
Ma'am, sorry, your this time is presentation with Shenawali. You after Shenawali.
Okay, ma'am. Thank you, ma'am. I will wait, ma'am.
Thank you so much.
Okay, okay.
Shenawali, are you in this room?
Can I start sharing the screen now?
All right.
Can I start, ma'am? Hello.
Yes, please.
Good afternoon, everyone. I'm Sneha Wali, and today I'm going to present the e-poster on topic development of marker-based methods for Diospyros paniculata extract and assessment of antitubercular activity. It's my research, farm research work, which I'm going to present here. Starting with the introduction beginning, the tuberculosis, as we all know, it is a chronic infectious disease which is caused by Mycobacterium tuberculosis. It is spreads through inhaled infected the infected droplets by the healthy person, and it still remains one of the primary causes of mortality and morbidity. And it is reported, the statistics mentions that more than 10 million individuals are infected with TB for the first time in every year. So no doubt there are many therapies and medications available which are reported against our tuberculosis, but there are many side effects associated, and there are drug resistant strains available, and there are also high prevalence TB. That's why there is need to find out drugs which are from natural source that have fewer or less side effects. There are many natural products such as curcuma longa and cannabis sativa, which are proven to show antimicrobacterial activity. The plant which I've chosen for my work is Diospyros paniculata, which is commonly known as panicle flowered ebony, and it belongs to the family Eboniaceae. It has various phytoconstituents, as mentioned, which relates to the biological activity, which are remarkable and significant. Coming to the need for study, solving the problem, why we have chosen this, there are many species under the Diospyros genus which have shown to have and reported the antimicrobacterial activity, but there is no evidence related to the plants which have chosen. And there are many chemotaxonomical biomarkers such as betulin, juglone, plumbagin, diospyrin, etc., related to Diospyros genus, which we can use for standardization, which will help in the identification of the plant material which we have chosen. So among this, juglone and betulin are the two biomarkers which I have chosen, and they are generally present in among all the genus of the Diospyros species. And there is no evidence that there is antimicrobial activity performed on this plant extract. So we have performed that. Coming to the materials and methods, the two biomarkers, juglone and betulin, were procured from Yuka Enterprises, Mumbai, and all the solvents used were of respective grades. First, coming to the collection of plant material, here we have chosen two plant materials, parts of the plant, that is leaf and the bark. It was collected, authenticated, and all the preliminary process like shade drying, powdered and all, and extraction was carried out by hot solvent method using methanol solvent. And the extract which were obtained, that means we have got two extracts. One is from leaf and bark, that was fractionated according to column chromatography and Koss et al. Method. Then for the extract and the fraction, preliminary phytochemical screening, that is the TLC, and chemical tests were performed according to the standard procedure. The extracts obtained were in vitro study was performed according to the MABA assay. Analytical work for quantification and method development was done by HPLC and HPTLC according to the instruments and the mentioned softwares. Coming to the result and discussion, the extracts revealed the presence of various secondary metabolites or the phytoconstituents. For fractions, I've done the test only for naphthoquinones and steroids and triterpenoids because our juglone and betulin belong to this phytoconstituent category. Then preliminary qualitative TLC was performed for the fraction because we have taken fraction for the analytical study. The extract were evaluated by MABA assay for their antimicrobacterial activity, where the bark and leaf extracts showed 50 and 25 microgram per ml respectively, while compared to the standard isoniazid, that is 1.6. Then coming to the HPTLC and HPLC, which was performed. HPTLC was performed like for juglone, we have developed the method and quantified.
Sorry, ma'am, the time is over.
Ma'am, 10 minutes are over so early.
Yeah, I mean 1 minute remaining.
Ma'am, I think 10 minutes were allotted, right, ma'am?
Yeah, 10 minutes.
Just 1 minute, I'll come.
Oh, yeah, all right.
Thank you, ma'am. For HPTLC, juglone was developed, the method was developed and quantified, and for HPLC, both juglone and betulin, the linearity range, LOD, LQ, and regression coefficients are mentioned. The amount of juglone quantified is mentioned here for HPTLC. For juglone HPLC, here it is mentioned in bark and leaf fraction. And for betulin, bark and leaf fraction, it is mentioned. Various trials were carried out, and the final mobile phase selected for juglone in HPLC was methanol water 50-50, and betulin is acetonitrile water 90-10. And for HPTLC, juglone is toluene and ethyl acetate 8:2, volume by volume. Coming to the summary and conclusion, whatever chemical test and TLC was performed, it detects the presence of various phytoconstituents that are present. And by performing HPLC, HPTLC, we have developed a method for juglone and betulin, and whatever amount of juglone is quantified by HPTLC and HPLC is almost same. And the extracts have proven to show the anti-TB activity. These are the references. I would like to thank KLE College of Pharmacy and BSRC Belagavi. Thank you.
Can you please explain how was the anti-tubercular activity carried out?
Yes, ma'am. It is according to the MABA assay, where the strain which was taken was H37RB Mycobacterium tuberculosis, and the broth which was used was Middlebrook 7H9 broth. And then the plates which were taken, ma'am, the serial dilution was done, and then paraffin was covered and incubated for 5 days at 37 degrees.
Okay, sorry. I thought it's you. Okay, bye.
So can I continue, ma'am?
Yes, please proceed.
Then for 37 degrees for 5 days, and then serial dilution was done for standards also and extracts also. Means first we have taken 0.8 microgram per ml, then double 1.6, 3.2, like that way till 100 microgram. And then it was incubated for 5 days, after which 25 ml of freshly prepared Almar blue reagent was added, ma'am. and then twin 80 was added and incubated for 24 hours again. So as in the image it is shown, pink is resistant and S is sensitive. Means pink means there is growth. Blue means there is no growth of the bacillus. For bark extract it was 50 and leaf it was 25.
Okay, Sneha, thank you.
Thank you, ma'am.
Okay, that's it, I think. Can we move to next presentation?
Yes, we can.
All right.
Padma ma'am is there.
There's no
Sorry?
question. You can do further.
Thank you.
Thank you, ma'am. All right, thank you, Sanawali. And we move to Anurati Basak with poster presentation. Anubrati Basak, you are in room now? Okay.
Hello.
Hello.
Yeah, the last presenter title, which number is that? Because the title said anti-tuberculosis, but I'm not seeing number 6 as anti-tuberculosis.
Ma'am, shall I share my screen again?
You are Sneha, na?
Yes, ma'am. I have presented just now.
Just now Sneha Wali is presented, but your title which is given for the reviewing is different. Method development, validation and quantification it is given.
Yes, ma'am. Can I share my screen again?
Yeah, you can share just once, then we'll see.
Yes, ma'am, sure. Ma'am, here it is. Development of marker-based method.
1 second. Development of
marker-based method for Diospora spaniculata and evaluation of its anti-tubercular activity.
Correct, but fine. I'll talk to them. No problem. You can. The other speaker can speak.
Thank you, ma'am.
All right. Can we go next presenter? Sanskriti Saini.
Hello.
Yes.
Hello. Am I audible?
Oh.
Yeah, it's Anuprati Basak.
All right. Anurati Basak. Okay.
Yeah. Can I start?
Yes. You can start.
Okay, a very good afternoon to all of the delegates present over here. I'm Anubrati Basak, an informed student from Pharmaceutical Chemistry, presenting JSS College of Pharmacy from Ooty. So my project is about the phytochemical and in vitro neuroprotective evaluation of Verticillium dalsei leaves. So we are all familiar with the disease that is neurodegenerative disorders that can be related with Parkinson and Alzheimer disease. For my project, I have particularly focused on Alzheimer disease. For that, I have chosen the plant that is Verticillium dalsei, and I have extracted the plant in the different extracts such as the N-hexane extract, the chloroform extract, ethyl acetate extract, and 90% V/V of ethanol extract of P-dalsei. And for that, after that, I have run the confirmatory studies, which is the preliminary studies of alkaloid and flavonoids and so on. Starting with the aim of my
project. The aim of the present research work is to investigate the phytochemical characterization and neuroprotective evaluation of the leaf extract of my desired leaf, that is Pathosodium dalsi. What I have done, first I have collected and authenticated of plant material that I have collected from the Pachukottai district of Tamil Nadu, and it was confirmed by Dr. Sija, Associate Professor of Department of Botany at the Government Arts College in Uti, and we verified the plant's authenticity and kept the specimen in the Cognosy lab. After that, extraction of plant material was done in two different extracts. These extracts were separated by the Soxhlet apparatus with inhexane, chloroform, ethyl acetate, and 90% of the V by V of ethanol. And all the extract was subjected to the preliminary phytochemical test and in vitro free radical scavenger studies. Next up is the preliminary cytochemical studies. We have made sure that all the extracts are subjected to the quality test of identification of the constituent like alkaloids, flavonoids, phenolic groups, tannins, saponins, carbohydrate, terpenoids, etc. Next up come the in vitro free radical scavenger studies. All the extracts of the P. dulce leaves were subjected to DPPH radical scavenger studies. Next, for the estimation of the total phenolic and flavonoid and alkaloid group, based on this phytochemical in vitro free radical scavenger, the ethanolic extract was selected for the quantitative estimation and as it shows the good results. After that, isolation and characterization of the leaf extract of P. dulce. We have done this isolation by TLC method and flash chromatographic method. For both, we use the mobile phase same, that is toluene and ethyl acetate, and the ratio of 9:1. And for the TLC, we have got 5 spots for the ethanolic extract. And in flash chromatography, we have got 3 fraction of region which imparted same color, and that are 10 to 13, 15 to 21, and 26 to 33. The next we did is what to subject this 3 fraction to the LCMS study. Here are all my all my results. The LCMS study shows that there is a constitution present of camphorol and then catechin, polyphenols, linoleic acid, and etc. in this extraction of ethanolic. Next is the part 2 that I have done is the in vitro studies. These determine the toxicity effects by using MTT assay for studying the Alzheimer's disease model. In that, I have used the medium DMEM and I have used the cultural medium for FBS in 96 well tire plates. And the same line that I've used is SHSY5Y neuroblastoma cell, which have a positive effect in causing Alzheimer's disease, and the extract was ethanolic extract of P. dulce leaves. Next, I have done the cytotoxic study, which shows the result. I have choose the standard drug that is memetine, and which shows the IC50 value of 64. Among all these values, the IC50 value of ethanolic extract was 96, which is the optimum value and shows a good effect. Result and discussion. By concluding my e-poster, we have find that above all the extract, the ethanolic extract of the dulce leaf shows a good effect for Alzheimer's disease, and it also causes free radical scavenger effect. We have isolated the constituent by TLC and FC, that is flash chromatographic method. The flash chromatographic method shows 3 fractions, that is 10 to 15, 15 to 21, and 26 to 33. All the 3 flash chromatographic method was then converted into the conveyed into the LCMS technique. Here we found presence of polyphenolic compound in the ethanolic extract of P. dulce leaves, and this ethanolic extract of P. dulce leaf was subjected to in vitro cytotoxic studies for Alzheimer's disease, where we chose the cell line of neuroblastoma cell, that is SHSY5 cell, and which study shows the viability of the study shows significant Alzheimer activity in ethanolic extract, where memethamine was stored as the standard drug. So reference, I have take the reference from Jagadish K. Kumar, who has done Indian Journal in Pharmaceutical Science about the topic antioxidant states. And thank you. This is all my e-poster.
There's a question, ma'am.
Yes. Can you explain about the in vitro studies you have carried out?
Yes, ma'am. Ma'am, I have done the in vitro studies where I have done the cytotoxic studies by MGT assay, where I have taken the Dalbuco modified eagle medium with the cultural medium as the FBS cell, where I found that it was able to convert the insoluble formation to soluble formation, which we can tell the viable cell concentration. And for that, the ethanolic extract gave a IC50 value of 94, which is very close to the standard drug, which gives the IC50 value of 64. So, ma'am, that's why we chose the ethanol one. That study I've done in vitro. And another, I have chosen the neuroblastoma cell, which is SH5 Y cell, and where it also shows a good IC50 value on the ethanolic extract.
Okay, understood. Thank you.
That's it, ma'am.
Yeah, that's all from my side.
How you chose this plant for the Alzheimer's disease? Any basis was there for you to choose this?
Yes, ma'am. This Alzheimer has a good antioxidant activity, and for Alzheimer, as we know that, old people are like most of the people, most percentage of the old people are having neurodegenerative disorder because of free radical formation, and which leads to the
Correct, yeah, free radical
neurodegeneration.
formation is there, I understand. But
Yes,
because of
ma'am.
antioxidant, as if you take that, then all plants can be used for Alzheimer disease. Because most of the plant will have antioxidant free radical scavenging activities there, very good activities are there. Okay, fine, we can proceed with the next one.
Okay, ma'am. Thank you.
Okay. Thank you so much for your presentation. And we go next for Sanskriti Saini. Are you in this room? Sanskriti Saini
Yes,
with the. All right. The time is only 10 minutes.
Is the speech is available?
No.
Yes, ma'am.
Your screen is not visible.
Okay. Is this available?
Yes.
Okay. Good morning to good afternoon to everyone present here. I'm Sanskriti Saini, M.Pharm student from Kurukshetra University, representing my review e-poster on harnessing therapeutic potential of transparent targeted nanocarriers in cancer therapy. So, as we all know that cancer represents a significant part of global mortality rate, and it offers requires the higher dosing medications. Cancer itself is a complex interplay which further is a the main risk factors which includes in the cancer is the lifestyle modifications, environmental factors, and the genetic mutations. So there are various and substantial available potent anti-cancer medications uh, like surgery and chemotherapy, radiation therapy, and hormone therapy, but it's still hindered by its selectivity and shows various adverse effects like low toxicity, solubility, and stability. So, to overcome all these challenges and to mitigate off-target effects, which is drawn by the conventional therapies, nanotechnologies and nanocarriers become a spare part to overcome all these challenges. And in this context only, the active targeted nanocarriers selectively demonstrate the therapeutic efficacy more than the passively target counterparts. So in this context, the transferrin ligand and transferrin conjugated nanocarriers uh, becomes more efficacious than the other ligand conjugated nanocarriers. So transferrin receptors, which is a transmembrane glycoprotein, already overexpressed in the malignant cells. And firstly, the transferrin conjugated nanocarriers initiate the binding process and interact with the receptors interact the transferrin receptor which is already overexpressed onto the cancer cells. Further, it recognizes the receptor site and binds to it which leads to the subsequent release of the therapeutic agents of that is the anti-cancer medications within the cancerous cells. So there are many therapeutic payloads like nucleic acids and the mRNA, which further bind to the nanocarriers and shows its effects. There are various advantages of using the transparent conjugated nanocarriers, such as its reduced toxicity, its prolonged the circulation of therapeutic payloads, enhance the cellular uptake, and improve therapeutic performance, reduce the off-target effects after targeting. One of the major aim to of selecting a transferrin conjugated nanocarriers for cancer therapy that it only targets the malignant cells and it reduces off-target effects to the healthy tissues. So with this, I conclude that transferrin conjugated nanocarriers are effectively delivered to the target cancerous cells and not to target the healthy tissues. Uh, so due to the transferrin binding affinity for the cancerous cell receptors and its enhanced efficiency and selectivity for the drug delivery. So though this approach helps mitigate adverse effects, offer the significant potential to enhance the treatment outcomes. Uh, uh, these are the references, and these have shown that transferrin conjugated nanocarriers, how it is recognizing the transferrin receptor, which is overexpressed and in the cancerous cells, its binding stage, and as the enters pass, how it targets to the cancerous cells and exhibits its anti-cancer action, and these all are the benefits which is exhibited from the transferrin conjugated nanocarriers. Thank you.
Sanskriti, you have presented a review, right?
Yes, ma'am. I have presented the review for e-poster.
Okay. Have you carried out any study or have you formulated this nanoparticle?
Not yet, ma'am. I have represented the e-poster, so I have go through the literature survey, which I have found that the already the lactoferrin targeted the other ligand, which is the lactoferrin, and its conjugated nanocarriers is used in the cancer therapy, and also this is also used in the cancer therapy, but not only to the limited cancer cells, which is the breast cancer cells and the lung cancer cells, not all cancer cells. So,
If you are proceeding with a formulation or if you are planning for a future study, and what would you do? What kind of study will you conduct with using this compound?
mainly, if not yet, I have chosen this project, so I'm not selected.
Okay. Thank you.
There is no question from my side. You can proceed with the next.
Okay, thank you, ma'am. We go next to the next presentation. Tarini Aisahu. Thank you for Sanskrit Saini. And Tarini Aish, can
Yes,
you
ma'am.
share screen your presentation?
Yeah. Yeah. Yes, ma'am.
All
Can you
right.
hear me?
Yes.
Okay.
The time is only 10 minutes.
Okay, sir. Ma'am, is my screen is visible?
Yes, visible.
Okay, okay. So good afternoon to all the and everyone. And myself, Tarini Sao. I'm from student in pharmaceutical chemistry. I'm from JSS College of Pharmacy, UT. So first of all, I want to thank you to the team of APR who given an opportunity to me to represent my work on this. So let's proceed. So my topic is insight into contemporary diabetic retinopathy management. Let's move on to contents. So these are my contents. So the introduction, pathophysiology, then various traditional strategies, then limitation challenges, then the role of AI and the landscape of in non-investigation, non-invasive therapy, then all. Then the introduction. So let's start about the disease. So what is the diabetic retinopathy? So it is the critical retinal condition which affect the person vision power with diabetes. So able to see properly, so you have to do the quick eye check. So here the physician use fluorescein angiography and or OCT to examine the eye, and they can use the dilating agent like cyclopentaloid to dilate the eye. Then move to the pathophysiology. Here you can see there are broadly manifested as through three pathways like phospholipase A2 enhancement pathway, COX2 activity enhancement, then oxidative stress. This all leads to direct and indirectly to the diabetic retinopathy complication, which further categorized into four stages. So let's see videography to understand it easily. So I'm playing this video. So let's see the stages. So it has mostly two stages, that is non-proliferative stage and proliferative stage. So what the actually thing happens in this stage is, in the non-proliferative stage, the swelling occur of the tiny blood vessels, and it is called as microaneurysm. Then it's in this blood vessel, it's and in the interior area, then it bleeds. So while it bleeds, we can see the dots and hard exudates often caudal wool spots also appear. This is broadly manifested as non-proliferative stage. So this let's move to the proliferative stage. You can see the proliferative condition. Here is the various pictorial conditions of the stages of the eye. Then here you can see the how the disease is gradually occurring and damaging the interior part of the eye, and in the proliferative stage, generally the person can see dark floaters, and the blood cell due the so as in the vitreous thing is happen to when the light enters into the eye, and it has to face this neovascularized blood vessel, and after that it reaches to the optic nerve. So for the particular thing that happens, the light can properly reach to the back of the eye. So due to this neovascularized, yes, neovascularized blood vessel, so as a result the person can see properly. Here you can see properly the various conditions is a normal eye. Then here you can see gradual appearance of the NPDR conditions. Then here is the pre-proliferative stage. Here in the proliferative stage, the blood vascular start appearing. Then in the proliferative stage, this vitreous body starts detaching from the eye. Then move to the treatment strategies. You can see the various treatment strategies here. We have laser therapy, anti-VEGF injections, steroid therapy, and vitrectomy. Actually, laser therapy use the laser to burn this actually bleeding things and all. And anti-VEGF injections is intravitreal injection, which is injected inside the eye, which restrict the VEGF, that is vascular endothelial growth factor. Then you can see in vitrectomy, it's involves the removal of the vitreous body from the eye and replacement with a clear liquid or gas bubble. Then you can see the current treatment strategies. These are the current treatment strategies generally. In these conditions, we have to gradually do the eye checkup with in this year. Then frequently we have to do intravitreal injections. So these are the one example, that is the Avastin. So it is a anti-VEGF injections, which is actually it is anti-cancer drug which is approved by FDA in 2004, but used as an intravitreal injection in 2005. So actually it's blocked VEGF factor. So these are the common side effects of this Avastin, that is eye redness, temporary blurry vision. These are the things. So it's the mechanism action of this. Actually, what it does, it actually blocks the VEGF receptors inside the eye, and so as a result, this VEGF receptor can't occupy the receptor, and the neovascularization can be stopped. So again, we have to see what is the limitation and challenges of this anti-VEGF therapy. So there are various clinical manifestations occurs with this use of this Avastin. So we have to see better way to treat this disease. So we can introduce the artificial intelligence. Actually, the artificial intelligence, it involves the detection, the early detection of this disease. So how it detects, it actually it uses various algorithms to and it matches the images of diabetic retinopathy with the patient's eye image, and try to diagnose very quick manner. So it gives an advantage to the physician for early detection. So and you can see it's various algorithms like Messidor, and it uses other database like IPAX, the Optha, and and diagnosis the disease. So actually, this is the data process for this. Actually, it has three sets: training sets, validation sets, and test sets. It utilizes these three sets and process the fundus image. Then it's do deep learning, then extract the feature, and it say whether this condition is referable or non-referable. That thing we have to see. Then here is the example. Here is the device. This picture represents a device that is an IDXDR. It's a fast AI-based software, and which utilizes the algorithm that is cloud-based server to diagnose the diabetic retinopathy conditions and diagnosis it deeper. Then we can see uh, this this is the combination of AI with OCT, that is optical coherence tomography. Actually, this optical coherence tomography is that angiographic test which takes the cross-sectional picture of the retina, and better diagnosis possible by this thing. So how it's do? It's this OCT is combining with AI, so and gives more wider image of the retina,
Sorry,
and
Tarini, your
yes,
time is 1 minute remaining.
okay, okay, thank you. Then we have to see various other things like visual prosthesis. It's a bionic eye actually which is implanted in inside the eye. And here is the landscape of investigational non-invasive therapies. Here you can see various drugs which are represented by various companies, but here Occupy Pharma's PX330 is ongoing clinical trials which gives effects. So we can see the drug. Here is the structure of the drug, which is actually inhibitor of the AP1 and or RE redox factor 1 inhibitor. So how it does is it has a unique mechanism of action. It inhibits the particular factor redox factor 1. So how it's actually what is the advantage of this? It maintains the normal level of the VEGF. So which gives the advantage over the anti-VEGF therapy. So you can see the various time phase thing that it has already completed the phase 2 of the clinical trial, and phase 3 is and 2 is already in the process. Then you can see it's the Zeta 1 phase 2 clinical trial of this thing. Here, these are the participant total 103 number of participants, and these are their characteristics. And here you can see broadly. Here, this is the demographic characteristics of the patients, and here is the optical study of these patients, and you can see there is a three-step change in this DRSS, that is diabetic retinopathy severity, so which indicates is a better option. And you can see the percentage of subjects for developing proliferative diabetic retinopathy is more in case of placebo as compared to the APX. So you can see here the adverse effect, and which is also less in case of APX treated drugs as compared to the placebo treated. So let's go to the
The time
summary.
is over.
Okay, ma'am. That's on. So these are my conclusion, and this is my reference. Thank you, ma'am.
Can you please move back to the conclusion slide?
Yes, ma'am.
Can you please move back to the conclusion slide?
Yes, ma'am. Tell me.
No. Let me see the
Yes, ma'am.
conclusion slide, please. You
Okay,
were
ma'am.
speaking in detail about diabetic retinopathy, and
Yes, ma'am.
suddenly you spoke about a compound. What is it? APX 3330?
Actually, it's a drug which inhibit secondary pathway, that is redox factor 1. So what actually it's do is it inhibits, by inhibiting this factor, we can indirectly inhibit this anti-VEGF. That is, we can indirectly inhibit this VEGF factor. So as an advantage, it can control the normal level of VEGF, so which is required for our body. So it's a better option as compared to the intravitreal.
Is this drug now under study, approved under the US FDA?
Yes, ma'am. Here you can see there is the various results of phase 2 study, and as considering these various studies, and the last time this meeting already happened, and FDA also approved this drug for this phase 3 level, and it's
Are
showing
you
effect.
working in this phase trial?
No, ma'am, I'm just studying the various articles and papers on this treatment and of this drug.
Presenting
And
the review on diabetic
yes, ma'am,
retinopathy.
it's my review paper only. Yes, ma'am.
Which company is this? The product has come to phase 3, APX 3330.
Sir, ma'am, is Ocufer Pharma. Here you can see the company, its logo. It's this company is doing this drugs.
Which company name you have told?
Ocufer Pharma.
Okay, fine. Okay, fine.
That's it.
Yeah, we can move to the next one.
Can I? Okay.
All right.
Okay, thank you, ma'am.
Thank you. And we move to a Pon Malar.
Yes, ma'am.
Yes. You can start your presentation now.
Wait, ma'am. Please wait. Is my slide is showing, ma'am?
Yes, it's visible.
Okay. Myself, Dr. Ponmala, Professor Sri Sairam Siddha Medical College and Research Center, West Tambara, ma'am. My topic is antispasmodic activity of some herbs commonly used in Siddha system. A comprehensive review. It is a review paper, ma'am. The main aim of this review is to focus on the medicinal potential of plants possessing antispasmodic activities with their proposed mechanism of action. Methodology. Siddha is a form of holistic medicine that is focused on promoting balance between your body and mind. The study taken through review paper and articles. Introduction. Many various kinds of illnesses and ailments, including gastrointestinal issues, have been treated with a variety of medicinal plants. Antispasmodic drugs are typically used to alleviate the symptoms of smooth muscle tightness and cramping in gastrointestinal disorders and other serious clinical settings. In the alternative medical system, especially in Siddha system of medicine, antispasmodic herbs were crucial in treating gastrointestinal disorders. The existence of chemical components that are physiologically active may be the cause of many applications. Spasm are very common in human beings. Spasm are continuous smooth muscle contractions may be induced due to endogenous acetylcholine and histamine. They can lead to discomfort, uneasiness, and could result into irritation and inflammation of the GI tract, posing a major health problem to the human being. It could even lead to threatening conditions such as gastritis and inflammatory bowel disorder (IB). Yes. Antispasmodics are used in Siddha system to treat such conditions successfully without side effects. Types of spasm as per pathology, acetylcholine-induced spasm are due to muscarine M3 receptor activation, which is a characteristic of vagal stimulation in the body. So mostly all the endogenous colic pain like biliary, gastrointestinal, ureter arise due to such acetylcholine-induced spasm. Second one is histamine-induced spasm are mediated by H1 receptor activation. This is due to lead poisoning, uremia, excessive gastric acid, or even bile secretion. Third one is barium chloride induced spasm are not mediated by any receptor, but they are mediated by increased calcium channel entry due to spasmogen or increased phosphodiesterase activity leading to calcium channel activity. Some herbs possess anti-spasmodic activity. Gugulu, botanical name, Comifera mucul, is a small tree or shrub with spinous branches. It is a gum resin obtained from incision of a bark. Processed gugulu inhibited the spasm induced by these spasmogens. Gugulu showed maximum inhibition of histamine-induced spasm and barium chloride-induced spasm. It is only a review paper, ma'am. Spasmolytic activity of and polyherbo formulation of three plant species, Apium graveolens, Helicteris isora, and Mentha piperita, was evaluated on guinea pig ileum model. The result indicated that polyherbal formulations displayed significant reduction of spasm induced by acetylcholine nicotine and histamine in guinea pig ileum. Additionally, intestinal motility in mice also inhibited by the polyherbal formulation and proposed molecular mechanism governed by inhibition of muscarine, nicotine, and histamine receptors. This is the pictures of Apium graveolens, means celery, Helicteris isora, in Tamil name of Valamburi, sorry, um, Valamburikai. And the third one is Mentha piperetta. This is Pudina in Tamil. Next, Panicculum vulgare. Panicculum vulgare in Tamil known as Perinjiragam or Sombu. Panicculum vulgare commonly used fennel is a perennial help and flowering plant with feathery leaves and yellow flowers used in traditional medicine for many ailments. It is one of the oldest medicinal plant in the world. It has been established that fennel oil possesses a significant antispasmodic and relaxant effect on contractions induced by methacholine tracheal chains of isolated guinea pig. It is an in vitro study, but my paper is a review paper, ma'am. This might be linked to the opening of the potassium channel. The essential oil has beneficial effect on primary dysmenorrhea. It is commonly used for dysmenorrhea patients and it reduces the pain. Next one is Acorus calamus in Tamil, Vasambu. Acorus calamus is a very important medicinal plant used for various disorders, especially it is commonly used in Siddha system for kids. Major chemical molecules of this plant is alpha azarine, calamine, methyl isoeugenol, and methyl isoeugenol, along with volatile and essential oils. Locally, this plant is used in memory, vascular, and skin problems. This plant is also used for the treatment of digestive and nervous related problems. Pharmacological activities of this plant include antispasmodic, anti-inflammatory, antidepressant, antipyretic, antiemetic, expectorant, carminative, transcoiler, and antibacterial activity. This is a picture of Vasambu, that is Acorus calamus. Spasmolytic potential of Acorus calamus exerted to calcium channel blockages has been reported. Antispasmodic activity of crude extract of Acorus calamus has been conducted using isolated rabbit jejunum preparation. High potassium induced contraction were inhibited by using Acorus calamus and showed antispasmodic activity. Furthermore, tissues were pre-treated with plant extract which caused rightward shift in the calcium to dose response curves. This effect was similar to standard calcium channel blockers such as verapamil in modern medicine. Last one is Anethum graveolens. Anethum graveolens sadakuppai. In Tamil, it's known as sadakuppai. It is a fragrant smell and it a lot of disorders. The common ethnopharmacological uses is antispasmodic and hiccup in infants. It has muscle relaxant, anti-emetic, and anti-convulsant effect. In primary dysmenorrhea, it reduces the pain and has the relaxant effect. Antispasmodic effect is isolator. Ratelium is produced by the quercetin and lutein constituent present in dill extract. Conclusion: it might be concluded on the basis of obtained information that in the spasmodic pain can be treated by traditional natural medicines which are effective, safe, and economical. Under this review, many plant species have been identified and reduces the spasmodic pain with low toxicity and side effects. The recent research data supports to use the herbal medicines for treatment of variety of ailments. Hence, more work is being carried out to identify the potential antispasmodic effect of different plant species, which deserves more awareness in terms of in vivo and clinical trial studies. The detailed literature analysis of medicinal plants could be the rich source of antispasmodic compounds and open up new areas for researchers and scholars in this field. Therefore, further pharmacokinetics and pharmacodynamics along with clinical trials should be carried out to validate efficacy and reveal the safety profile of traditional medicine to manage spasmodic disorders. This is my reference, ma'am. Thank you, ma'am. Excuse me, ma'am.
Yes, ma'am.
Any questions, ma'am?
Yeah.
Tell me, ma'am.
In your practice, what is the antispasmodic you try to give for your patients, ma'am? Which plant would you prefer among the plants you listed?
Sadakuppai, Anethum graveolens is commonly used for dysmenorrhea, ma'am.
Okay.
Another plant, Acorus calamus vasambu, is a very good medicine for infants colic, ma'am.
Okay. Is there any
Funicular.
adverse effects you have observed for these plants when given them?
Excuse me, ma'am.
Did you observe any adverse effect of these?
Not yet, ma'am.
Okay.
Till now, no, ma'am. In future, if any present, we will document it, ma'am.
Okay.
Thank you.
When you like, I would like to know about your conclusion. Like, would you recommend combining the extracts of few plants you have listed and go for an antispasmodic activity, or how would you go for further research from this topic? What is your idea?
In future, I will definitely carry out this type of plans for in vivo studies, ma'am.
In vivo studies.
Yeah, ma'am.
But it is being commonly used already, right?
Yes, ma'am. Yes, ma'am. Yes, ma'am. But anyway, a type of standardization is needed for in this in current era, ma'am.
Okay. Okay. Okay.
Thank you. Thank you so much. Can I leave, ma'am?
1 second. I
Stop.
wanted to know whether any of this drug you have patient or only just a review you have collected.
Yeah, ma'am. All from my experience, ma'am. This type of plants are well work in our patients, ma'am. That way I select this plants, ma'am.
In Acorus, which part of the plant you are using? Whether leaves, rhizomes, or
No,
which
ma'am.
ones?
Rhizomes, ma'am. Rhizomes, ma'am.
Rhizomes
Rhizomes are
only you're using.
rhizomes, ma'am.
Okay, okay, fine. Okay,
Thank you.
we can go to the next.
Okay. Thank you, ma'am. Thank you so much.
Thank you, Ponmolar, and we are going to next presenter.
Thank you, ma'am.
Yes, thank you so much.
Sagar
Sagar
Tamahan.
Tamahan. All right.
Yes, ma'am. Yes, ma'am.
Okay, ma'am. Myself, Sarkant Mohanty, first time farm student, Department of Pharmaceutical Chemistry, JSS College of
Sorry,
Pharmacy.
your your slide is not visible yet.
Yes, ma'am. My screen is visible, ma'am.
No.
It's my screen. Now it's visible.
No, it's stuck on your folder.
Okay. Okay, ma'am.
Can you stop share and
Okay,
reconnect again with
now visible.
Sir, actually, your screen is visible, but your slide is not visible. Please click on the slide.
Okay. So now visible?
No.
Please double click on the
Yes,
slide.
ma'am. Yes, ma'am. Actually, ma'am, it is stop. I will. Yes, ma'am. Now it is visible.
No, it isn't. Or you can share your PPT to
Okay, ma'am.
committee.
Sir, could you please share the slide with me so that I can share the slide?
Okay, sir.
Yeah.
Asla.
Hello.
Now it is visible now.
Yes, it's visible now.
Thank you. Actually, it is a net problem. That's why, ma'am. Okay. Okay, ma'am. Can I start, ma'am?
Yes, you
Hello.
can start now.
Good afternoon, everyone. Myself Sarkant Mohanty, first time student, Department of Pharmaceutical Chemistry, JSS College of Pharmacy OT. Today I'm going to give presentation on the title that in silico studies of some actinic derivatives used as breast cancer agents in human epidermal growth factor HC2 Breast cancer is one of the malignant tumors which mainly affect the female population. About 20% of the breast cancer cases are due to the overexpression of human epidermal growth factor receptor HER2. In general, actinic derivatives play an important role in anti-tumor activity due to their interlocking properties, and mainly that HER2 positive breast cancer is morely dangerous for the female. And here we have designed some novel actinic derivatives to check their binding affinity against the our targeted receptor HER2 receptor by using the scavengers Maestro 12.6 version. Most of the compounds are significantly bound to the active side of that receptor HER2 target with significant Gleim score minus 8.28 to minus 9.31. When compared to standard drugs like tamoxifen, lidocaine, to check the stability of the docking complex, prime MMGBSA was performed, which show significant digibind values. 100 numbers of MT stimulation for the compound AB27 was performed using Desmond model. In the in silico ADME study is also performed by using quick proof model and produce satisfactory results. As for our in silico results, the compounds AB27, AB20, and AB1, AB11 are significantly active against HER2 target and employed in the future development. Here is my introduction part. Many types of cancer can be treated by chemotherapy, and search for new chemotherapeutic agent still plays a major role in the fight against cancer, because we know that cancer nowadays cancer has more mortality rate according to WHO data. For about 1 in 5 women affected with breast cancer agent, and according to WHO data, breast cancer caused 6 lakh 70,000 deaths in globally in 2022. And human epidermal growth factor receptor 2 inhibitors always useful target for the breast cancer. The cancer cells have too much for a growth promoting protein known as HER2 on their surface. These cancers, known as HER2 positive breast cancers, tend to grow and spread more aggressively other than other cancers. The benefit of anti-HER2 therapy is demonstrated to one of the most promising molecules for targeted therapy. And breast cancers can have up to 25 to 50 copies of HER2 gene and up to 14 to 100 folds increase in HER2 protein, resulting in 2 million receptors expressed at the human cell surface. In general, we here we taken that 9 substituted actinic derivatives, because that 9 amino actinic have that various biological activities, and it is specially as anti-tumor due to their DNA interacting properties. In continuous of our previous research work, Kaliranjan R. et al. On searching new potent psychotoxic agent, we have designed 9 substituted actinic analogs by molecular docking studies by using scavengers. The results reveal that the newly designed 9 substituted actinic derivatives exhibited good inhibition with HER to exhibit anti-breast cancer activity. And here is the materials and methods we took for the our research work. The 3D crystal structure of HER2 receptor. Here is the PDBI we retrieved and prepared by protein preparation result of EPIC model. The above protein was prepared by protein preparation model of Schrödinger suite. Missing chain atoms are adding by using prime model. Finally, a restrained minimization of protein structure was carried out using our OPSL3 force field, and the ligand AB1 as actinic substitution are generated and subjected to leak probe model. The docking studies were performed by a glide model. In silico ADME scanning was performed by quick probe model. And here we use that the glide docking program using SP mode. The MD stimulation studies for the significant active compound also performed by using Desmond model. And we got that from our research, we got that result and discussions here. The molecular docking studies for designed ligands to protein active sites were performed by Scoringer for determining binding affinities for the ligands. The designed analogs were docked in HR receptor in order to their potential activity against breast cancer. From acridine derivatives 1 to 41. So the good affinity to receptor when compared with the standard acridine derivatives with anti-cancer activity like lidocaine and anti-breast cancer agent tamoxifen. The glide scores against HER2 are shown in the table. Here is the table 1. Here is the table 1. In that table 1, we took that parameter, our glide score, hydrogen bonding, glide energy, then our binding property, then covalent bond, hydrogen bonding, like that. In that we can see that AB 20, that acridine substituent 20 and acridine substituent 27 and 11 has lowest glide score, that is minus 9.312 and minus 1.78 and minus 0.50, minus 9.10. And here the 9 substitute acridines are located in the hydrophobic pocket with the amino group is located in the hydrophobic pocket, and the interaction between the drug and that our target ligand is BL 734 to 862, which is the active site. And here is the figure 1 is the structure of compounds AB acridine substituent 1 to 41. Here is R is AR can be 41 structures, and that ligand interaction of compound AB acridine substituent 20. We saw that the active sites of these compounds. And figure 1 is the docking studies for 9 substituent acridines. Top 10 out of 41, we selected only top 10 who is having that binding and free energy using our prime and prime model. And table 1 is the in silico ADM screening for 9 substituted acridines. And here is the parameter is the molecular weight and no, donor hydrogen bonding, then acceptor, then that our partition coefficient log p, and that the role of height is
Sorry, sir. Your time is 1 minute left.
okay, ma'am. And here is the MD stimulation studies for significant active compounds. We taken the acridine substitute 27 as our for the MD stimulation studies, and here is the conclusion is that generally 9 amino acridine derivatives exhibited various biological activities, and mainly it is anti-cancer agents, and that the present studies clearly demonstrate that some derivatives out of 41, that the acridine substitute 27, 20 and 11 have significantly HER2 inhibitors to be used for after further studies for and refinement for breast cancer activity. And that here I am. Thank you, everyone. Okay.
Sagarkant, right?
Yes, ma'am.
Okay. What is new in this study, Sagarkant? HER2 receptor studies. Many studies have already explored about HER2 receptor activity, and many drugs
Yes,
have
ma'am.
been studied, right? What is new in your study? What is the novel?
And that acridine, before acridine has many biology activity, but we studied that if we substitute that acridine at 9 position, then we can see that 9 amino acridine substitute has breast cancer activity, anti-breast cancer activity. Ma'am.
Okay. Are there any further literatures reported for the same derivative?
From our previous researcher, we studied that some potent psychotropic cytotoxic agents can be formulated from 9 separate activities, ma'am, analogs.
Okay. What do you think about
Based,
the activity, anti-breast cancer activity? How good it would be when compared to the conventional treatments available?
ma'am, nowadays that breast cancer is more prominent in every time. That's why we have to synthesize a Nobel that is less side effect and more potent towards that HER2 receptor, because HER2 receptor plays a vital role in that breast cancer, ma'am.
Okay, Sagarkan, thank you.
Okay, ma'am, thank you.
See, you're already synthesized or you're just done the docking studies, correct? So any plan of synthesizing and checking
Yes,
the
ma'am.
activity?
Our further plan is to synthesize that drug, and now only, ma'am, we started the docking scores and that glide score, binding energy.
Okay, what is the cost of preparing one product, one molecule with the activity which you have found? How many steps are involved in the synthesis of this particular molecule?
Can you repeat me?
How many steps are there for synthesizing this molecule?
Ma'am, we can synthesize, ma'am, that active side, ma'am, after the studying that, ma'am, out of 41, these threes are, ma'am, more potent drug than. Then
Correct.
yes, ma'am. And then here is 27. We can calculate. We can that aromatic substitution. We can conclude that the 27 substituent number 27 we can synthesize, ma'am.
Okay, fine. We can proceed with the next one.
All right, that is Isma. Can we move to next center?
Yeah.
Okay, thank you so much. And we move to S. Chitra.
Yes,
Can
ma'am.
you share your presentation, please?
Yes, ma'am.
Thank you. I will inform you. It's only 10 minutes to presentation.
Yes, ma'am. Good afternoon to all. Ma'am, screen is shown, ma'am. Screen is visible. Can I proceed, ma'am?
No, ma'am, your screen is not visible. Your screen is visible, but your PPT is not visible. So please click on that.
Okay.
All right, it's visible now.
Okay, thank you. Myself, Dr. Chitra, Professor, Sri Sairam Siddha Medical College and Research Center, Chennai. My topic is a holistic scientific validation of standardization, Vediyanabedi Chenduram, a herbo-mineral formulation. Introduction. Our Siddha system of medicine encompasses various formulations. They are derived from plant, metal, mineral, and animal sources. There are 32 kind of internal medicine and 32 kind of external medicines are there in our system. Chenduram is categorized as one of the 32 internal medicines, boosting an imperative self-life is 75 years. Vediyanabedi Chenduram is a traditional herbal formulation utilized for addressing diverse health conditions including but not limited to anemia, fever, jaundice, dysentery, dropsy or generalized anasarca and ascites. My objective is to explore the analytical properties of Vediyanabedi Chenduram. The method is to find out the shape, size, and elemental composition of Vediyanavedi Chenduram. Rationale of the study. There is a prevailing public concern regarding the perceived toxicity of these medicines stemming from a Western disregard for traditional medicine systems, as evidenced by various studies. Some studies conducted in the traditional system of medicine supporting the safety of the human health. While there has been some research studies conducted on Vediyanavedi Chenduram, but limited instrumental analysis work has been undertaken thus far. Therefore, the present study is concentrated on the in-house preparation and comprehensive characterization of Vediyanavedi Chenduram, such as scanning electron microscopy, high-resolution scanning electron microscopy, transmission electron microscopy, X-ray diffraction, and X-ray fluorescence, and energy dispersive X-ray analysis. The ingredients of the Vediyanavedi Chenduram shows on the table. Annavedi, it's a Tamil name. The nature of the drug is inorganic salt. Ferrous sulfate is the chemical name. The quantity was taken for the preparation is 35 gram. Vediyuppu, the next drug, it's a naturally occurring mineral source of nitrogen. The chemical name is potassium nitrate. In English, it is saltpeter. It was taken in 7 grams. Then the only one plant, lemon, that is citrus lemon is the botanical name. Parts used is plant. Fruit juice added to this for the preparation of this formulation. Next method of preparation, Annavedi, that is ferrous sulfate, and Vediyuppu, potassium nitrate, were purified separately. Then mixed with lemon juice and ground well, it was taken in a waxy stage. Mixer was placed into a mud vessel and covered with another mud vessel. It was then wrapped in 7 layers of cloth cover, that is called sea lemon in Tamil, and subjected to calcination using 70 cow dung cakes. 3 pudams, that is furnace techniques, were subjected and finally powdered. Vehicle is honey. Dosage is 130 milligram twice a day. Duration is 45 days. Indication: anemia in Tamil Pandu, jaundice Tamil in Kamale, dropsy or generalized anasarga in Tamil Sobai, fever Suram, dysentery called in Tamil Seedavedi, and ascites is Peruvedi. Apart from the preparation indication, Annavedi is used for ulcers, abscess, or cancer in the womb, Karpasya Vrenam called in Tamil. And Vediyuppu is used for uterine abscess, that is Karpasya Katti in Tamil, and infertility. The scanning electron microscopy, SEM of EABC. The SEM images were taken at magnification of 8000x, 50,000 and 50,000x, which gives respectively 10 microgram, 5 microgram, and 1 microgram represent the micrograms. The following SEM images of EABC exhibits variation in both particle size and agglomeration. The distinct differences in morphology as depicted in the micrograph indicate the presence of more than one crystalline phase in the sample, attribute to the diverse chemical material present in the sample. These are the pictures, the SEM images of EABC. Then high resolution scanning electron microscopy shows the table, the slide shows the pictures at the magnification of the 35,000 and 50,000 and 10,000. This is the nanoparticle picture shows in these slides. These are all the structure reveals the pronounced agglomeration of the nanoparticles. Then transmission electron microscope, TEM of Vediyanavedi Chenduram. The presence of nanoparticles was confirmed, which already seen through the SEM. The particle size is 2 nanogram, 2 nanometer and 5 nanometer and 20 nanometers. The pictures are shown in the slide. Then EDAX of Vediyanavedi Chenduram. The table shows the element composition, the atomic percentage and atomic error, weight percentage. The element composition of the drug sample was assessing using EDAX. The EDAX analysis reveals that Vediyanavedi Chenduram has mass percentage of iron, oxygen, indium, and sulfur, which are the 38.7%, 70%, 35.41%, 16.03%, and 5.05% respectively. Then XRD characterization of Vediyanavedi Chenduram. The X-ray diffraction spectra illustrate the structural composition presence in the prepared Vediyanavedi Chenduram. This is shown in the graph. The phase purity and the crystallinity of the Vidyanabhidi Chenduram treated electrodes were examined by the X-ray diffraction. It is a versatile method that helps to study the properties of the materials. It reveals information about the composition, structure, and orientation of the crystals in solids, powders, and liquids. The direction of diffractions depend on the size and shape of the material building black, while the intensity of the wave depend on the type and arrangement of atoms in the crystal structures. In this preparation, many crystals are composed of tiny crystal materials are present in the VABC. These graph shows ensure that the property. Then XRF of VABC, the table shows the compound of the metal oxides present in the Vidyanabhidi Chenduram. The XRF analysis showed the absence of heavy metal compound and the presence of oxide forms of Fe, sulfur, and silica. Aluminium, magnesium, and sodium in elemental form, which is the minimal amount. Then XRF study has shown the presence of Fe2O3. All the
Sorry,
metallic.
you only have 1 minute remaining.
Tell me, ma'am.
1 minute remaining, ma'am.
1 minute. Okay. All the metallic forms or element converted into its oxide form heating process. This shows there was no existence of natural raw material. The presence of oxide forms of iron and sulfur elevates its therapeutic value. Conclusion. The current study assessed the physicochemical properties of traditional Indian medicinal Vidyanabhedi Chenduram. The results from XRD SEM studies serve as excellent physiochemical fingerprints, offering a robust validation of this medicine. The near nano size of Vidyanabhedi Chenduram enhance absorption, potentially leading to improved bioavailability. The study represents a sincere effort to scientifically validate herbomineral traditional Indian medicines, addressing the need for proper standardization and quality control measures in a competitive international landscape. In conclusion, SEM and TEM analysis of Siddha formulation Vidyanabhedi Chenduram provides insights into nature and size of the particle within the drug. The structural modification of ingredients occur during the preparatory phases, eventually transferring into a final product. These particles found the finalist Vidyanabhedi Chenduram as predominantly within the nano range. Consequently, I can deduce that this formulation can be classified as a nanomedicine. These characteristics suggest potential advantages such as enhanced bioavailability and an extended shelf life. These are all my reference. Thank you. Any questions? Is there, ma'am?
In this showroom, you have added
Yes, ma'am.
ferrous
It was a good presentation. This Annabedi Churnam has been used. Vedic Annabedi Chenduram is used since long time, right, ma'am? It is a well-established medicine.
Yes, ma'am. Yes, ma'am.
Yes. Then, why did you go for this standardization? Or what was your objective, ma'am?
The limited studies are heavy metal analysis. There is no toxic element present in the Vidyanabhedi Chenduram. That's why I take out for the standardization method, ma'am. Then the medicine is then a particle conversion process. So I want to prove that. So I take out this study, ma'am.
Okay. Okay, ma'am. How are you going to proceed with your study, ma'am? What is your future?
My future study is to in fibroid uterus how this drug is work on it, ma'am.
Okay. Okay, ma'am. Thank you.
Thank you, ma'am.
No, I wanted to know ki this ferrous and potassium you have added, they are strong alkalis, no? So whether that has not impact on your study? Hello?
Hello, ma'am.
You have added ferrous salt and potassium salt, correct?
Yes, ma'am. Ferrous sulfate and potassium nitrate.
Nitrate. So that doesn't have impact in your because they are not safe to use to that concentration, because concentration you have used is too much.
Ma'am, it is a safe one, ma'am. It is a hepatoprotective drug, ma'am. Already proved that. The toxicology studies were done, ma'am.
Okay, fine. Yeah.
In animal model, ma'am.
Okay, fine.
Okay.
Yeah, thank you. We can go for the next one.
Okay, ma'am. Thank you so much.
Thank you so much. And we move on to next presenter. Please join Gaurav Chandra. Are you in this room?
Yes, ma'am.
All right. Let's start the presentation.
My screen is visible, ma'am?
No, it's not visible.
Yes, ma'am.
All right. It's visible now.
Good afternoon, everyone. My topic for presentation is advances in microsphere hydrogel system for enhanced ocular disease. Firstly, we will start with introduction in microsphere and hydrogel system, advantage, application, mechanism of action, future direction and challenge and conclusion. Introduction. The eye with its complex structure and unique present a challenge for effective drug delivery. Ocular drug delivery is a specialized field dedicated for overcoming this challenge to administer medication for treatment of various ocular disease. For common elements like dry eye syndrome and more severe disorders such as glaucoma and macular degeneration. Traditional methods for ocular drug delivery, such as eye drop, ointment, often suffer from limitations such as poor viability, rapid clearance, and need for frequent administration, leading to the issue with patient compliance and treatment outcomes. Emerging technologies such as nanoparticle liposome hydrogel and microsphere-based system have shown promise and improving drug retention. This and bioavailability. This enhancement not only offer hope for the other effective treatment, but also hold the potential to reduce the dosing frequency and minimize the side effect and enhance patient comfort and compliance. There are various routes for ocular drug delivery system. Microsphere as hydrogel system. Microsphere hydrogel system combined with the properties of microsphere application including drug delivery. In this system, microsphere, which are tiny particles, are embedded within a hydrogel matrix, while hydrogel provide a three-dimensional network of hydrophilic polymer that can retain large amount of water. Key component. Microsphere are solid spherical particles with diameter typically ranging from few micrometer to hundred of micrometers. They are often made of biocompatible polymer or other material that can encapsulate drug. Microsphere can release drug through various mechanisms, including diffusion and degradation of polymer matrix. This particle provide a means of controlling drug release kinetic and protecting drug from degradation. Hydrogels are the three-dimensional network of hydrophilic polymer that can absorb and retain large amount of water. They are highly biocompatible and mimic the natural environment of tissues. Hydrogel can be designed to adhere well to ocular surface, providing prolonged contact time and enhancing drug absorption. They offer a comfortable and biocompatible platform for drug delivery application. When the microsphere and hydrogel are combined, then a combined system is formed. When microsphere are incorporated in hydrogel matrix, they form a composite system that leverages the benefit of both components. The microsphere provides a controlled drug release, while the hydrogen matrix enhances drug retention and tissue adhesion. The combined system allow for sustained and localized drug delivery to the target site. The property of microsphere, such as drug release, kinetic, and mechanical strength, can be finely tuned through formulation parameter to meet a specific therapeutic goal. Advantages of microsphere hydrogel system. Firstly, that is controlled release. Second, enhanced retention and bioavailability. And application of microsphere hydrogel system in ocular drug delivery. Firstly, treatment of glaucoma by delivering intraocular pressure lowering agent. Therapy for diabetic retinopathy by controlling the release of drug to mitigate inflammation and vascular abnormalities. Treatment of ocular infection by localized delivery of antimicrobial agent. Mechanism of action. The mechanism of action of microsphere based on the hydrogel are drug encapsulation within, firstly, drug encapsulation within the microscope. The process begins with the encapsulation of drug within the microsphere. This can be either hydrophilic or hydrophobic compound depending upon the composition of microsphere. Microsphere act as a reservoir for the drug, protecting them from degradation and providing sustained release kinetic. So dispersion of microsphere within the hydrogel matrix. The microsphere containing the encapsulated drug are then dispersed within the hydrogel matrix. The hydrogel matrix provide a scaffold for the microsphere, ensuring their uniform distribution and preventing aggregation, adhesion to the ocular surface. Upon administration, the hydrogel component of the system adhere well to the ocular surface due to its biocompatible and adhesive property. This adhesion ensures prolonged contact time between the drug-loaded microsphere and to the ocular tissue. Controlled drug release. Within the hydrogel matrix, the microsphere releases the encapsulated drug in a controlled manner. Factors such as composition of microsphere, the porosity of the hydrogel matrix, and the environmental condition influence the release kinetic. The sustained release of the drug from the microsphere hydrogel system prolong the therapeutic effect and reduce the frequency of dosing. Tissue penetration and targeting. The hydrogel based on the microsphere in permeate the tissue. Additionally, properties of the microsphere and the hydrogel matrix can be engineered to target a specific tissue or cell within the eye, maximizing the therapeutic efficacy while minimizing the systematic effect. Biodegradation and clearance. Biodegradation and clearance. Over the time, both the microsphere and hydrogel matrix undergo biodegradation and clearance for the ocular environment. Future direction and the challenges for microsphere hydrogel system in ocular drug. First one is optimization of formulation parameter, enhance stability and cell life, personalized medicine approaches. What are the challenges that we face? Biocompatibility issues, controlled drug release, and scale-up manufacturing challenges. Conclusion: microsphere hydrogel system represent a promising approach for enhancing ocular drug delivery. By combining the advantages of microsphere and hydrogel, this system offer controlled release and increased residence time. With further research and development, microsphere hydrogel system hold a considerable promise. These are the references, and thank you.
No questions from my side. We can proceed further.
Yeah, there's no question from my side. Okay.
Before we continue, can we welcome our 8 keynote speaker? There is Dr. M. Sunita Reddy. Good evening, ma'am. Can you hear us, ma'am?
All right. For Q&A, the last author, we have to wait and we continue the a keynote speaker, Dr. M. Sunita Reddy, Principal, Center of Pharmaceutical Science, GNTUH University College of Pharmaceutical Science, India. Hello, Mom.
స్క్రీన్ చేస్తున్నారు మాట్లాడాలి వినపడట్లేదు కదా సెట్టింగ్స్ పెరుగు
Sorry, Mom, the screen is not visible. Do you have any trouble?
Hello, can you hear me?
Yes, Mom.
Yeah, shall I share my PPT?
Yes, of course, Mom.
Yeah. Can you see my PPT?
No, it's not visible, Mom.
Yeah?
It's not visible, Mom.
Not visible. 1 second, okay? 1 second. One second.
Sorry, Mom, if you have any trouble, you can share your PPT to our committee. All right, it's visible now.
Sorry, mom. Your sound is. We cannot hear your sound.
Hello, can you hear my voice?
Yes, I can hear your voice.
Okay. You can see my screen, isn't it?
Yes.
Yeah.
Yes, we can see
And you
your
can hear
screen.
my, yeah, okay. Sorry about that, some trouble. Okay, I'm Dr. Sunita, from Professor and Principal from the JNUH University College of Pharmaceutical Sciences. I'm going to talk about innovations, research challenges and opportunities. Okay, and what is actually, this is a scientific research. Scientific research is required for all pharmacy students and to excel in their pharmacy profession. And we can start our research program as scientific research from undergraduate to the even for PhD, up to PhD also. Like undergraduate, we need to start from like a institutional, and then again graduate like a pre-doctoral. Then again we can go for the postdoctoral. Postdoctoral will be like become career development awards, and also we can work as a faculty, and we can do the our scientific research also. This is we can successful, we are in end of the day we will be in a successful independent research investigator. And during this period we should be expecting like best education, scientific knowledge, and creativity, basic research skills, and also we need to have a like technical writing, and how we can have a grants also we can, I will be talking about, and how what we need to do in the publications and how the collaborations. This is all we are going to talk in the scientific research as a career option. So our scientific research will start with like a choose your what is your research topic first. Okay, after that you will be might be finding like few topics. Out of that you need to narrow down and find your actual area where you wanted to work, and after that identify the research problem and develop a clear research questions. Then you need to create a design how to excel in that kind of research. So what is actually research means? Research is like a creative systematic work undertaken to increase the stock of knowledge, including knowledge of human culture, society, and use of this stock of knowledge to devise a new application. Okay, and it is a careful consideration of study regarding a particular concern or particular problem. So how choosing the research topic? We need to start with the academia from like universities like us, and you will be able to make your research topic even at 12th grade, and undergraduate or graduate postdoc, or as even the faculty also. When you are working in industry, you can be having the research topic related to analysis, product, project, or anything. And if you are working in a government or intergovernment, you can be having choose a research topic useful for the director, coordinator, officer, or anyone. And even you can excel in a also as an NGO also in a scientific research career you can get it. Okay, research topic versus research paper. It is a one of the complex area, and to have a research topic, you should have an idea. Then you should have a plan. You need to collaborate. Then you need to take care of the action. Okay. So, but the most important in research is ethics. Okay, ethics should be like you should whatever the research topic you select, it should be you should have integrity, you should have a competence working towards that, and also you should have a non-maleficence, and also you need to justify your work, and you need to do all this research with a dignity and benefit. This whatever the research you do should be beneficial to the society, and whatever the findings you should be able to tell that you are responsible for those findings, and you should be honest enough to produce them and reproduce them. and you should maintain the confidentiality, privacy until you publish and all you patentable. Okay, and the research process will start with a research idea. Then you need to do the literature reviews. Then you need a theoretical formulation of the research problem, and empirical research questions, that what questions, what you want to do, how you are going to do it, all these need to be done. Then research design planning, how which plan or which design work for suit for your research should be planned enough. And then you start the data collection, then you do the work, then you do the data analysis, then you need to do the all research questions asked by the professors and mentors and peer review groups. Then you need to do the theoretical interpretation of your results, and these results should be societal useful, and you need to compare with the research what is already existing and how your research has a novelty that you need to do. Then you do for the conclusion. Okay, and in a research publication, a paper publication, one is a very complex thing now. And if you want to publish a peer journal with impact factor, you need to have a lot of things need to be taken care. One is, first thing is you need to finish the research, actual research, and then you write a manuscript. The manuscript you can submit. The manuscripts are reviewed by several people, and these several people will be asking you several questions and feedback you need to do, and you need to clarify their doubts. And one decision is done and they are satisfied, that will be published. Otherwise they ask you to rewrite your manuscript. When they ask you to rewrite your manuscript, you have to be concentrate on what you need to do or how you need to do, that is more important. And you, after again, if it is rewrite, then again you have to take care of the problems, what is happened, or what are the questions asked by the reviewers, has to be put it in your mind and do the correction of manuscript. Then you need to make a decision, and then once the paper is submitted to the publisher, then they will make you a decision to accept or reject. If it is reject, you need to submit it to the later by modifying or doing some other research and suitable for that manuscript. Once it is accepted, the publication will come into the journal or review, a peer journal, and you need to choose the journal which has an impact factor and which is a UGC journals, or if it is in India, or a Google journals, or a Web of Science. Those journals are more popular and more acceptable worldwide. And when you are writing a successful research paper, you need to have certain guidelines. First one is you need to have an innovative ideas, and you need to justify your research work. Then explain the different scientific terminologies and make a bibliography, very important bibliography, which is giving the reference. And you strictly follow the all specifications of approving a publication authority of a journal, and thoroughly you have to be proofreading from the top to bottom of your journal, and correction, even spelling mistakes and also grammatical mistakes, and a scientific area where you are projecting, that also you need to be corrected. Then develop a depth of knowledge of the subject. So to do this one, you need to develop a depth of knowledge of the subject. Then the writing should be simple and avoid unnecessary things, and have a clear understanding of the content, and using a unique style in writing. So these are all will make you to publish a very good journal. And during the publication or during submission of PhD, we will have a plagiarism. This plagiarism is an appropriation of another person's ideas, processes, results, words without giving appropriate credit, including those obtained through the confidential review of other research proposals and manuscripts. That is plagiarism. Plagiarism is done by different softwares, and they recognize the whatever the publication or whatever the thesis or whatever the dissertation done by a student or a scientist, where they got it and what are the things they have copied or what are the regenerated, that can be recognized. What may be plagiarized? The plagiarization can be words can be plagiarized, ideas can be, findings, writing, graphical representation, any diagrams, graphs, printed, all these you have to be unique in your publication or unique in your PhD. There is in India, we always care about UGC care journals, and in all over the world, you will be Web of Science or Google Scholar or some peer journals, and those you can take care of them. So there is a for every either PhD or every master's program, we need to provide a synopsis. A synopsis is simple defined as a brief discussion and summary of general survey carried out about a particular project or a topic. And what is a synopsis is like example of short synopsis of a story, like in Jack and Jill, and or anything like a formulation and evaluation of certain drug. A small, precise, brief discussion and summary with a conclusion, that is called a synopsis. And guidelines for the synopsis is more important because this synopsis should able to give the what work you have done, what work you are presenting. That is should be contain title of your dissertation and topic, and you should have some kind of introduction and where you reviewed, what references you have taken, and what is the problem you are selected for your thesis or you are selected for your project, and what is the need, why the research is required in that area, and what is the objectives of that area you have to have, and you need to have a objectives and draw a line or a boundary for the research work, and what methodology you are going to apply for your objectives, and how you are going to analyze, what are the tools you are going to do, what are the where the sources you are going to do, and what are the sampling, and what is the bibliography, what is expected outcomes, and what you got it, what conclusion you have it overall, that all need to be contained in the synopsis. Okay. And dissertation and thesis writing, it should be very important, because without that the actual acceptance of PhD or acceptance of paper or journal is very difficult. And what are the initial pages should be like a title page, what is approval sheet, what is abstract should contain everything about your work start and objectives and what is outcome, and acknowledgement, who helped you, who is the guide, who are your peer group, and who are your lab mates, and who helped you, who are the students or who are the other professors who is helping, and whom you are dedicating this thesis or dissertation. And you should have a table of contents, tables of figures, tables of list of tables, and it should everybody should able to find after seeing these contents. Okay. And what dissertation and thesis is like a purpose. What is the purpose? And perhaps the biggest difference between a thesis and dissertation is the intended purpose. A dissertation is a commonly required to obtain a master's degree, and thesis is usually done by the doctorate students or a PhD students, focuses on original research. And there is a difference between thesis and dissertation. The thesis is shorter than the dissertation, and a original research work that helps to obtain the PhD degree, a simple a one part of a research, and dissertation is a constitute an original research project, helps to obtain the doctorate degree, and it can be a part of a master degree which you have extended in a dissertation also. Steps in writing thesis, we have to remember a lot of things to know is understand the goal of the thesis, and also know the qualities of good thesis, and understand the structure of a good thesis, and understand the language of a good thesis, and you need to seek help for your thesis also. And the don'ts, what are the things which you are not supposed to be in the thesis are never plagiarized, and never produce a falsify or cook up a record or data, and do not use passive voice, use always active voice. Do not use words like a quite, some, considerable, great deal. All these are not acceptable in thesis. And do not use abbreviations which are not defined anywhere. And don't present a conjectures as a fact. That is a more important in a thesis writing. And what are the components of technical report that you need to have for a technical writing? For example, you should have an executive summary. What is the summary? And methodology, introduction, your results, conclusion, reference, and abstract, background, appendix, figures, and tables. That is a more important. And you need not to put each and everything, but you need to put only optimized things which are there and which you need to present. And writing a technical report is used to convey the important findings and test results to be controlled audience. Okay, and you should be able to understand by the other author and employee also. And not only writing, presenting for thesis and also for a publication of journal, you can have a projects, you can do a grant writing. Grant writing is more important because if you get a grant, you are able to do the research without any boundaries. If it is for a grant proposal, you have to be very careful in your objectives and outcomes, and also your novelty. And whatever the novelty, you should be useful for the society also. That is more important. And you need to remember many C's for a grant writing success. For example, you should be conscious, be constant terminology correctly, and you have to be cohesive application to get funded. Be your own consultant and complete their sentences. Be correct and be comfortable. Be current and contemporary. Be calendar sensitive. Don't be funding contingent. Be creative and clustered, but not competitive. Collaborative with the partners. Represent a community of practice. Become a known commodity. Explain the valuable credentials. Don't just copy and paste. Connect with their past recipients. Offer a solid corporate foundation. Contribute back and be courteous. Use careful word choices. Circulate your application. Understand the chain of command. Leave some cash on the table for your work. Commercialization is more important. Consider the funders' perspective. Have a just cause. Display cross-country value. What is the use? And be confident, not too cocky. And understand the chain of grants. Proof of consent first, then commercialization. Calculate your value before and after. So these are the more important C's that you need to remember to get any grant from anywhere or any place. And winning characteristics will make you to expel or excel in your profession. For example, you should have a strong plan and address the needs, evidence of organization capacity, and you should have a justifiable evaluation plan, and you should have a sufficient and necessary budget required for your proposals or your work. Reasonable sustainability plan should be there, and it should be reproducible, and you
Sorry,
should
ma'am.
able to yeah, go ahead.
The presenter are waiting for their presentation, so you can first
Yeah, I'm
your
going to
presentation.
conclude. Yeah, I'm going to
Thank you so
conclude in a
much.
minute. Okay. And these are the silent points to build a better scientific research career. And time and data management and networking skills, all these are required. And don't struggle for survival or existence, but struggle for excellence. That is more important. And that's all my presentation. Thank you. Thank you for the opportunity. Hello.
All right. Thank you so much, ma'am, for your
Yeah.
time.
Okay. Okay, ma'am.
And then after this, we have to continue our oral presentation.
Okay, okay, ma'am. Go
With
ahead.
presenter. The
Okay.
next presenter is Gaurav Chandra with the title of Advances in Microphase Hydrogel System for NSAIDs Ocular Drug Delivery. Gaurav,
Okay. Go ahead.
yeah. Thank you so much, ma'am.
Okay. Okay, ma'am. I'm leaving.
Yes, thank you so much, ma'am.
Gaurav just completed before ma'am's presentation. So, ma'am, as per the list given, the next presenter should be
Vishal
Vishal
Kumar.
Kumar.
Yeah.
Yes,
So
ma'am.
sorry.
May I
Gaurav
audible,
has
ma'am?
already, yes. Gaurav has already completed.
Oh yeah. So sorry. We can go with Vishal Kumar right now. Are you ready?
Yes, ma'am. Hello, ma'am.
You can start your presentation right now.
Okay.
Go.
And is this visible?
Yes, it's visible.
Can we start?
Yes, you can start now. It's only 10 minutes.
Okay. Good afternoon, everyone. My presentation topic is the role of pharmacists in detecting and resolving drug-drug interactions. Introduction: Pharmacists play a vital role in ensuring the safe and effective use of medication for patients. One critical aspect of their responsibility is detecting and resolving drug-drug interactions. Drug interactions occur when two or more drugs interact in way that alter the effectiveness or safety of one or both medications. These interactions can lead to adverse effects, reduced therapeutic efficacy, or even treatment failure. In this context, pharmacists serve as as a medication expert who possess extensive knowledge about the pharmacology of drug and their potential interactions. Through their expertise, pharmacists actively identify potential drug-drug interaction by reviewing patient medication profile and including prescription medications, over-the-counter drugs, and supplements. Upon detecting potential drug interaction, pharmacists employ various strategies to resolve them. This may involve adjusting doses, altering medications schedules, or recommending altering therapies to mitigate the risk of adverse outcomes. Additionally, pharmacists educate patients about the importance of medication adherence, potential side effects, and the significance of disclosing all medications. The aim of the study to evaluate potential drug-drug interaction in admitted patients. Objective is the main objective. The main objective of the study to find out potential drug-drug interactions with the help of potential hospital prescription and to categorize the identify interaction as to assess its severity. And the second is to intervene a strategy from pharmacist perspective to promote rational and efficacious use of drug in order to improve therapeutic outcomes and quality of life of the patient. And third is to observe routinely drug-drug interaction in elderly patients, and software will be used for the assessment and classify the potential drug-drug interactions. The significance of the study: first is patient safety, second is clinical decision making, third is healthcare resource utilizations, fourth is research and academic contribution, fifth is pharmacovigilance. And the methodology: the study involves collection of information about the medication prescribed to the patient in nephrology, cardiology, and gastro department at Max Super Specialist Hospital in Dehradun, Uttarakhand, India. This study is observational. Is the breakdown: first is data collection. We will gather information based on the set of format designed for this study, and second is study type. It is a prospective observational study. The third is data sources. We will collect prescription detail from the inpatients department at Max Hospital, Dehradun, Uttarakhand, India. And fourth is data entry. The collected data will be entered into a software tool that check for interactions between different drugs. This tool helps identify potential issues when certain drugs are taken together. Fifth is classification of interactions. Sixth is scope of medication review, and seventh is software used. There are two specific software tools will be used. First is Epicurate and second is Medscape. And next is primary and secondary outcomes. Primary outcomes is a incidence. First is incidence of drug-drug interactions. Measure the overall occurrence of drug-drug interaction in admitted patients during this study period. This could be expressed as the number of drug-drug interaction per patient or as a percentage of the total number of prescribed medication. And second is secondary outcomes is: first is severity of drug-drug interactions, second is types of drug-drug interactions, clinical manifestations and commonly implicated drugs, and fifth is patient's characteristics, and sixth is physician awareness and management. Thank you.
Thank you so
Okay,
much.
Vishal, what were the kind of drug interactions you identified during your data collection?
Man, actually study is going on, so study is in process.
So you have only presented your protocol.
Sorry.
Have you only presented your protocol for this conference? There is no result at all. Am I audible, Vishal?
Hello.
Am I
Yes,
audible?
ma'am.
Yes. Have you only presented the protocol for this conference?
Yes, ma'am.
Okay. How do you classify drug-drug interactions?
Ma'am, actually, we use the software. We use softwares for the categorize drug-drug interactions, Medscape and Apocalypse.
Okay. Say if
Yes.
two drugs are interacting, there
Yes,
is an
ma'am.
interaction. What kind of results will your software give you back? It will identify the
Ma'am,
drugs
software identify
interacting. After
and
that, what are the other results?
software identify in different categories like major, moderate, and minor. So according this data, so we can identify the potential drug-drug interactions.
After identifying the interactions, what would be the inference? What you are going to do?
After observe the potential drug-drug interaction, we can discuss with the doctors, and we adjust the treatment and modify the treatment of the patients. Like we reduce the like एक second, 1 second. Like we change the medication and use the alternative drugs also.
Okay, Vishal. Thank you. You can proceed further.
All right, did this one or anything else?
You can move further.
If there's no question,
It is over from my side. We can move further if I don't think ma'am is available. We can move further.
all right, we move to Sudala Srisha. The time is yours.
Yes, ma'am. Afternoon.
You can share your presentation right now. All right. Thank you.
Thank you. Yes, good afternoon to everybody. Myself Siddhartha Sirisha. Today I'm going to give a presentation on the topic of use of box bands and design for optimization of process variables in antiphoretic delivery of naproxen sodium. And here, naproxen sodium, it is one of the NSAIDs we are taking for our study, and also it is an ionizable drug, and we are formulating the first into the transdermal drug delivery. Why? Because so many NSAIDs having the main disadvantage that is they are going to formation or they are producing the gastric ulcers in a stomach. So to that disadvantage can be overcoming by application of the dermal delivery, and we are preparing the transdermal patches. But the main disadvantages for the dermal delivery, that is mainly permeation through the skin. So the skin having the statin carnium, that is a carnious having the upper layer. So whatever the drug that can be crossing this layer, it is a very big question, very big question in the ionizable drug. So for that purpose, we are using the different types of enhancing method, like a chemical enhancing method and as well as the physical and physical system. So many enhancing methods we are following. But in this study, we are going to follow the physical enhancing method, that is antiphoresis. So the antiphoresis is nothing but it is the whatever the ions going to form on the dissolution that can be cross the skin by the application of the current. And we are using here the small amount of current, that is a 0.5 to 1 milliampere per square centimeter of the area. And whatever the current we are applying and whatever the pH of the medium we are using and whatever the polymer concentration we are going to prepare for the transdermal patches, that can be optimized by using the software, that is Buck Benton design, by using the Minitax software. And this is the main objective for the my study, that is we are going to prepare the transdermal delivery to success the permeation across the skin. And this is the antophoresis. And antophoresis nothing but that is the electrical driving of the charged molecules into the teeth will be accomplished by the small direct current, that is approximately we are going to give the 0.5 milliampere per square centimeter through the drug containing electrode in contacting with the skin. And the most popular electrodes are we are using silver chloride electrode. And here the three mechanisms to follow the antophoresis process, that is the electrical repulsion when the charges species are there, when the drug having the negative charge, and also we are giving the negative electrode to that drug solution, there is a repulsion. Why? Because that is a negative repulsion is there, then it is easily repelling. So based on that mechanism, the drug can be reaching into the deep tissue, that is the electrical repulsion. And the electroosmosis. Osmosis nothing but that is only for the uncharged molecules from the high concentration up to the lower concentration. And these are the mechanism, electrical repulsion and electroosmosis. And in some cases that can be transferred through the polarity and as well as the valency and the mobility of the charged species, also one of the mechanisms to follow the antophoresis process. And here, this is a table for the preparation of the transdermal patches by using the different concentration, different amount of the polymer, that is HC and CV15 we are using. This is a hydrophilic polymer, and also we are using the polyethylene glycol, it is a plasticizer to give the plasticity to the patch. And methanol and the dichloromethane, these two are used for the solvent, for the solubility of the drug and as well as the polymer to form into the patch. And the patch can be prepared by the solvent casting method. At the second table, we are having the independent variables. So for the box Benton design, it is a 15 trial. Why box Benton design? For the optimization purpose. It is a trial and error basis. So it is taking the last so much of time and as well as the so much amount of material also we are using. So it is to concise it, to decrease the time and as well as to decrease the amount of polymers, we are using these softwares to directly optimizing the process variables by using the software. So here we are taking the two variables, that is a dependent variable and as well as the independent variable. And here the level also there, that is -1, 0 and +1, that is a low concentration, medium and a high concentration levels. And here the three independent variables, that is we are deciding, that is the current density, medium of the pH and the polymer concentration. And we have a three levels, that is a low, medium and a high level. And similarly dependent variables, whatever we are expecting, that is a Y1, that is a Q4, cumulative amount of drug permeable in the 4 hours, and the Q24, that is a Y2, and that is a cumulative amount of drug permeable in 24 hours, and Y3, that is a log time. So these three dependent variables and the three independent variables, that is a 3 into 3 factorial design. And here it is a total chart we having, that is a 15 trials, like a different levels of the independent variables. So based on that, we are conducting the ex vivo studies, ex vivo permeation studies by using the pig ear skin. And based on that, we are getting the values, that is a Q4, Q24 and the log time. And these are the dependent and independent variables. And here, and before that, the evaluation of preliminary patches, that is a normal patches having we have evaluation parameters like a physicochemical parameters, weight variation, thickness, drug content, and flatness, folding endurance, moisture studies, mechanical studies, all we are conducting for the preliminary purpose. And this is the antophoresis mission we are conducting in our laboratory. It is having the one electrical supply mission, and this is a branch diffusion cell. And this branch diffusion cells having the two compartments, that is a receiver compartment and the donor compartment. In the donor compartment, we are placing our formulation, that is a patch we are using, and the below, that is a receptor compartment containing the medium of the pH, that is a buffer we are placing, and in that we are placing the magnetic field for
Sorry,
the
ma'am.
rotating purpose. Yes.
You have 1 minute remaining.
Okay. And in the donor compartment, we are placing the electrode, and here the naproxen sodium, it is having the negative charge, and also it is having the acid dissociation constant is a 4.15. So based on these, whatever the pH and the pKa we are having, so based on the two values we know the theoretical calculation. Based on the theoretical calculation, that is a Henderson-Hasselbalch equation. Substituting these two values, we are getting the ionized state. How much percentage of ionized state of the naproxen sodium in that medium? That is a 99.94%. So that means the 7.4 pH, according to the theoretical purpose, it is used as a medium. And similarly, we are taking the three levels of the current density and the polymer concentration and as well as the pH of the medium. And this is a counter and the surface plus for the Q4. And this is a Q24 counter plus and the surface flat. And this is a log time and the counter and the surface flat. And these are the factorial responses. So based on the factorial response, whatever the first figure that can be shows, the current density can be increases, the Q4 is also increases. But when the polymer concentration up to certain point, that can be increases, and after that it can be decreases. The same situation here can be seen in a Q24 also. Whatever the current density increases, the Q24 value is also increases. But in a polymer concentration, when the polymer concentration increases, at a certain point, the polymer concentration increases, the
Time
Q24 is also increases.
over,
Yeah.
ma'am.
Yeah, it is about a half minute. It is about to complete it. This is a conclusion. So based on
Alright.
all the values, we are taking the 0.5 milliamperes of the current density and the pH 7.4 and the polymer concentration 1550 mg of polymer can be optimized for the preparation of the transdermal patches. And this is a last exposure studies we are concluding. So compared to without antiphoresis and with antiphoresis, we are getting the 1.34 folds of increase in the drug release. And this is the conclusion. And acknowledgement, we are acknowledged to DST Women Scientists for giving the financial support for the carried out the work. These are the references, and thank you.
All right.
Okay. Transdermal patches for naproxen is available already in the market, right, ma'am?
No, ma'am, actually, diclofenac sodium we have available.
Naproxen sodium is not available.
No, ma'am. Diclofenac sodium is available, but antiphoresis is not applicable till now. So we are introducing the antiphoresis also for the ionizable drugs. Maybe the drug dose is also maybe decreases. In a diclofenac sodium, we have a 75 mg patch is available, ma'am. That is a 10 into 10 patch. 100 square centimeter patch is available.
Okay. Who's the target population for whom you are designing this formulation, ma'am?
I'm not
Target
getting it.
population, what kind of patients?
Yeah, ma'am. Actually, for the naproxen sodium is mainly used for the psoriatic arthritis patients, for the arthritis in a knee joint patient, and as well as the mainly in a cancer patient. In a cancer patients also we are applicable. Why? Because they are having so much of pain. When any operation purpose also and wound healing purpose, any person having the pain at any site, we are giving the transdermal patch.
It is for a localized area in the body, right? Or for pain management for a particular joint or a localized area. Pain management we can do for this formulation. Is it so?
Yeah, ma'am.
Okay, say if it is a rheumatoid arthritis patient or as you said, psoriatic arthritis patients where multiple joints are involved, in that case we have to go for multiple patches and how this works for them. And that is my
Yeah, ma'am.
question.
Yeah, in any place we are just sticking the patches. If the pain is relieved, so at that time we can easily removing the patch. So that means withdrawing of the drug action is possible. That is the main advantage in this transdermal patches. when we are taking the tablet, we are not getting the withdraw the drug action. But when the patch is we are attaching into the skin, that can be easily removed. So that means whatever the low amount of at the low amount of drug, we are receiving the highest drug action.
After applying the patch, how are you going to give the current, ma'am? How is it?
It is a battery system. It is a battery system like a wristwatch. You know the wristwatch. So in upper side we are placing the positive, and the down part of the wrist we are placing the negative. And also the whatever the based on the drug ionization, and which part having the positive, the drug having the positive charge, then whatever the drug solution you can place at the positive charge side, and there is electrical repulsion, and whatever the opposite side that is a negative, that can be attracting the positive drug molecule.
Okay, okay. So far you have conducted animal studies alone. Is it so?
Yeah, ma'am. Yeah, we are already done that one by using the rabbit.
Okay, okay. Okay, ma'am. Fine. Thank you.
Thank you.
Thank you so much. We move on to the next presenter, Hannah Laleng Zuali Fanai. Are you in room?
Yes, ma'am.
All right, you can start your presentation.
Is my screen visible?
Not.
Hello?
It's not visible, ma'am.
Okay.
All right, you can open your PPT.
Is my screen visible?
Yes, it's visible, but your poster is not open yet.
Ma'am, is my screen visible?
No, your poster is not open yet. The screen is still your folder.
Ma'am, is it visible now?
No. Or you can send your PPT to the committee. You have to double click your PPT.
Ma'am, is it visible?
No, it's not visible. It's stuck on your folder.
Okay, ma'am.
Or you can open your poster, then you share screen in Zoom. Not share screen first and then open your poster.
Sorry, Hannah. Can you send your PPT to the committee?
Yes, ma'am. I'm sending.
Ma'am, please stop sharing your screen, ma'am, so that I can share the screen with you. Have you shared, ma'am?
Alright, it's visible now. You can start, and the time is only 10 minutes.
Okay. Good good afternoon to the participants. Today my topic, I'm Hana Lal Engzwali Fanai, and today my topic on is network pharmacology approach and molecular docking prediction to investigate the possible mechanism of benzylidine derivatives against scavenging reactive species via sirtuin-3 in Parkinson disease. So my study aims to hypothesize the role of new molecules as a potent modulators of sirtuin-3 and its co-activating genes in Parkinson's disease. Parkinson's disease is a progressive neurodegenerative disorder characterized by the selective loss of dopaminergic neurons in the substantia nigra pars compacta of the brain, resulting in motor and non-motor symptoms. Although the pathological conditions of the neurodegeneration are vague, the network pharmacology approach was to establish uh, encounter network target multi-component mode, which provide holistic understanding and identification of pharmacological and disease pathological mechanism of the design molecules. Using the databases, the targets genes were collected for the design molecules, and the resulting targets were further re-evaluated for authentication via pieces of literature. The design molecules were further evaluated for drug likeness and probable adverse effect using tools and databases. The gene ontology, including molecular function, biological pathways, and cellular components, and the KEC pathways studies were performed for the collected genes prior which were collected using protein-protein interactions. The computational study involves the probable in silico studies involving the binding affinity of the design molecules with the sirtuin-3 protein, PDB-ID 4VT, the MMG-BSA studies and molecular dynamic studies at 100 nanoseconds were performed to find the stability of the molecule protein stability in the given simulation environment. Targeting the sirtuin-3 and the co-involved genes could modulate the activity of the mitochondria, which is responsible for the balancing of reactive oxygen species and oxidative stress, which results in cellular deaths. So the materials and methods. First, we prepared the ligands and protein genes involved in the PD. So 11 substituted benzylene-based hydroxy derivative named as B1 to BH11, BH1 to BH11 were prepared using the ChemDraw and Maestro leak prep molecules in Schrodinger suite. So, and the synthesized ligands were subsequently converted into usable data with SMILES and other physiochemical attributes. The Swiss target prediction database was searched for probable targets with 70 to 90% molecular similarity. The target genes were obtained from the therapeutic target database, and the gene IDs were obtained from the UniProt database. And the drug likeness and ADMET profiling of the design ligands. The Lipinski rule of 5 method was used to evaluate the drug likeness ratings. The compounds ADMET profiles were determined using ADMET SAR 2.0, and furthermore, the quick prop module in Schrödinger suit was also used to obtain the ADME study for the design compounds and identification of adverse effect. The potential adverse effects were predicted using the design compounds, SMILES, and the advert PrEP database. Analysis was done on probable activity PA and probable inactivity PI values. If the PA value was discovered to be higher than the PI value, the phytoconstituent adverse effects were taken into account while maintaining the value of 0.8. Identification of protein pathway and construction of the network. The string database was used to further analyze the collected genes to determine the network of protein-protein interaction involved in the development of PD. Additionally, the gene ontology, including molecular function, biological pathway, and cellular components, KEC pathway database was used to identify the proteins and regulated pathways associated with PD. Using cytoscape, a network was built between the target gene's pathway and compound. For molecular docking, Schrödinger's suit was used for molecular docking studies. BH1 to BH11 were generated as a three-dimensional structure using the ligand designer Maestro tool. The leak prep module was then used to create the structure with a pH range of 7.4. The compound's energy was reduced using the EPIC module, and the force field employed was OPLS3. The protein data bank provided the human certin 3 bound to ACSES peptide with the PDB ID F4FVT by utilizing the protein preparation workflow tool of the Schrödinger suit. The protein was replenished with the missing hydrogen, water, and amino acid. The grid box was generated using the glide and receptor grid generating modules. Molecular docking was performed with a great precision using the ligand docking module from the Schrödinger suit. Molecular dynamics to verify the stability of the compound BH1, the protein interaction at 100 nanoseconds time intervals. The detailed molecular dynamics simulation study was carried out in the Desmond program of the Schrödinger. The BH1 interacted with the atoms and the protein structural core. It was discovered that BH1 docking score among all the design compound was the highest. And these are the results. So these are the design molecules, BH1 to BH11, and drug likeness and Lipinski rule of 5 were also applied, and the probable adverse effect in which BH1 to BH7 have the shows cardiac failure, nephrotoxicity, hepatotoxicity, and BH8 to BH11 shows myocardial infarction and hepatotoxicity. And these are the network between the molecules, genes, and pathway gene ontology studies and molecular docking studies in which BH1 is compared with resveratrol. And for conclusion, the molecule BH1 has the highest H count with 17 genes, which were modulated via FOXO signaling pathway interaction. The design molecule BH1 was found to have the highest binding affinity of minus 7.463 kilocalorie per mole, whereas the standard molecule resveratrol was found to be minus 5.04 kilocalorie per mole. Certain 3 or BH1 complex displayed the hydrogen interaction uh, of these ligands, and the root mean square division RMSD of the ligand protein complex reach a continuous peak between 20 and 100 nanoseconds. 1,015 biological processes, while the regulation of neuron death was found with the lowest force discovery rate by modulating 19 genes, and 91 molecular functions were obtained with the lowest force discovery rate. The enzyme
Sorry,
binding
ma'am.
pathway. Yes, ma'am.
You have 1 minute remaining.
Okay. The enzyme binding pathway was found to modulate 26 genes, and 61 cellular components were obtained, among which neuron projection was found to modulate 27 genes. And numerous studies have claimed that FOXO signaling pathway in FOXO one as a potent antioxidant, which is involved in the downregulation of the reactive oxygen species, one of the main cause of the Parkinson disease. Our study marks the compound E14-fluorobenzidine benzylidine, 3,4-hydroxymethylbenzene urea BH1 as the potent modulator of the mentioned pathways and genes. On the note, the design molecules can also be used as a Parkinsonian agents. Thank you.
Can you please, Hannah, tell us about your future direction of these studies?
Future studies will be, this is only based on the in silico studies, so no further studies have been done yet, so maybe we would probably go for the in vitro.
Thank you. Good luck. We can
Thank you,
proceed
ma'am.
further.
That's all right. Okay. We go on to the next presenter, Dr. Ramani Mani.
Yes, ma'am.
All right. You have start your presentation now.
Yes, ma'am. My PPT is visible, ma'am?
Yes, it's visible. Let's start.
Thank you, ma'am. Thank you. Good afternoon to one and all. Myself, Dr. M. Ramani, Research Scholar, Department of Siddha, Tamil Nadu Dr. M.G.R. Medical University. My guide, Professor Dr. N. Kabilan, Professor and Head, Department of Siddha, Tamil Nadu Dr. M.G.R. Medical University. Co-guide, Professor Dr. K. Kanakavalli, Principal, Government Siddha Medical College, Arambakam. My topic is effect of Amartadi Churnam on sodium oxalate-induced urolithiasis in Vista rats. Introduction. Urolithiasis at any level of urinary tract is a common disorder as it has worldwide distribution. The worldwide prevalence, incidence, and composition of the calculi varies and have changed in the last several decades. The prevalence of 13% in North America, 9% in Europe, 19.15% in Asia, and 15% in India. Calcium oxalate is the most frequent component of the calculi, 75 to 90%, and followed by uric acid, 5 to 20%, calcium phosphate, 6 to 13%, and struvite, 2 to 15%. The incidence of the urolithiasis reaches its peak in population aged over the 30 years. Males are more likely to suffer from the urinary calculi due to the variations in socioeconomic status, geographic locations, and dietary habits, genetic background, and prevalence among races and nationalities also exist. Genetic mutation, dietary habits, and especially metabolic syndrome may contribute to the formation of different type of calculi. Need of the study, urolithiasis has affected in mankind and can persist with serious medical consequences throughout their lifetime. This may cause obstruction, hydronephrosis, infection, hemorrhage in the urinary tract system. Supersaturation of the urine with a stone-forming constituent is considered to be one of the major reasons for calculogenesis. Many traditional medicines have been used in the treatment of renal stone, but a lack of inadequate scientific evaluation in understanding the mode of action exists till now. So this present study and effort has been made to evaluate sodium oxalate-induced urolithiasis in Siddha polycarbonate formulation, Amirtadi Churnam on Vista albino rats. Aim and objectives to study the effect of sodium oxalate-induced urolithiasis in Siddha polycarbonate formulation, Amirtadi Churnam on Vista albino rats. Materials and methods, preparation of the study drug, standardization of the study drug, toxicity studies and efficacy study. Efficacy study, in vivo evaluation of the sodium oxalate-induced urolithiasis in Siddha polyherbal formulation Amirtadi Churnam on Vistar albino rats. Preparation of the study drug. The ingredients of the Amirtadi Churnam was identified and authenticated by Department of Pharmacognosy at Siddha Central Research Institute, Arambakam, Chennai. Standard operation procedures for Amirtadi Churnam. Amirtadi Churnam is a traditional Siddha polyherbal formulation in Anubhavaiti Deva Ragasyam, page number 684. Author J. Sitaram Prasad, published by Ratnanayakar and Sons. This is the ingredients of the Amirtadi Churnam. This is the authentication certificate issued by SCRI, Chennai, Arambakam. All the drugs purified as per the Siddha literature, Saraka Suddhi Muraigal, and the preparation of single saccharide as per the Siddha literature. These are all the ingredients of Amirtadi Churnam and part use and quantity. Preparation of the study drug Amirtadi Churnam. All the above mentioned ingredients were purified as per the Siddha classical literature and taken into an anhydrous form, pound well and grounded it in iron mortar separately. Steaming process Pitavial Murai. The Churnam was purified as per the Siddha literature, Pitaviyal process. Then added equal quantity of sugar to the above mixture and grinded well. The powder was sieved through a mesh 80 to 100 particle size. The prepared test drug was stored in a clean airtight container. It was labeled as Amritadi Churnam AMC. Vehicle water, 1 gram twice a day dosage, duration 24 days, and indication, Meganir urinary disorders, Pramegam urological disorders, renal stone including Madhumegam diabetes, Vellai, leukorrhea, and Vettai, gonorrhea. Standardization of the study drug. This was done as per the protocol for testing of Ayurveda, Siddha, and Unani medicine by PLA as per the PLM guideline under the Department of Ministry of Ayush, Health and Family Welfare, New Delhi. This study was carried out in Siddha Central Research Institute, Arambakam, Chennai, and Drug Standardization Laboratory, Department of Siddha, Tamil Dr. M.G.R. Medical University, Guinea, Chennai. The toxicity studies were carried out in Arambakam, Chennai 106, Siddha Central Research Institute. This is the IAEC number. Acute and subacute toxicity studies were carried out as per the following guideline, OECD 423 and 407. Evaluation of anti-urolithic activity by sodium oxide-induced urolithiasis. A total number of 48 number of Vista albino rats were received from Tamil Nadu Veterinary and Animal Sciences University, Madhavaram, Chennai. The body weight variation of the rats selected for the study on the day of randomization did not exceed plus or minus 20% of the mean body weight of each sex. Grouping was done based on the body weight and standard pellet laboratory animal diet, RO water, corncob bedding material, polypropylene cages, stainless steel racks, and were used. Animals per cages, 4 animals per cage. Temperature ranges 18 to 25 degrees Celsius. Humidity ranges 30 to 65.
Sorry, ma'am. You have 1 minute left.
Cycle 12 to 12 hours dark and 12 hours night. These are all the methods. Grouping of animals. Group 1 serves as distilled water. Group 2 sodium oxalate 70 mg per kg body weight intraperitoneal distilled water. Group 3 received sodium oxalate 70 mg per kg body weight intraperitoneal plus standard drug cystone 750 mg per kg body weight per oral. Group 4, 5, 6 received sodium oxalate 70 mg per kg body weight intraperitoneal as well as Amritadi Churnam low dose 180 mg per kg body weight and mid dose 900 mg per kg body weight and high dose 1800 mg per kg body weight per oral. The treatment period was 7 days. Rats from each group were placed individually in metabolic cages 24 hours for urine collection. Urine samples was analyzed for creatinine, uric acid, magnesium, oxalate concentration. 7th day blood was withdrawn by retroorbital puncture from each rat. Serum parameters were analyzed, sodium, potassium, creatinine, and uric acid. Statistical analysis was set up. P-value less than 0.05 for all tests. Group 3 to Group 6 was statistically compared with Group 2 and Group 2 was statistically compared with Group 1 to find the treatment-related effects. This is the body weight and biochemistry parameters, blood and urine biochemistry parameters, and urine quantitative parameters, pH parameters, organ weight. Results. The results indicated that the administration of Amritadi Churnam in rats with sodium oxalate-induced
Sorry,
urolithiasis
ma'am. Your time is over.
the growth of urinary stone, restored all the elevated biochemical parameters and restored the urine pH to normal and increased the urine volume significantly when compared to the normal group. This is the histopathological slides. Group 1 rat was found with minimal tubular epithelial cell degeneration. Group 2 showed dilatation of the tubules, mild to moderate multifocal tubular epithelial cell degeneration, necrosis, and presence of numerous tubules showed crystals, whereas Group 3 to Group 5 showed dilatation of the tubules, mild tubular epithelial cell degeneration, presence of crystals in tubules, and Group 6 was found minimal tubular epithelial cell degeneration and presence of crystals in few tubules. Conclusion, the study results show Amritadi Churnam caused diuresis from 180 mg per kg body weight and 1800 mg per kg body weight having an anti-urolithiatic activity against sodium oxalate-induced urolithiasis. The study supports the usage of Amritadi Churnam in urolithiasis. The utility could further be confirmed in other animal models. These are all the references. Thank you for your patient hearing. Thank you, ma'am. Thank you.
Thank you.
Okay, I have only one question. Sodium oxalate and the churnam was administered simultaneously for the animals?
Yes, ma'am. Group 3. Ma'am, sodium oxalate Group 3, sodium oxalate 70 mg per kg body weight intraperitoneal and standard drug cystone 70 mg per kg body weight per oral. Group 4, 5, 6. Group 4 in low dose of amidadi churnam. Group 5 mid dose of amidadi churnam. Group 6 high dose of amidadi churnam.
Okay, ma'am. Okay, thank you.
Thank you very much, ma'am. Thank you, ma'am.
We can proceed, ma'am. Next candidate.
All right, thank you so much. And we move on to Dr. Yash Sharma. You can
Hello.
start to share screen now.
Am I audible?
Yes.
My screen is available. Is it?
No,
Is it?
it's not, sir.
Just a second.
All right, it's visible now. You can start. It's only 10 minutes.
Okay. Thank you so much. My name is Dr. Yash Sharma. I will be talking about my topic, that is the therapeutic potential of Ocimum sanctum, that is the tulsi, the seed cake extracts, in activating the p53 apoptotic pathway against the oral cancer cell lines, or should I say oral squamous cell carcinoma. I'll start with a brief discussion about the topic, that is the oral cancer, which presents a significant global health concern, and its rank among the topmost concerned malignancy worldwide, with stagnant survival rate despite the reach advancement. There are the developing countries like India, Pakistan, and Brazil that bears a disproportionate burden of the oral cancer instances, with prevalence highest in these regions. And natural therapies utilizing the plant-derived products such as flavonoids, phenols has shown promises in cancer treatment, and they have reduced the side effects associated with the traditional treatments. Defatted neems and the Karela seeds like Azadirachta indica and the Momodorica genetia have demonstrated the anti-proliferative properties against the oral cancer cells, highlighting their potential in the previous studies against these cancers, against the oral cancer cell lines. So the main objective of this study was to explore the anti-proliferative effects of the Ocimum sanctum seed cake, that is the oil cake and the fatted seeds, against the oral cancer cell line, which is considered as an agro-industrial waste, to illustrate its role in upregulating the p53 apoptotic pathway in the oral cancer cells. This is the work plan. First of all, we have collected these seeds, then we have prepared the seed cake extract by using the Soxhlet extraction method. Then we have observed a cytotoxic potential activity on the cell lines. And then with respect to our study, we have observed its selective index to check whether they have any therapeutic index or not. And further, we have proceeded with the scratch assay, that is the anti-migratory effect. And after that, we have observed the in vitro antioxidant activity on KB cells. After that, we have observed the anti-angiogenic activity on the chick chorioelenteric membrane model. And furthermore, we have observed that which one has the highest anti-proliferative activity. We have checked the presence of the bioactive compounds in them by using GCMS profiling. And then with respect to the p53 apoptotic pathway, we have observed by using the DNA fragmentation assay and the real-time PCR, that is a apoptotic and anti-apoptotic gene evaluation. These are the results which indicate the cells that were treated with the ethanolic, acetone, and aqueous extract had shown maximum anti-proliferative efficacy in the aqueous extract of seed cakes of osanctum, that is the 227.39 microgram per ml, as well as the standard commercial compound, that is 5-fluorouracil, was also taken in consideration in order to validate the data. The cytotoxic effect were also checked on the fibrillar cell line, that is L19 and L929, to observe their toxicity on this fibrillar cell line, and it was found they have not shown any kind of cytotoxic effect. Anti-migratory effect was observed in order to check the gap filling of the cells in the gaps of the cells, and it was found that aqueous extract has shown lower migration rate at the 24-hour of treatment and then at 48-hour of treatment. With respect to 5-fluorouracil, that has shown the maximum high rate. So this signifies that these extracts have anti-migratory effect. Anti-angiogenic activity was observed in order to check the inhibition of the vasculature pattern and the dissolution of the blood vessels on the chick chorioelectric membrane model, and it was found that with respect to this blue arrow, it is signifying the inhibition of the vasculature pattern and as well as the dissolution of the blood vessels, where you can see the growth of new vessels pattern are there in the control X. So it was found that the eggs that were treated with the aqueous extract had shown maximum anti-angiogenic activity. Afterwards, we have observed the presence of the bioactive compounds in the aqueous extract that has shown the maximum anti-proliferative efficacy, and it was determined that the DSMF has shown the presence of many of the compounds like alkaloid, esters, alkene, triterpene, squalene, azathiothane. In order to confirm the apoptotic activities in p53 gene expressions, first of all, we have done the DNA fragmentation assay with the treated cells with the extracts of the O sanctum. It was found that the extracts that were treated with the ethanol acetone express had shown the laddering pattern which signified the apoptotic pathway. As well as we have already discussed about the selective index, which provide us that whether our drug is safer or not. So this data signifies that higher the SI value, it means the safer the drug. So which signifies that aqueous extract had shown higher SI values. This is the gene expression data, which provide us that the cells that were treated with the ethanol, acetone, and aqueous extract and the standard commercial available drug, it signifies that the treated with the aqueous extract has shown the higher upregulation of p53 gene, whereas caspase 3 and 8 found to be highly upregulated in the cells treated with the acetone extract, as well as the apoptotic and anti-apoptotic genes were also studied, and it was found that the cells that were treated with the aqueous extract had shown high BEX expression, whereas BCL2 found to be low. So in conclusion, O-sanctum seed cake extracts contain the bioactive compounds like ethyl alpha D, glucopanoside, linoleic acid, ethyl ester, scalene, and tetracontin. It exhibits anti-proliferative effects on the KB cells as confirmed by MDT and the SCATch assay. The aqueous extract has shown the safer drug profile based on the selective index evaluation. Seed cake extracts demonstrate the antioxidant activity, particularly noted in the aqueous extract. In the fertilizex, that is the chick chorioelectric membrane model, the aqueous extract has inhibit the blood vessels formation, indicating the anti-angiogenic potential. Polyphenol induce the p53 dependent apoptotic pathway in oral cancer cell lines, as we can see with respect to our data. And in vitro and in vivo study supports that anti-cancer efficacy of these aqueous seed cake extracts, and we can say that they can be a promise herbal remedy for the oral cancer patient. Thank you so much.
So after the study, you're recommending tulsi for treatment of oral cancer?
Yes, generally they are seed cakes, which has been left over after the extraction of the oils. There are many industries that are, extracting the oils, and they have the leftover of the seed cakes. So I'm saying that I know that tulsi has that kind of potential, but the seed cake has also the potential for the oral cancer, and it can be utilized in any form, whether in a tablet or in any conditions.
Could there be any possible adverse effects out of this?
No, we have not seen till now, because we have already observed the efficacies on the cell line, that is on L929, and as well as on the chick chorioelectric membrane model. If they would have any kind of efficacy, we have observed. But till now, there is no data regarding that.
Okay, thank you. We can proceed with the next.
All right, thank you so much. Yes, Shamar, and we moving to Vikas Kumar. Vikas Kumar, are you in the room? Hello, Vikas Kumar. Okay, we move on to the next presenter. Next presenter is Ruby Santra. Ruby Santra is in the room?
Yes, ma'am.
Yes.
Hello.
Can you please screen your presentation, please?
Yes, ma'am.
Okay.
Just a second.
Is my screen visible to everyone?
It's not visible, ma'am. It's any trouble, ma'am? Sorry, ma'am.
Is the next candidate available, ma'am? Please proceed.
All right. We move on to the next presenter. It is Vivek Bakshi. There's Vivek Bakshi in this room.
What about Vikas Kumar Pandey? Yeah. Did you miss that candidate, or is he not available, ma'am?
Yeah, it's not available now. There is no in this room.
Okay.
How about Vivek Bakshi? There's in the room.
I don't think he's available. He isn't.
All right, we move on to Mekanti Manasareka. Mekanti is not also in this room. And then we move on to Gopi Patel.
Yes, ma'am.
All right. Can you please
Yes,
share
ma'am.
your... All right.
Is it visible, ma'am?
Yes, it's visible now.
Okay. Good evening, one and all present over here. Myself, Gopi Patel. My presentation topic is a review on preparation, evolution, and application of bioenzyme. As we talk about the today's scenario that we have using the different chemicals on daily basis, that is we can say as a in household product, that is the shampoo, floor cleaners, cleansing solution. We are consuming the harsh chemicals on daily basis, right? And also we are using a large amount of pill waste that are generated from the different fruits, vegetable industry, and also we are wasting the different peels from in our kitchen product also. So for that, we increase the use of bioenzyme that can be helpful in resolve this problem efficiently, because these bioenzymes are non-toxic, non-hazardous, non-corrosive, eco-friendly also, and completely it is a natural one. As we prepare this bioenzyme in our lab basis also, in home also, and we can convert this lab basis to large scale production also, and easily synthesized by anaerobic fermentation of that just including with the jaggery and different peels from fruits, vegetable, and we can use also a flour from waste materials also. And chemically, this bioenzyme is nothing but a mixture of complex organic substance that just we this say as that contain a protein, carbohydrates, and other metabolites also that occur from the naturally. So this bioenzyme, just example like amylase, protease, lipase, cellulase, different bioenzyme that we can prepare from the different peels of fruits and vegetables, and we can use as a flower petals also. It is a liquid solution that originate from the different peels extract. Now, these are the some material that require for the preparation of bioenzyme, that is the fruit peels, vegetable peels, and one jaggery, that is one jaggery that is mainly it is used for the highly source of carbon that used for the increase the reaction rate also, and we can use some pinch of yeast for increasing the rate of reactions also. Then second one is water, that normal water we can use for the preparation of bioenzyme, but this bioenzyme is require plastic container with screw cap. These are the some preparation of bioenzyme. First step is preparation. Second step is require filtration. And after filtration, we need to characterization by different qualitative analysis. And also we can perform the test of microbial properties, that antimicrobial properties, and also fungal and bacterial activity we can check. Now, how we prepare this bioenzyme, that is just we have to collect the peels of fruits and vegetables, and we need to divide in a smaller piece so that the surface area increase and rate of reactions is also increase. So the ratio of this material, that jaggery peels of fruits and vegetables, that is in ratio of 1:3:10. If we take 30 grams of peels, then jaggery required 10 grams and water is 100 ml, and we put in a airtight plastic container, mix thoroughly, and one optional ingredient, that is yeast, just is required for the increase the rate of reaction, and gas will be produced in the, that is the fermentation process is going on, and we have to use the plastic container. We cannot use the any metal and glass, because gases is evolve, and glass may be broken down, and the material, this bioenzyme is react with the metal. So only we can use the plastic material, airtight container. So the gas is to be released, and a lid of the plastic container was open once in a day for a minute, and lid was closed again. After some days, the gas will be considerably decreased after the 1 month. So there are two parts are divided into these plastic containers. That is, one is the colored liquid and second one is solid residue. After filtration, this solid residue, that is the, we have to dry it, and again with that use for the biocompost, and color liquid, that is nothing but our bioenzyme solution. And these are the some evolution parameter characterization that will be performed for the as a qualitative analysis. Parameters like pH, density, viscosity, total dissolved solid, phenolic compound, is whether it is present, alcohol, alkaloid, carbohydrate, protein, and antimicrobial test, that is like antibacterial test and antifungal test, we can check by after preparation of bioenzyme. One application of this bioenzyme that we can use as a biocompost also, that is if we use 60 gram of raw pills, then total 12.25 grams of that enzymes are that is used for the as a biocompost. Also we can use as a soil stabilization, as per the literature review, the soil stabilization that is highly that is used terpene pills is a good stabilizer, and all bioenzyme that is based on the pH that can be used for the soil maintenance also. Also, it is used for the treatment of contaminated water. that observed in that if we use bioenzyme in water, that we can observe through the pH and TDS evolution parameters, and also the way highly used this bioenzyme in household product, that is natural pesticides, floor cleaner, shampoo, laundry, mold remover. Now, one important note for bioenzyme, that we have to used in a plastic bottle only. We have to keep in a dark place only. And after the fruit peels is at the beginning, fruit peels are float on the top and gradually sink into the bottom. And we have to release the gas after opening the lid once in a day for a minute. After the third week of onwards, open the lid on alternate days. So, and after that, the gas activity will reduce and all pills will come down when it's ready. So based on that, we can confirm that our bioenzyme is ready, and then we'll need to filter out, and solid residue we can use for the biocompost. And these are some key references that are used for the preparation of this review article on preparation application of bioenzymes. Thank you, ma'am.
So, what are the applications of these bioenzymes, ma'am? How are we going to use it? Is it for human use or animal feed use? Or
Yes, these bioenzymes are mainly used in a household product like shampoo, floor cleaners, and it's also used in pharmaceutical application also, but it requires some standardization and purification and isolation process that require if we need to use for pharmaceutical purpose. But we can prepare in household, highly used household product.
Okay, what are the benefits of this?
These benefits are, as we talk about that, we are using the very hazardous chemicals in our floor cleaning, and also this one bioenzyme is eco-friendly one, and anyone can prepare at home also. It's not required just highly educated persons are skill required, but it's requires some basic materials, that is waste material and only jaggery. That's all available in our home also.
Okay, okay. Thank you.
Thank you, ma'am.
Okay, we can go for the next candidate. Thank you.
Okay, thank you so much. We move on to the next presenter, Priya. Are you in this? All right, you can start your presentation now.
Thank you. Is my slide visible?
Yes, visible, ma'am.
Okay, hello all, good afternoon. Today I'm going to present on structure-based design, in silico ADME, synthesis and pharmacological screening. Just a second. Screening of indole analogs as potential candidates for non-small cell lung cancer from College of Principal K.M. Kundnani. Introduction. There are around 2 million cases of lung cancer each year, among which 85% is contributed by non-small cell lung cancer. This type of lung cancer is mainly characterized by malignant cells in tissue of lungs, which is majorly observed that overexpression of epidermal growth factor receptor. Epidermal growth factor receptor, when activated, results in cascade of mechanism leading to cancer cell growth and proliferation, and also other mechanisms such as angiogenesis. There are FDA approved EGFR inhibitors, such as first generation inhibitors as erlotinib, which is used in normal non-small cell lung cancer disease. But then resistance occurs, and second and third generation EGFR inhibitors come into the picture. However, it is observed that EGFR goes on a continuous mutation. This leads to
finding of newer scaffolds in target. So we are going to focus on indole derivatives because it is observed that around 30% or 20% of FDA approved drugs have indole in their scaffold. For example, vincristine, vinblastine, which are indole alkaloid, and osimartinib, which is the third generation EGFR inhibitor itself. So hence, the aim of our project was to study 3 amino alkylated indoles and their action against EGFR for the treatment of non-small cell lung cancer. And the objective was to focus on in silico pharmacokinetic properties of the designed analogs, and then investigation of binding interaction and affinity between the designed inhibitors and EGFR through molecular docking studies. Hence, we were virtually screening our analogs and then synthesizing and characterizing the most promising analogs from the above computational studies, and then also assess their biological activities against human lung cancer A459 cell line. The material and method for the project was computer-aided drug design, which included ligand preparation and designing. We employed 2D sketcher of Schrodinger's Maestro, and then we minimized these ligand using light prep under force field of OPLS 2005. And then we carried out in silico ADME studies using QuickProp version 5.5 and also protein preparation using Schrodinger's Maestro again. And then molecular docking studies was conducted using Gold version 5.2.2. And then after the virtual screening of the 72 analogs, we synthesized the most promising one and characterized them using melting point, IR, and proton NMR. And then to validate the above information, we carried out a biological screening using cell line A549. Our results and discussions were as such that all the molecules who followed the range of quick prop and they also followed Lipinski's rule of 5. And we have presented the favorable representative compounds. And also in molecular docking, we first conducted a validation study on the 2 protein PDB IDs, 6-LUD and 1M17, which were of 3rd generation and 1st generation co-crystallized ligands, osimartinib and erlotinib, respectively. And RMSD was below 1. So we could say
Ma'am,
that.
you only have 1 minute.
Okay, so the RMSD was in range, and similarly the docking results showed that it binded with active site residue similarly to that of the comparative standard. And then in synthesis, we carried out a green reaction using ethylene glycol and ultrasonication at 40 Hertz, which resulted in good amount of yield, and the atom economy was higher. And we characterized them using proton NMR and IR. And in biological activity, we found that IM23 and IM45 were showed a comparable result with gefitinib, which were our standard for comparison, and also a first generation EGFR inhibitor. And in conclusion, we can say that our 9 analogs showed a favorable or promising EGFR inhibitory potential, and 2 of them showed a wonderful result in cell line studies. And my future prospect will be to carry out detailed cell line study on these compound and to have a multi-targeted approach. Thank you. Any questions? Hello. Hello.
Ma'am, you can stop sharing your screen, ma'am.
Okay.
All right, we move on to Dr. Mekanti Manasareka. Are you in this room?
Hello.
Yes, you can start now.
Hello.
Yes, I
Yeah,
hear
can I
you.
start now?
Yes, you can start now, ma'am.
Yeah. Yeah, good evening to all. Myself, Dr. Mekanti Manasreka, Associate Professor in Aditya Bangalore Institute of Pharmacy Education and Research. So today I'm going to present the alkaline water
Sorry,
therapy.
ma'am.
Excuse me?
Yes, the screen is not visible yet. Okay, you can go on. The
Yeah.
screen is not visible.
Yeah. Now, ma'am, is it fine?
Yes, it's fine.
Yeah. So actually, my main core of research is on pH of water, ma'am. So recently we have submitted one proposal also to Indian Council of Medical Research regarding alkaline water therapy. So generally our pH will be in between 7.35 to 7.45 for like isotonic to our blood. But my concept is, so why can't we range our pH like to alkaline so that we can prevent our body getting from metabolic disorders. So today I am going to present one research study. It's a small pilot study which we have conducted on a small group of population. So with this evidence, we supported the documents to ICMR India for the further grant, ma'am. So here the pH is usually we maintained 8.5 to 9. So it's like a alkaline water. So we are categorizing the entire group of the patients to two groups, like test as well as the control. Here the test group is given with all anti-diabetic medications, thyroid medications, as well as hypertensive medications. So these are the common metabolic disorders we can see day to day in our life, correct? So here the two groups, one group is taking the alkaline water, one more group is taking the normal RO water. So rest of the treatments as well as the medications, diet, lifestyle modifications, everything is same. So when we observe the group of population for a over period of time, we have seen like there is an improvement in quality of life of the patient, and there is a reduction in the complications, and the random blood sugar levels as well as HB1AC levels, there will be a drastic decline, ma'am. So with this concept, we can provide like, so why can't we take the alkaline water in day to day our life, like day to day in our life activities, so that we can prevent our body from getting any metabolic disorders. So this study also provided that like a detailed evidence based information regarding how the pH of the alkaline water that we are consuming is affecting our insulin pump. So the pH is the pH what we are providing to the patient, that is almost 8.5 to 9, so which is like alkaline in nature. So it can stimulate the beta cells of our pancreas to produce some more insulin content, ma'am, so that we can provide, so it's like a evidence based support. So even we have applied for the diabetic association also, so World International Diabetic Association. So yesterday I have gone through them like from Berlin. So they have told like, why can't we suggest it like a supportive therapy? So it's like a small pilot study. Here you can see, ma'am, here objectives you can see. Next slide, please. So objectives, next slide, results. Direct to results, please. So regarding the methodology, it is like from June to December 2023. So in results, you can see the symptomatic improvement. Sir, can you please go to next slide, sir? Next slide, please. So here, this is the allergic status of the patient. So you can see the symptomatic improvement in the next slides. Here, regarding the adverse effects, ma'am. So regarding the adverse effects, only one patient he have withdrawn from consuming the water because there is a severe nausea and vomiting and GIT disturbances are there. So apart from that, no adverse drug reactions we have observed. Some small metallic taste as well as excessive salivation we were observed first 2 weeks of intake of alkaline water. Later it got settled down, but one patient got withdrawn from this pilot study. Only 15 patients have given their complete details regarding our study. So you can see that symptomatic assessment and that I have done along with the percentages. So type 1, type 2, hyper and hypothyroidism along with comorbid conditions, we have tested this water. So actually we are planning it in a large scale. So we are planning it over 800 to 1000 people, ma'am, like population. So we have already sent the proposal to ICMR, also Bengaluru, and we are waiting for their reply, ma'am. So I hope this study will give a new concept to the people. So why can't change our pH so that we can get rid of all metabolic disorders? Thank you. Any questions, ma'am?
Any question, ma'am?
Any questions, please? Sorry, I have given a very small time, almost it's like 5 to 10 minutes, so I couldn't able to discuss more. But I am open for the questions, please. So it's a pilot study which we have conducted over 16 patients, in which one got withdrawal and only 15 patients were participated in the present study. So with this evidence, we applied for ICMR grant. So it's like a community trial. So we are implementing in a large scale over 800 to 100 patients. Any questions, ma'am? From anyone, please?
McCanty, ma'am, you can stop sharing a screen because ma'am is not available.
Okay, sir. Okay.
Yes, ma'am.
Thank
Thank
you.
you for your presentation, ma'am.
Thank you, sir. Thank you.
All right, we move on to the next presenter is Lima Labu of. Yes, you can share and you can start your presentation, please.
Yeah, ma'am, is my slide visible? Hello?
Yes, it is visible.
Okay. So good evening all. Myself Lima Lobo from PS University Bangalore. I'm here to present my poster presentation on the title Olanzapine and trihexyphenidyl induced hypertensive heart disease associated with cardiomyopathy. So starting with a brief summary, this case report will presents a concerning scenario of a 31-year-old male patient with schizophrenia who developed severe cardiac complications shortly after initiating the olanzapine and trihexyphenidyl therapy. The patient lacked any history of heart issues, making the rapid decline in his cardiovascular health particularly alarming. So, what were the critical observations that we noted were prior to the olanzapine therapy, the patient's medical history was absent of any cardiac risk factors, suggesting a potentially normal baseline cardiovascular function. And following olanzapine, shortly after starting the treatment, the patient experienced a surge in blood pressure, irregular heart rhythm, and concerning results in tests assessing heart function. So these abnormalities pointed towards cardiomyopathy, a condition characterized by weakening of the heart muscle. So coming to the introduction part, generally antipsychotic medications have remained the primary treatment for schizophrenia, effectively alleviating the psychotic symptoms. However, first generation antipsychotics have significant extrapyramidal side effects, prompting the development of second generation antipsychotic. So olanzapine is a second generation antipsychotic with some efficacy advantages in addressing both the positive as well as negative symptoms, and it has known to exert the favorable impact on the cardiovascular electrophysiological parameters. So why trihexyphenidyl is given? It is given to manage the extrapyramidal side effects of some antipsychotic, but it also has its own risk profile. So this case report examines a rare occurrence of olanzapine and trihexyphenidyl-induced hypertensive heart disease in a patient with no cardiac history. We also explored the clinical presentation, diagnostic challenges, and management of olanzapine and trihexyphenidyl-induced hypertensive heart disease. So what was the objective? The objective is likely to document and analyze the rare occurrence of hypertensive heart disease with associated cardiomyopathy. And the case report underscores the importance of vigilant monitoring during the antipsychotic therapy. Coming to the case description, a 31-year-old male with a 3-year history of deteriorating mental health, he presented with the symptoms of paranoid schizophrenia, and the symptoms were social withdrawal, self-talk, and poor self-care. Alongside, he had vitiligo. So initial assessment revealed that he had a moderate self-care, depressed mood, slow speech, and delayed reaction. Although his mood was low, he didn't exhibit any unusual perception, and basic laboratory tests and vitals showed no abnormalities. So initially, the management proceeded, which involved olanzapine and trihexyphenidyl, intended for his suspected psychosis. However, on the second day, a significant concerns arose, that was Mr. P's blood pressure increased to 230/130 mmHg, and investigations revealed enlarged heart walls and irregular heartbeat and moderately impaired heart pumping function of ejection fraction 50%. Then he was stabilized with amlodipine and monitored closely. His blood pressure fluctuated throughout the following days, and eventually he responded to the medication. So what were the findings that were considered are echo report showed that concentric left ventricular hypertrophy, thickening of the heart's left ventricle walls, and a reduced left ventricular ejection fraction of 50%. Pathology showed that calcification on the posterior mitral wall leaflet annulus. So all these were on the second day after the olanzapine therapy. ECG showed left bundle branch block, and heart rate was high, that showed tachycardia. So olanzapine, coming to the discussion part, olanzapine exhibits lower cardiotoxicity than any other antibiotic. However, olanzapine has been linked to ventricular hypertrophy in animal studies. So evidence suggests that trihexyphenidyl also may exert direct effects on cardiac function, potentially influencing the heart rate, contractility. So this case will align with an iatrogenic etiology, that is of unknown cause. So the patient's lack of pre-existing cardiac risk, symptom resolution after the treatment modification, and presentation of hypertensive heart disease support a causal link to the olanzapine and trihexyphenidyl. So this case underscores the importance of clinicians remaining vigilant regarding the potential cardiac side effects of antipsychotics, particularly when used in combination with drugs like trihexyphenidyl. So coming to the conclusion, in clinical settings, the possibility of olanzapine and trihexyphenidyl-induced cardiac complication should be contemplated when a patient exhibits symptoms such as dyspnea or other indications of heart failure. So in such cases, we have to discontinue the olanzapine and trihexyphenidyl, which is advisable, and standard heart failure management should be initiated as part of routine care. So regular evaluation of cardiac function is necessary for individuals undergoing antipsychotic treatment. Moving forward, interdisciplinary collaboration involving clinicians, pharmacists, and other healthcare providers is essential to optimize the medication therapy and minimize the risk of adverse cardiovascular events in vulnerable patient population. So thank you. That's it about the case report.
All right, thank you. And we move on to the next. Glory Douza.
Thank you.
Thank you so much, Lima. Glory, are you in this room?
Yes, ma'am.
All right, you can start the presentation now.
Yeah. Can you see my screen?
No, ma'am.
Are you able to see my
Alright. Yes, I see your screen now. Let's start now.
Okay. So today I am presenting my poster presentation. First of all, good afternoon, everyone. So my poster presentation is on the topic NT-proBNP in heart failure with preserved ejection fraction. So what is heart failure with preserved ejection fraction? It is nothing but it is a significant challenge in clinical practice where the left ventricular ejection fraction is greater than 50%. It is a clinical condition where the left ventricular ejection fraction is either normal or it is greater than 50%, but it will exhibit signs and symptoms of heart failure due to excessive left ventricular filling pressure. So the pressure wave that is expelled from the left ventricle is reflected back to the heart as arterial stiffness increases. So what happens is the left ventricular load will be increased and hence the stiffness in the wall is seen. So because of that, reduced coronary flow reserve, elevated ventricular diastolic filling pressures, reduced ventricular diastolic function, and pulmonary vascular congestions are the result of these hemodynamic consequences. So another most important consequences that can be seen is breathlessness is a consequence for the patients. Heart failure prognosis assessment, diagnosis, and prediction have all made extensive use of natriuretic peptide. So there are many other cardiac biomarkers that can be used to diagnose heart failure with preserved ejection fraction. But NT-proBNP is playing a main role in this. And recent studies have shown that it is having more effect than other cardiac biomarkers. So increase in the concentrations of NT-proBNP over time have been related with a higher probability of subsequent mortality and morbidity. There is a limited real-world information on the usage of and distribution of NT-proBNP levels in patients with heart failure, especially in those after a worsening heart failure event, that is WHFE. So according to the 2022 American guidelines on heart failure, it is possible that in patients with acute symptoms, NT-proBNP is measured prior to discharge in order to better track the patient's trajectory and create a post-discharge plan. So what are the objectives of my review article is evaluate the effectiveness of NT-proBNP levels in diagnosing heart failure with preserved ejection fraction, considering its sensitivity, specificity, and predictive value compared to other diagnostic criteria and biomarkers. Investigate the prognostic significance of NT-proBNP levels in heart failure with preserved ejection fraction, including its ability to predict adverse outcomes such as hospitalization, cardiovascular events, and mortality. Assess the utility of serial NT-proBNP measurements in monitoring disease progressions and response to therapy in heart failure with preserved ejection fraction patients. Explore the influence of NT-proBNP levels on the treatment decisions and management strategies in heart failure with preserved ejection fraction, including its role in guiding therapy initiation, optimization, and monitoring. So, why NT-proBNP? Natriuretic peptides, including B-type natriuretic peptide and N-terminal pro-B-type natriuretic peptide, are released by ventricular cardiomyocytes in response to increased wall stress and neurohormonal stimulation, serving as a biomarker for myocardial stretch and biomechanical stress. International heart failure guidelines recommend measuring NT-proBNP for various purposes such as prognostic stratification, illness severity assessment, diagnosis, and categorization as it provides enrichment for cardiovascular outcomes in heart failure trials. NT-proBNP is underutilized in clinical settings despite its association with mortality, morbidity, risk in heart failure patients, and age-dependent cutoffs are favored due to the association with age, gender, and renal clearance compared to BNP and MR pro-NP concentrations. NT-pro-BNP demonstrates higher diagnostic accuracy for asymptomatic cardiac dysfunction, especially in older individuals with multiple risk factors for heart failure compared to MR pro-ANP. The inclusions for natriuretic peptide testing is guideline-based heart failure management offers economic benefits and aids cardiologists in identifying high-risk patients, assessing their patient status, optimizing heart failure management, enhancing prognosis assessment, reducing mortality and hospital readmissions, and achieving cost savings per year of life. So what is associations between HFPF and NT-proBNP? First of all, it is used in the diagnosis. So the guidelines, the heart failure guidelines, international heart failure guidelines recommends that natriuretic peptides are a key component in diagnosing heart failure with preserved ejection fraction, with studies showing how they're utilizing it in cardiac dysfunction even when levels are within the normal range. Diagnostic performance, that is, some of the meta-analysis have indicated NPs have a great reasonable diagnostic performance in chronic heart failure with preserved ejection fraction context. Comorbidity influence on NP levels, that is, several comorbidities like COPD, kidney diseases, and diabetes affect the levels of natriuretic peptide, potentially complicating interpretation. So, obesity also may lower the NP levels, while older age is associated with higher levels of NPs. So, in clinical settings or in emergency settings, it is used. Why? Because when the patient comes with a complaint of breathlessness, the clinicians will either go for a test where get confused whether it is because of any asthma or any other condition. So this is a test that is the NT-proBNP levels is like the criteria that is going to distinguish between heart failure with preserved ejection fraction and any other breathlessness symptoms. So breathing not properly study demonstrate the clinical utility of NPs, showing their correlation with severity of heart failure symptoms and their reliability in predicting heart failure diagnosis. So it is also acting as a prognostic value, and it is having some limitations. So prognostic values before the patient gets discharged, again the values of
Sorry, ma'am.
Yes, ma'am.
You have 1 minute left.
Yeah, okay. So they can check the NT-proBNP levels and they can follow up the patient later. So the conclusion drawn in this review article is it plays a critical role in the landscape of HFPF, providing prognostic information, diagnostic clarity, and a useful treatment monitoring tool, as the review concludes. So there are still difficulties and some research that should be done to show that NT-proBNP is much more superior than other tests. Thank you.
Good good afternoon or good evening. Sorry, I'm Prof. Dr. Nala from Malaysia. So the time difference will be 2.5 hours. Okay, thank you for the presentation. I'm just wondering, because your title, you're mentioning about the comprehensive review. So I would like to know how many papers that you have gone through to review this topic.
Ma'am, I have gone through almost 60 papers, 45 papers. So some of the papers are going to tell us that no N-type pro BNP levels are not that much important, because in the clinical settings it is like not of much use. Now they are not using it. So my review states that we have to use it so that the relevance will be more.
Okay, what I mean, if you said that already 45,
Yeah.
what type of paper that you look into, among those 45 papers?
Like what type of studies, ma'am? Like there are some research papers also.
No, I mean, it's like, it's based on the like random, controlled trials, or
Okay.
cohort trials or what?
There are some cohort trials also. One study which was done in China, that they have used this as a biomarker to check whether it is having a significant role in the diagnosis or not. So they have stated that furthermore research has to be done. There are some future prospects also in the study, where they're saying that furthermore research has to be done whether even low levels also, what is the correlation between low level of NT-proBNP with heart failure. So there are some trials, ma'am, currently going on.
All right. Sometimes when we're doing review, there will be some method that we apply. So I'm not sure what type of method that you apply. Just like doing like just a review, or because we have like different type of review. We have the systematic review, we have a scoping review. We also have those review that involve in the meta-analysis.
Yes, ma'am. Mine is a comprehensive review, ma'am.
Okay, all right. Okay, because of the time, so I end up my session of questioning. Thank you.
Thank you.
Okay, thank you so much, Glory. We move on to the next presenter, Sweta Misra. Are you in here? Alright. The voice is not going out, Ahmad. There's no sound at all.
Shweta Mishra was unable to share the video, so I am sharing the screen. Is it visible?
Yeah, it's visible to see the video, but the sound is.
Can I play the video?
Yeah.
Okay.
Rebes. Wait for a minute.
So are we having any videos or not?
Are we waiting for any of the participants to share the screen?
Or we can next move to the next speaker, ma'am.
Yes, please continue with the presentation. If any of the participants is having issue regarding sharing the screen, they can take it at the end.
Alright, we move on to the next presentation. There is Dr. Anand Ramesh. You can start now, sir.
Yes, I can start now.
Thank you.
My study is about clinical radiological evaluation of postmenopausal women of osteoporosis in tertiary care center hospital of Western Maharashtra. I am associate professor in Department of Community Medicine at a VPMC Medical College, Nashik, Maharashtra, India. So the topic covers about the osteoporosis, the problem in women. Menopause, the natural end of women's reproductive years, can usher in roller coaster of health challenges. There are some important problems like hot flashes, sudden sensation of intense heat radiating throughout the body, and night sweats. Beyond these immediate symptoms, menopause can increase risk of developing certain long-term health conditions like decreased density of bone in women, which is also called as osteopenia or osteoporosis. Menopause, however, doesn't have to be time solely of difficult challenges. One of the key concerns of bone health after menopause is osteoporosis. The main key player over here is estrogen hormone. Estrogen plays a vital role in regulating bone health. Rapid bone loss because of decrease in estrogen contributes significantly to osteoporosis. There is increased risk of bone fractures. It is also a silent threat because the symptoms go unnoticeable till the risk actually arrives. Risk factor is thin or bald body flame, lack of exercise, low calcium and vitamin D intake, sedentary lifestyle. But it can be easily prevented by treatment and good exercise. The earlier easiest way to diagnose osteoporosis is DEXA, that is called as dual energy X-ray absorptiometry. It is a gold standard technique. It gives us three results. One is normal, which indicates no osteoporosis. Second is osteopenia, and third is osteoporosis, where bone mineral density is
Hello.
very low.
Um,
Hi. The methodology that we have used is we have focused on postmenopausal women of age 50 and above. We have gotten their DEXA scans done. The DEXA scan gives us a T score, where higher T indicates that bone density is normal. 0 indicates it is as good as a young adult, and lower indicates it is weaker than a normal adult. We have included postmenopausal of age. We are excluded women who were not willing to participate and women with normal bone density. After careful sample size calculation, we arrived at a sample size calculation of 100. Data was gathered and through a pre-tested questionnaire entered into Microsoft Excel, analyzed using softwares like EpiInfo version 7.2.1. Statistical analysis involved utilization of both students t-test and chi-square test. Statistical significance was less at probability of less than 5%. Results, I've summarized the results over here. As we can see, as the age increases, the risk of osteoporosis increases. The trend continues in older women groups with highest percentage of osteoporosis in women is found in over age of 70. The second table that we see here, we have three statistically significant parameters over here. One is the mean age. As the age increases, the chances of osteoporosis are higher. That is statistically significant, as we can see from the table. The body mass index is also significantly associated with osteoporosis, and exercise minimum 20 minutes a day for 5 days a week is also significant. Exercising a day will decrease risk of osteoporosis. I conclude saying that the crucial role of bone health awareness and osteoporosis prevention measures for women going through menopause and beyond. We observed that osteoporosis prevalence of 58% in low bone density cases in early postmenopausal women. Early intervention and consistent monitoring can significantly reduce the risk of fractures and ensure better bone health throughout the later years in these women. Women with osteoporosis tend to be older, have higher BMIs, and engage in less exercise compared to women with osteopenia. Our findings suggest that age, BMI, and exercise habits may be an important factor in bone health. We recommend regular screening for low bone density via DEXA scans, following good lifestyle for all women aged 50 and above to maintain good health and better quality of life. These are references for my study. Thank you for patient listening.
All right, thank you so much, Dr. Anna. So I just proceed with a question.
Yes.
From your data now, I might have missed it. I mean, correct me if I'm wrong. From the participant that you have, did you notice the number of the respondent that in terms of comparing whether they are menopause or not menopause yet?
I did not get your question, ma'am. Please, can you repeat it?
Okay, from the total number of the respondent that you have,
Yes.
how many is actually menopause? How many is not menopause yet?
We have included all menopausal women above 50 years of age, ma'am. Both. Even if the menopause has been not been achieved at 50 years, we have excluded that lady and not considered them as participant.
Okay, so all of the participants are all menopause women.
Menopausal and above 50.
Okay, and that menopausal is like defined clinically by the medical doctors?
Yes, I am myself a medical doctor, postgraduate.
Okay, all right. If you look at the osteopenia, it is slightly different than the pattern of the osteoporosis. What do you think it will be the difference?
By definition, it is different, ma'am. If we see
No,
the bone
no, sorry. I'm not asking about the definition. I'm saying that from the pattern, from the results.
Yes.
I mean, if you look at the table, you can just show the table.
Okay.
Where you put the osteoporosis and the osteopenia.
Okay.
I mean, you can share the screen if you want.
Yes.
Not this one. It's the table.
The previous one. This one?
Yes.
Okay.
Okay, so if based on the number, I mean, we can see that for osteopenia,
Okay.
it will be more in the range of, it's not like more than 70. It
Yes.
is at the younger age for this particular, population.
Okay.
And then for the osteoporosis, I mean, from this, so we can see that, when it's more than 70, that it looks like, they're having higher tendencies to the osteoporosis.
Yes.
So my question now is, what do you think it will be the difference, from the osteopenia and osteoporosis in terms of the age distribution?
We can say that, we can contemplate that, madam, today's people who have osteopenia, if we do not give them any treatment or attention, they will progress to osteoporosis tomorrow.
Okay. In
So
terms
it is
of
better that we start them on treatment of vitamin D and calcium. We improve their diet and also give them some moderate exercises to start with.
Okay, but if we just
Today's people who are normally after pregnancy, we see most of the women do not get an DEXA scan, but most of the women have a lump while walking, or they also have, say, neck pain or lumbar pain or difficulty in walking, or they have knee pain. All these pains they have, but they do not get a DEXA scan done. If they get a DEXA scan done, we can see that their bone density is very low. It is called as an ice cream stick phenomena. If you may say, if you may allow me, if we see an ice cream, if we are eating an ice cream in a hot sun, the ice cream starts to melt because the stick is not able to handle the weight of the ice cream. So similar phenomena will be observed in this female, where the bone skeleton is not ready, where the bone skeleton is not able, the strength is not able to handle the weight of the fat and muscle over it. So we'll have to strengthen the bone and also strengthen the muscle and decrease the fat to maintain good women's health.
Okay, thank you.
Thank you very much.
Okay, we move on to the next presenter, Dr. Amrut.
Thank you so much, ma'am.
Yes, thank you
Let
so
me
much.
share my screen, please. Yes, I hope this is visible.
Yes, it's visible, sir.
Yes, so namaste and good afternoon, everyone. I am Dr. Amrut Arun Swami. I am Associate Professor in the Department of Community Medicine at Dr. S. Tantia Medical College, Sri Gangananda, Rajasthan. I am here to present our research work titled Evaluation of Contraceptive Usage Among Females of Reproductive Age Group in Urban Slum of a City in Western India. So the contraception or family planning, what we will be discussing here, it has got two goals, two basic aims. One is to achieve the desired family size, and second is
for spacing pregnancies effectively. The annual report of Government of India, we expected the increase in the population of 1.21 billion in 2011 to 1.5 billion in 2025. The total fertility rate in the India has reduced from 2.9 in 2005 to 2.3 in 2013. The total fertility rate is nothing but how many number of children a couple will have. Okay, so it is now observed that has reduced from 2.2 in 2015 to two children per woman in 2019 and 21. This 2 children per woman of total fertility rate, it amounts to less than the total fertility rate of 2.1, which is required to maintain the same population. So if the total fertility rate is 2 and less than 2, we can predict that in the coming years, the population will be on a declining trend. The unmet need of contraception is reflected through around 21% of the females, they have the unmet need of family planning according to the district level health survey 3. The early age of marriages play a major part. Still, many states and many corners of India, women less than 18 years of age, they are getting married, and 22% of the females married in adolescence before age of 18. Teenage pregnancy after these underage marriages remains a concern, with 5.6% of deliveries occurring among the teenager. Spacing between births is an often inadequate, with around 60% of the births having less than recommended 3-year gap, and the young adults 15 to 25 years of age group contribute around 52% of the total fertility, highlighting the need of targeted interventions. So these were the most important contributing factors. So we conducted this study and we tried to find out what was the prevalence and method and the health seeking behavior for contraception in our urban slum. This study was conducted in an adapted slum of a city served by the Urban Health Center of a medical college in Western India, focused on married couples where the wife has the age of 15 to 49 years. The couples who were married and living together and women aged 15 to 49 years were included. Couples unwilling to participate and individuals with medical reasons preventing them using any contraceptive methods were excluded. All participants gave written informed consent and we used a pre-tested semi-structured questionnaire. Sample size was calculated using Cochrane's formula with a prevalence of contraceptive uses 67%, and considering the absolute error of 7%, we got the sample size of around 180 to considering 10% dropouts, we rounded up it to 200. The sampling technique that we used was systematic random sampling with a sample size of 200 sample size, and with around 1000 houses in our urban slums with the eligible couples, the sampling interval came to 5. So what we needed to do is we needed to line list all the houses with in our urban slum, and we started from the random house first, and then we tried to find the eligible couple every 5th house in the slum, which was the sampling interval. So this way, from these 1000 houses, we got the 200 required number of samples. The information was gathered through pre-arranged questionnaire entered into Microsoft Excel and analyzed using EPU. The results are: the mean age of the participants was 35 years. Median education status was 12th grade. The table 1 depicts the contraceptive usage and the method used. We observed that around 71% of the participant couples were using or had previously used contraception. It is to note that if the 60% of the eligible couples, they are using the contraceptives, that amounts to the total fertility rate of 2.1. So the 71% usage here that we observed is better than what is expected for maintaining a good population balance. The most popular method was barrier method like condoms, chosen by nearly 40%, followed by oral contraceptive pills, chosen by 27%, and intrauterine contraceptive devices, chosen by copper T, chosen by 20%. We observed that the shampoo sachet marketing strategy also worked for barrier contraceptives like condoms. So many customers, they prefer to buy the 3 condoms which are cheaper for them as compared to buying a total pack of 10 or 20 condoms. The contraceptive services procured from government hospital were more common because of the availability of the urban health center and all the health workers and medical social workers, they were requested to spread awareness regarding
Mister,
availability of
you
contraceptive.
only have 1 minute.
Okay, thank you. The reasons for not using contraceptive, the most common reason was desiring pregnancy, followed by family pressure, lack of awareness, and others. And what we observed, the contraception, there was the participants' age. Those who used contraceptives, they were older in age as compared to those who never used. The significant value of 0.19 was observed. In conclusion, what we conclude is the study found a better rate of contraceptive usage among urban slum women, indicating a positive trend towards family planning within this population. These findings highlight the importance of readily available and accessible contraceptive services. Integrating such services into all government facilities alongside continued promotion of women's health and family planning and further support informed choices and empower women in these communities. Awareness about contraceptives and terminal methods of sterilization is to be given in the communities for better acceptability. These are my references. Thank you for patiently listening. That's all from my side.
Okay, thank you so much, Dr. Amrut. Okay, I'm Prof. Dr. Mahir. Maybe like a suggestion from my side.
Yes,
You know,
ma'am.
I think it's also not applicable to you, but applicable to the other participant as well. I mean, it is good if we have slide with less wordings, you know, rather than put everything, all the words there, because, you know, for us it's a bit difficult to focus when there are too many wordings. And then,
Yes, sure.
yeah, and then can just please simplify the thing. I mean, we can simplify using some pictures, some flow chart and all that will attract people more. Okay. And since of the short of time, so we couldn't really appreciate the remaining findings that you're not able to present today. But I can see
No
that,
problem,
you know,
ma'am.
but I can see that, you know, the pattern on how people, you know, use for contraception. So it started from the very young age of 15 years old, because I
Yes.
can see yours is from 15 and 49,
Yes, ma'am.
Okay. Okay, with all that, I wish you all the best, yeah?
Thank you so much. Thank you, ma'am.
All right, that it's mom. Can we move to the next presenter? All right, we move to the next presenter, Kamini Baswar.
Yes, ma'am. Hello, can you hear me, ma'am?
Yes, I can hear you.
Hello.
Hello?
Yes,
Yes,
can you
I
hear
can hear you.
Okay.
You can start your presentation now.
Yes, ma'am. Just sharing. Ma'am, can you see the presentation?
No, it's not visible yet.
We couldn't see.
Is it visible?
No.
No, it's not visible.
Hello.
No,
Yes,
it's
yes.
not, ma'am.
We can hear you, but it's just that the things that you wanted to share is not pop up on the screen.
Okay.
Hello, Miss Kamini, are you there?
Yes, ma'am, just 2 minutes.
Sorry, Kamini.
Ma'am, just 2 minutes.
All right.
Is it visible, ma'am? Hello?
Still
No, it's
no.
not.
Is it
It's not,
visible?
no, it's not
No,
visible.
you're not even try to share screen, Mom.
Now, ma'am?
Still
No.
nothing. Okay,
No?
something to guide you is, I mean, if you look at the screen, you can see there's a button of share screen, correct? Can you see, can you see the small icon at the bottom? They put the participant, chat, share screen, record. Can you see that one? Okay, so if you can see that one, so you click the share screen.
Yes.
Okay, okay. What are the things that come up from your side? Because from our side, we couldn't even.
Start now and cancel. The two options. Start now.
Okay, so did you click the share screen?
Yes, ma'am. I have already shared from here.
Yeah, but from our side,
Or
there's
can
nothing.
we going to the next presenter that have been prepared? After this, then we come to Kamini. Can we go to Sweta? And after Sweta,
Uh, Kamini will presentation.
Yes, ma'am. Yes, ma'am.
Okay.
Is Sweta videos aware? Hello?
Yes.
Yes.
Sweta doesn't able to share the screen, so I am sharing the screen. Is the videos available? Like, is it visible?
Okay.
Yes,
Yeah.
the video is visible.
Yeah, but the
Can I play
voice.
the video?
Yeah, but the voice of the video.
Okay. Can I play now?
Yes, you can, but we hope this time we can hear the voice.
You can't hear the voice, ma'am?
No, I mean, can you just play it for a while?
Yeah, sure.
No, the video was okay. Okay, can you just play? No voice. From my side, no voice.
No voice.
Yes.
Just a minute, ma'am.
Yes.
Is voice now audible? Is audible now?
No, it's not.
No, it's not audible, huh?
No, we can hear your voice, but not the voice from the video.
Please just give me a minute. I am working on it. Ma'am, is voice is audible now?
From KB Institute. A very good morning, respected delegate.
Okay.
Yes, it's visible now.
Okay, can I play the video?
Yes.
Okay.
I am Shweta Mishra, PhD Research Scholar student from KB Institute of Pharmaceutical Education and Research, Gandhinagar, Gujarat. The topic of my today presentation is entitled as Isolation, Identification and Characterization of Forced Degradation Product of Alkalogluten. Chemical stability of pharmaceutical drug or pharmaceutical molecule is an important parameter as it affects the safety and efficiency of the drug product. In this regard, FDA and ICH emphasize the requirement of stability testing data to understand the quality of drug substance or drug molecule and its change under the influence of various environmental factors. One among the method is force degradation study. Force degradation study is also known as stress testing, in which drug substance or drug product is exposed to different stress conditions like acidic, basic, oxidation, photolytic, and thermal to determine the stability of drug substance or drug product. Force degradation study helps to predict the stability of any active pharmaceutical ingredient or formulation product, to identify the degradation product, to establish the intrinsic stability of the drug molecule, to develop and validate analytical method, to identify impurities related to drug substance or excipient, to identify the impurity developed during the storage of the drug product in various environmental conditions, to generate more stable formulation product, to choose the correct storage condition and packaging of formulation product. In the present study, we aim to isolate, identify, and characterize post-degradation product for a drug named acalabrutinib, which is currently used for the treatment of adult patients with mantle cell lymphoma. The pathway of research work pursued by me till date is presented herewith in the form of flowchart as under. Step 1. Literature review of acalabrutinib drug. Step 2. Procurement of acalabrutinib API and formulation product. Step 3. Preparation of post-degradation solution of acalabrutinib drug. Step 4. Method development and validation on HPLC system. Step 5. Method transfer to preparative HPLC system. Step 6. Purification of post-degradation product of acalabrutinib drug. Step 7. Structure confirmation of post-degradation product of acalabrutinib drug. Now, I would like to elaborate my research work step-wise, which I've already mentioned it previously. Step 1. Literature review of acalabrutinib drug. A literature review is a comprehensive summary of previous research done on acalabrutinib drug till date. Literature review revealed that few analytical methods and bioanalytical method has been reported for the estimation and quantification of acalabrutinib drug in bulk and pharmaceutical dosage form. But a literature review does not reveal isolation, identification, and characterization of forced degradation product of acalabrutinib drug. Therefore, it was thought to identify, isolate, and characterize the forced degradation product of acalabrutinib drug using various spectroscopic techniques. Step 2. Procurement of acalabrutinib API and formulation product. The acalabrutinib API of high quality is procured from pharmaceutical laboratory or pharmaceutical company or any other outside agency in a cost and time efficient manner. The marketed formulation product of acalabrutinib capsule 100 mg is purchased from manufacturer via agent-like distributor or from private or public supplier. Step 3. Preparation of forced degradation solution of acalabrutinib drug. Weigh about 100 mg of acalabrutinib drug and transfer to 100 ml round bottle flask. Add 4 ml of methanol and sonicate to dissolve and add 1 ml of 1 molar HCl or 1 molar NaOH. The mixture was refluxed at 60 degrees Celsius for 12 hours and then cool down the solution at room temperature. Step 4. Method development and validation on HPLC system. The forced degradation solution of acalabrutinib drug is injected into the HPLC system to study the degradation pattern and to calculate how much percent of acalabrutinib drug is degraded in acidic and basic stress condition. The method is developed and validated on the HPLC system as per ICH guideline. Step 5. Method transfer to preparative HPLC system. The developed and validated HPLC method is then transferred to the preparative HPLC system to collect the outlet degradation solution of acalabrutinib drug. In preparative HPLC system, the chromatographic separation was achieved on chromanic C18 column at 35 degrees Celsius with a flow rate of 10 ml per minute and injection volume 1000 microliter. The final mobile phase used was water containing 0.1% formic acid is to methanol in the ratio of 75 is to 25 for separation of acid and base degradation product of acalabrutinib from acalabrutinib drug. The detection was carried out at 230 nanometer. As seen in the chromatogram, the retention time of acid degradation product TP1 of acalabrutinib and acalabrutinib is obtained around 3.26 minutes and 7.29 minutes respectively. Similarly, the retention time of base degradation product TP2 of acalabrutinib and acalabrutinib is obtained around 4.25 minutes and 7.29 minutes respectively. Step 6. Purification of forced degradation product of acalabrutinib drug. Through preparative HPLC system, the outlet acid degradation solution was collected and neutralized and the resultant solution was lyophilized to get crude solid sample of acid degradant product TP1 of acalabrutinib. Similarly, through preparative HPLC system, the outlet base degradation solution was collected and neutralized with 1 molar HCl and the resultant solution was extracted with chloroform. The resultant organic layer was collected and evaporated to get crude solid sample of base degraded product DP2 of alkylabrutinib. Step 7. Structural conformation of forced degradation product of alkylabrutinib drug. The solid residue of acid degradation product DP1 of alkylabrutinib is dissolved in methanol and infused into the mass spectrometer. The mass spectra and proposed fragmentation pathway shows that alkylabrutinib, whose mass to charge ratio is about 466.3 Dalton, on reaction with acid, generate acid degradation product DP1 of alkylabrutinib, whose mass to charge ratio is obtained around 390.2 Dalton, and its two coordinated fragmentation ion is observed, whose mass to charge ratio is obtained around 269.2 Dalton and 136.1 Dalton. Similarly, the solid residue of acid degradation product TP1 of alkylabrutinib is dissolved in DMSO and then it is proceeded with 1H NMR study. The NMR spectra, along with its interpretation, is shown over here. The solid residue of base degradation product TP2 of alkylabrutinib is dissolved in methanol and then infused into the mass spectrometer. The mass spectra and proposed fragmentation pathway shows that alkylabrutinib, whose mass to charge ratio is about 466.3 Dalton, on reaction with base, generate base degradation product TP2 of alkylabrutinib, whose mass to charge ratio is obtained around 442.1 Dalton, and its three protonated fragmentation ion is observed, whose mass to charge ratio is obtained around 330.3 Dalton, 198.3 Dalton, and 95.3 Dalton. Similarly, the solid residue of base degradation product DV2 of alkylabrutinib is dissolved in DMSO and then it is proceeded with 1H NMR study. The NMR spectra, along with its interpretation, is shown over here. Conclusion. From the spectral data results, acid degradation product TP1 of alkylabrutinib was confirmed to have molecular weight of about 390.2 gram per mole, molecular formula of about C21H19N5O3, and have a chemical name which is mentioned in the subtitle. Similarly, the base degradation product TP2 of alkylabrutinib was confirmed to have a molecular weight of about 442.1 gram per mole with molecular formula of C24H23N7O2 and have chemical name which is mentioned in the subtitle. The developed chromatographic method was validated as per ICH guideline and the method proved to be suitable for stability testing and quality control of the drug alkylabrutinib in pharmaceutical industry. This brings me to the end of my presentation. Thank you for sparing your precious time and hearing my research journey to date. Thank you. A very good morning, respected delegate. I am
All right, thank you so much for the presentation, and we will going next to Kamini.
Yes, ma'am.
Kamini, are you here?
Yes, ma'am.
All right. Or if you can be share screen your PPT, you can also give our committee your PPT.
What, ma'am?
You can share your PPT to our committee.
Okay, ma'am. Is it visible, ma'am? Hello?
Hello?
Is it visible?
No, it's not visible.
Is it possible if you just share the slide with the secretariat, so maybe other people can open your slide from others, from their channel?
Uh,
Are you using a mobile handphone or using a computer?
from computer, ma'am.
From computer. Okay, can you see the icon there share screen?
Yes, ma'am. From my side, I have shared actually.
Okay, but still from my side it's just dark only. It's just total black.
Hello. Hello.
Hello, yes. We cannot see anything here. Can you just pass your presentation to the secretariat so
Shall
that
I
they can
shall I shall I mail to the committee, ma'am?
yes, can you just the committee
Okay,
so that
just 1 minute. Just 1 minute. Yes.
Could you please share me the your presentation so that I can share?
यस सर ईमेल आईडी सर वही जो ड्रग डिस्कवरी की है वही वाली आपकी
You can share me in WhatsApp itself, ma'am. I have texted you now.
अच्छा ओके सर व्हाट्सएप कर ओके इज इट 6369 नंबर सर
Yes, ma'am. The same number
yes
where I texted you.
yes okay okay I will send sir
Ma'am, this is your presentation, right, ma'am?
Kamini yes sir
Just one slide is there.
No, sir. I think 21. No, sir.
No, for me it's showing just only one slide, ma'am. Could you please check that?
Wait, sir. One more time I'm sending.
Ma'am, your slide is not.
Just one second. Sir, just check now. Hope you get the full slide. Hello.
Yes, ma'am.
Sir, I have shared the again. I have shared the PPT. Can you just check?
All right. Yes. Wait a minute. All right. You can start now, ma'am.
Ma'am, can okay.
Okay,
Your
can you start
PPT
now?
is also
Yes,
showing
ma'am.
now.
It is
Okay.
showing. Okay, showing. Ma'am, can you just hello?
Yes.
Ma'am, can you just share in that presentation mode, ma'am? Can you
All
just
right.
put the slide in a presentation mode? Yes.
Okay.
Yes, ma'am. Okay. Okay, so good evening, everyone. My name is Kamini Bhavshar. I'm PhD scholar from the Spur Jamia Hamdard, New Delhi. And thank you for giving me this opportunity to present my research work, which is influence of cognitive impairment on quality of life in adult persons with epilepsy, a cross-sectional study. Next slide, sir. Next slide. Yes, apart from me, there are other co-authors. Okay, from the introduction, epilepsy, according to the International League Against Epilepsy, ILAE, epilepsy defined as a brain characterized by any of the following condition. It is a disease which is at least two unprovoked seizure occurring greater than 24 hours apart, one unprovoked seizure, and the probability of further seizure, which is similar to the general reoccurrence, which is at least 60% after the two unprovoked seizure, occurring over next 10 years and a diagnosis of the epilepsy syndrome. So epilepsy affects around the 50 million people worldwide, and it is in a high economic country. There are estimated to be a 49 per 1 lakh people diagnosis with the epilepsy per year, and it is mostly seen in a low to middle income countries, and this figure can be like a high as a 139 per 1 lakh. And epilepsy is a chronic condition with numerous comorbidity like social and the psychosocial consequences, and it has a tremendous impact on quality of life. So the assessment of the quality of life that has been an important indicator for the chronic neurological disorder. Next slide. Okay, so the cognitive impairment of the epilepsy that has an impact on a quality of life. So first we can see it affects the cognitive impairment affect the daily functioning. So daily functioning, the cognitive impairment can affect the memory, attention, problem solving ability, and making it challenging to perform the daily basis. So it increases the dependency and decrease the quality of life. Second, employment and education. So cognitive impairment interfere with the employment opportunity, like the job performance, decrease the poor job performance, and education attainment. Third, social relationship. So cognitive impairment, it interact with the social interaction and the relationship. Uh, uh, and the difficulty with the communication, memory recall, understanding social cues. It may lead to the feeling of isolation and the loneliness and the frustration, which leads to the poor quality of life. Fourth, emotional well-being. So cognitive impairment, it contributes to the emotional distress like anxiety, depression, frustration, and which negatively affect the mood and the overall emotional well-being. Fifth, healthcare management. So cognitive impairment, it also affects the individual's ability to manage with their epilepsy effectively, like difficulty in adhering to the medication schedule and understanding of the treatment plan and recognizing the seizure trigger, and which impact, and that has a negative impact on the overall quality of life. Next slide. Okay, so coming to the methodology, our objective of the study is to assess the influence of cognitive impairment on quality of life in adult persons with epilepsy, and it is an observational cross-sectional study which include the 74 persons with epilepsy, and the study site was AIIMS, New Delhi. Total 74 persons with epilepsy who visited the neurology department of the AIIMS between November 2022 to February 2023. They were included, and the quality of life were measured using quality of life in epilepsy inventory, which is QALY 31 scale, and enrolled patients were evaluated for cognitive impairment by using Montreal Cognitive Assessment Scale, which is MOCA. Next slide. Inclusion criteria were the age were patients with age between 18 to 50 and a diagnosis of epilepsy by a neurologist according to the International League Against Epilepsy, and all type of epilepsy we have included, and the patients with having 5 years of education and understanding of the study consent from, or if not, then a caregiver who could ensure compliance with the study and provide information required for the psychometric assessment, and patients having experienced at least one seizure within 12 months. And the exclusion criteria were the patients with the history of a severe neurological disease other than epilepsy, and the patients with uncontrolled and untreated diabetes and thyroid disease, or a history of a drug or alcohol abuse, and clinically unstable and life-threatening medical conditions, or a surgical disorder within 1 year, and medication history of a psychoactive or the central nervous system depressant drugs, and also the patients with abnormal renal and the liver function, they were excluded from the study. Next slide. So coming to the quality of life epilepsy inventory, quali 31. So we have measured the quality of life through quali 31 scale, which include the 7 multi-item scale, which is emotional well-being, social functioning, energy or fatigue, cognitive functioning, seizure worry, medication effects, and overall quality of life. So first of all, the score of the quali 31, they were converted to the raw data to a 0 to 100 for each subscale, the 7 subscale, and with a higher score, they reflect the higher quality of life, and lower score reflect the lower quality of life. So the total score is calculated as a weight mean of subscale. And next slide. Quali 31, which measure the 7 parameter: emotional well-being, social functioning, energy, cognitive functioning, seizure worry, medication effect, and overall quality of life. Next slide. This is a questionnaire, which include the 31 questions. And the questions are subdivided into the emotional well-being, social functioning, energy, cognitive functioning, seizure worry, medication effect, and overall quality of life. So next slide. This is also next slide. Next, this is a questionnaire which include the 31 question total. And also we have taken permission from the RAND Healthcare. Next slide. So coming to the neuropsychological assessment, so enrolled persons with epilepsy were evaluated for cognitive impairment by using a MOCA, which is Montreal Cognitive Assessment Scale. And why we have used this scale only? Because this scale has a higher sensitivity, reliability, lack of ceiling effect, and good detection of the cognitive heterogeneity in neurological disorder. And cognitive impairment in epileptic patient is often manifested as a memory barrier, falling of executive function, psychomotor speed, naming ability, and visage spatial ability, and so on. Next slide. So the MOCA, which mainly measure the 7 parameter, which is visual, spatial, and executive function, naming function, attention, language, abstraction, delayed recall, and orientation. This 7 parameter, this MOCA scale includes. Next slide. So this is a questionnaire which include the 7 parameters or 7 subscale we can say, which is visual, spatial, executive, naming, memory, attention, language, abstraction, delayed recall, and orientation. And also we have I have taken a certificate training from the MOCA, and this is the certificate of completion. Next slide. So we have calculated p-value using a univariate Mann-Whitney analysis. 74 persons with epilepsy, they were first divided into the cognitively impaired and cognitively unimpaired through MOCA. And from them, we have find out the p-value of the overall quali 31 scale. So we found the overall quali 31 had a significant difference in cognitively impaired and cognitively unimpaired patients. So we can see here with p-value we have found the 0.000, and we can see that in a cognitively impaired group, significant decrease we can see the overall quali 31 score. Next slide. Quali questionnaire reliability test. So quali 31 subscale, we have found the seizure worry, energy fatigue, and had a good internal consistency with a Cronbach alpha value which is 0.7875 and 0.7875 respectively. And the social functioning, overall quali 31. Hello.
Hello.
It is not visible, sir.
Ma'am, just a minute, ma'am. There is still 1 minute left, ma'am, for your presentation.
Yes, sir. There is only two slide remains, sir.
Okay.
Two slide only. Yes. Okay, next slide. Sir, you just share in a slide share mode only. So in a Spearman correlation analysis between a MOCA and the Quali 31, so go to the 18 slide, sir, 18 number slide. Okay, so in a Spearman correlation analysis between a MOCA and 31, we have found that the total MOCA positively correlate with the cognitive function. Next slide. And in a linear regression, a multivariate analysis, we have found that in a cognitive function of the Quali 31, which positively correlate, and it is a positive predictor variable of a total MOCA with a 57% variance. And this data suggests that the patient with the epilepsy with a higher cognitive function in a Quali 31 have a higher MOCA score. So this is a summary of finding. Next slide. Next. So again, yes, the summary of finding, we see that the overall Quali 31 score significantly decreased in the cognitively impaired patients with the persons with epilepsy, and in a Quali 31, a cognitive function was significantly correlated. It is a positive predictor of the total MOCA score. So we can conclude that the cognitive function which influence the quality of life in persons with epilepsy. Next slide. These are the references of my study, and thank you so much. Thank you everyone for the patience. Actually, there were some technical issue with the sharing, but thanks for keeping the patient.
Yes, ma'am. Thank you, ma'am.
Any questions, sir?
Okay, thank you so much, Ms. Kamini.
Yes, sir.
Is the one presented is actually is a part of the other, the part of your big study.
What, ma'am?
The one that you have presented just now, was it a part of your study? Meaning it's
Yes,
like
ma'am.
you may have other objective, it's like your study is quite big, but you only presented just a part of your study.
Ma'am, I didn't got your question. Can you just simplify it? And can you just speak loudly, ma'am?
Okay, the one that you have presented just now, was it
Yes.
just part of your study?
Okay, so you're asking me that is it the only study, or there are other study also in India?
No, I'm asking, your study, the one that you just presented just now, was it a part
Yes,
of
ma'am.
your big study, or your study is only focusing on what you have presented just now?
No, ma'am. My PhD research include there are so many other objective also. This is just one objective which I have present, ma'am.
Okay. I
Yes,
do
ma'am.
expect, yes, I do expected that.
Yes,
I've been
definitely, ma'am. It is just a one. I can say one objective. Apart from this, I have four other objective also. So it is a big project, ma'am.
Yes, yeah. No, I detected that because, from your presentation, when you presented exclusion and inclusion criteria,
Yes,
it's
ma'am.
like for a study that only do a cross-sectional,
Yes, ma'am.
and using the questionnaire that you have checked just now,
Yes, ma'am.
they need that too much strict, on the inclusion and exclusion. So from there, I know this is can be just one part of your study, which is one objective of your study.
Yes, ma'am. You're right, ma'am. Actually, it is just the one part. Apart from the, I have experimental part also, some microRNA biomarkers and other part also actually from the same patients, ma'am. So this is just a questionnaire part which I have presented, ma'am. Apart from the, there are three more objective in same patients, ma'am.
Okay, and then when you're using the linear, this is my last question. Okay, when you're using the linear regression, so you only have
Yes, ma'am.
one independent variable. When you're using the
From this questionnaire, ma'am, there is only one parameter actually, one like a variable, ma'am. I've selected.
Yeah, so
Others I got, ma'am, as statistically insignificant. That's why I have taken only one which is significant from this question only.
yeah, sorry. I mean the other independent variables. You're not tested.
No, I have tested, ma'am, but they were statistically insignificant. Actually, not significant. That's why I have taken only significant one.
Okay, so there's only one, which is the cognitive part, but
Yes, ma'am. Yes, ma'am. From quali only cognitive function actually. That is only statistically significant.
okay. Which of those variable? Other than that, can you just list those that not significant?
Yeah.
I mean,
Yes,
for
ma'am.
the
So they are, ma'am, actually clinical variables and clinical and social demography, which is education, occupation also, cognitive function, which is from the MOCA, actually different one, and from here cognitive function. So total four we found the statistically significant.
Okay, but the one that you presented just now is only one, right?
From this objective only one. Apart from this, there are like three I have told, the education one and other occupation. One second.
Yeah, because I'm just wondering.
Yes, ma'am. There are actually one biomarker also, two biomarkers, and there is only also the education and occupation also. So four other we have found the significant. So actually from quali only there is one. From quali only there is one.
Okay, for the linear regression one, huh?
Yes.
Okay, all right then. Okay, due to the time, I want to ask you more. Okay, thank you so much.
Thank you so much, ma'am, for your patience, and sorry for that technical issues. You have to wait, ma'am.
Yeah, it's okay, Kamini.
You have to wait. Okay, sorry. Okay.
Thank you so much, Kamini, for the last presenters today. And we have to end this event now. Thank you all for joining us today and making this conference a grand success. Hope you had a great session today. I would
Thank you
like
so
to
much,
thank
ma'am.
Yes, thank you so much. I would like
Thank
to
you.
thank all the honorable dignitaries, committee members, and invited speakers who blessed us with their presence, and I am also thankful to all the attendees of this conference. Your presence and engagement have been the cornerstone of this conference's success, and we are looking forward to the future collaboration. Thank you once again for your valuable contribution and for making this conference a memorable one. See you in the next event and good evening, everyone.
Yes,
Okay,
thank you, ma'am.
thank you all.